So good morning everyone. So am I connected to all of you? Yes, so sorry for the delay today. Today we would first start with nephrology and at the end we would complete rest of your rheumatology. Basically the arthritis part, okay? So nephrology. Now kidney is one of the most important organs that we have. And why is it so important? What is the importance of having kidneys? Yes. Why is kidney so important to us? This kidney has so many functions. It is not merely an excretory organ, not just excretion of the waste products, it has n number of other functions like blood pressure regulation, acid base balance, acid base balance. Then, yes, RA and gout we would do. Arthritis we would do in the end. Let me start with nephrology. Okay, arthritis we would do in the end because if I start arthritis right now, nephrology would go haywire. Okay, so we would start with nephrology, and after doing nephrology, we would do rheumatology, arthritis. Okay, yes, Haldwani, fine. So arthritis, gout, RA, all those things we would do at the end. Fine. Yes, so excretion, blood pressure regulation, acid base balance, then the electrolytes and the water balance, then various endocrine functions like it regulates the acid base balance, sorry, the there's renin production, there's vitamin D, the active form of vitamin D which is produced in the kidneys, and then we have hormones like erythropoietin which is synthesized in your kidneys. Okay, so there are various functions in your kidney, not just merely your excretory function. Fine, so kidney is one of the most important organs that we have. Fine, now for doing all these functions there should have some functional unit, and the functional unit of the kidney is your nephron. Okay, the functional unit of the kidney is the nephron. Okay, and how many types of nephron do we have in the kidneys? We have two sets of nephron, and the two sets of nephron are the cortical nephrons and the juxtamedullary nephrons. Fine, so we have two sets of nephrons in our kidney, the cortical and the juxtamedullary. I've started it just now. Okay, so I'm just telling you the functional unit of kidney, which is the nephron, the cortical nephrons and the juxtamedullary nephrons. Fine, now how many parts of the nephron are there? How many parts does your nephron have? The various parts of your nephron are, if this is a rough diagram, okay, if this is a rough diagram, so the parts of your nephron would be Bowman's capsule, then the proximal convoluted tubule, PCT, then the loop of Henle, the descending limb, the hairpin turn, the ascending limb, then the distal convoluted tubule and the collecting ducts and the collecting tubules or the collecting ducts. Okay, so these are the various parts of your nephron. Fine, now I just told you that we have two sets of nephrons, the cortical and the juxtamedullary. Now how are your cortical and the juxtamedullary nephrons different from each other? Cortical and juxtamedullary nephron. Now the most important difference between them is the location of their glomerulus. The parenchyma of the kidney. If this is your kidney, the parenchyma of your kidney is divided into the outer cortex and then is your medulla. Okay, the cortex and the medulla. Now when we talk of the cortical nephrons, their glomerulus lies in the cortex and they have a short loop of Henle. If this is cortex and this is medulla. Fine, so for the cortical nephrons, the glomerulus lies in the cortex, so they are known as cortical nephrons, and they have a very short loop of Henle, okay, which would not go deep down the medulla. On the other hand, when we talk of the juxtamedullary nephrons, their glomerulus lies at the corticomedullary junction, and they have a long loop of Henle which goes down deep into the medulla. Okay, so if this is cortex, this is medulla, then their glomerulus lies at the corticomedullary junction. Yes, all of you. Jabalpur. Okay, I've just started. Okay, so the two sets of nephrons are cortical and the juxtamedullary. The difference is the location of the glomerulus of these nephrons. For the cortical, it is in the cortex and they have a very short loop of Henle. Okay, so they have short loop of Henle, they have a very long loop of Henle. Fine. Now, which types of nephrons are more, cortical or juxtamedullary? Yes, which are more, cortical or juxtamedullary? Yes, which is more in number, cortical nephrons or the juxtamedullary nephrons? Which is more in number? Yes, the cortical. What is the ratio between them? 85 to 90% nephrons are the cortical nephrons. Only 10 to 15% are your juxtamedullary nephrons. Fine. Questions have been asked on this. What percentage of nephrons will have long loop of Henle? So 10 to 15% will have long loop of Henle. 85 to 90% will have a short loop of Henle. Similarly, their blood supply. Since the cortical nephrons are more in number, so their blood supply is more. So they take up 85% of your renal blood flow. Okay, 85% of the renal blood flow goes to the cortical nephrons and only 10 to 15% of the renal blood flow goes to the juxtamedullary nephrons. Okay, so 85 to 90% of your renal blood flow goes to your cortical nephrons. Fine. Now one more difference is there, and that is regarding their capillary networks. In the kidneys, we have two sets of capillary network. One is glomerulus, which is arising from the afferent artery. The other group arises from the efferent artery. Okay, so the afferent artery, afferent gives rise to glomerulus, from which arises your efferent artery, which would then give rise to another set of capillaries, which gives rise to the venous system. The venous system. So this group of capillary from which your venous system arises is different in cortical and the juxtamedullary nephron. This set of capillary in the cortical nephrons is in the form of peritubular capillary network, and the venous system of juxtamedullary nephrons arises from the specialized group of capillaries known as vasa recta. Vasa recta. Okay, so we have peritubular capillary network and vasa recta. Peritubular capillary network and vasa recta. Fine. Yes. Now, why should they have different sets of capillaries? Because the function of these two types of nephrons is different, and that is why we need to have two sets of nephrons. The cortical nephrons, their function is simply filtration and the formation of urine. Okay, the function of cortical nephrons is filtration, whereas the function of juxtamedullary nephron is to produce the hyperosmolarity of interstitium. Interstitium. As we go down the parenchyma of the kidney, from the cortex to the medulla. If this is your cortex, the inside is your medulla. As we go down, the osmolarity increases. In the cortex, the osmolarity is almost same as your blood, roughly around 300. But as we go down deep the medulla, the maximum osmolarity that we have is 1200 milliosmoles. 1200 milliosmoles. Okay, so this osmolarity of the renal parenchyma, which is important for concentration of the urine, is produced by the countercurrent multipliers, which are actually the loop of Henle's of your juxtamedullary nephrons. Okay, I would just repeat this. I hope this much is clear to all of you. All of you have written it down. Yes? Yes or no? These differences are clear to all of you. Yes? Yes, so what is the function of juxtamedullary nephrons? The function of juxtamedullary nephrons is to produce the hyperosmolarity of the interstitium. Okay? as we go down deep into the medulla, okay? This is the cortex. Cortex inside is your medulla. So as we go down deep the medulla, the osmolarity keeps on increasing, and maximum that we have in medulla is around 1200. It starts from 300, which is the same as the blood, and as we go down deep the medulla, it is around 1200. Fine. So this concentration or the hyperosmolarity of interstitium, the parenchyma, is produced by the juxtamedullary nephrons, okay? And the loop of Henle of juxtamedullary nephrons, they are known as counter current multipliers. And this osmolarity, which is produced by the juxtamedullary nephrons, it is maintained by the vasa recta, which are your counter current exchangers, okay? So we have vasa rectas, which are the counter current exchangers, and we have the loop of Henle's of juxtamedullary nephrons, which are the counter current multipliers. So counter current multipliers and counter current exchangers are the loop of Henle's of juxtamedullary nephrons and the vasa recta, which helps in maintaining that, okay? And this is very important because if we would have had a single tubule, straight tubule, okay, the maximum gradient that could have been produced by the most important part of the nephron, which helps in producing the hyperosmolarity, is the ascending limb of loop of Henle, okay, which has a very important transporter, the sodium potassium 2 chloride channel, okay? It is the most important part which is responsible for production of the hyperosmolarity, okay? If we would have had a straight nephron, the maximum difference it could have produced was 200, okay? It can produce only a gradient up to 200. So that was the maximum we could have achieved. But because of this specialized system that we have in the nephrons, the maximum gradient that can be achieved is from 300 to 1200. So this is the maximum gradient that can be achieved, and this is because of the countercurrent multiplier, okay? The details of countercurrent multiplier and exchanges, I think they would be taught to you in physiology, okay? Yes, now how much is the cardiac output? The cardiac output is around 5 liters per minute. Out of this cardiac output, how much goes to the kidney? Out of this 5 liters per minute which is coming, how much goes to the kidney? Yes, out of this 20% goes to the kidney, okay? So out of this cardiac output, 20% constitutes the renal blood flow. So the renal blood flow would be equal to approximately 1 liter per minute. So approximately 1 liter per minute Yes, Agra, I just told you the difference between cortical nephrons and the juxtamedullary nephrons. The function of juxtamedullary nephrons is to produce the hyperosmolarity of the interstitium, and that is why their loop of Henle's are known as countercurrent multipliers. And the vasa recta, the specialized tubes, are there to help in maintaining that hyperosmolarity. So they are known as countercurrent exchanges. So vasa rectas are vasa recta, countercurrent exchanger, and loop of Henle of juxtamedullary nephrons are countercurrent multiplier, okay? So you need to remember only this: vasa recta, countercurrent exchanger, loop of Henle, countercurrent multipliers, okay? Now cardiac output is around 5 liters per minute, out of which 20% goes to the kidney, that is roughly around 1 liter per minute, and again 20% of, sorry, out of this 1 liter, 400 ml is your solute, the cells, and 600 ml is around your plasma. Roughly 600 ml is plasma. Okay, so this 1 liter, 40% would be cells and 60% would be plasma, and only the plasma is what would get filtered. Okay, only the plasma would get filtered. So out of this 600 ml, again 20% is what is going to get filtered. So 120 ml per minute is what is going to get filtered through your glomerulus, and this is what is GFR, glomerular filtration rate. Okay, is this clear to all of you? So out of the 5 liters per minute of your cardiac output, 1 liter goes to the kidney. In that 1 liter, 400 ml is cell and 600 ml is plasma. Only the plasma would get filtered, not the cells. Okay, cells do not filter, it is the liquid part which gets filtered. Fine, so plasma would get filtered, and 20% of this is what is going to get filtered. So 600 ml, 20% of this would be 120 ml per minute. So 120 ml per minute is the filtration which is occurring in your glomerulus. So that is your GFR, glomerular filtration rate. Fine. So 20% from the cardiac output goes to the kidney, and 20% of the plasma would get filtered. So 20-20. And finally, out of this 120 ml per minute, how much would be your urine output? Out of this 120 ml per minute which is being filtered, how much is being excreted as the urine? Out of this 120 ml per minute, how much is being excreted as the urine? Yes? Out of this 120 ml per minute, how much is being excreted as urine? Yes. Out of this 120 ml per minute which is filtered, so how much is being excreted as urine? Yes, only 1 ml per minute. So less than 1%. Roughly 1 ml per minute is your urine output. So you just need to imagine that out of this 120 ml which was being filtered, more than 99% will be reabsorbed. More than 99% will be reabsorbed. So this is very important. So whatever is being filtered, more than 99% would get reabsorbed, okay? So kidney is so efficient, and it has to reabsorb 99% of the stuff which gets filtered. Is this clear to all of you? Okay, and so this reabsorption part is very important, and the most important substance to get reabsorbed in the kidney is your sodium. Most of the other things would be reabsorbed linked to this sodium reabsorption. Okay, so now how many sites of sodium reabsorption do we have? How many sites for sodium reabsorption do we have in the kidney? Yes. How many sites of sodium reabsorption are there in the kidney? Sites of sodium reabsorption in the kidney, yes. There are 4 sites, it's not 3. A little faster, okay. I don't mind being faster, but if I be fast, then some of the students would not understand. Okay, so I need to be slightly slow. Yes, actually we have 4 sites of sodium reabsorption: the PCT, the ascending limb of loop of Henle, DCT, and the collecting tubules. Okay, now what percentage of sodium is reabsorbed at all these sites? What percentage of sodium? At PCT, ascending limb of loop of Henle, DCT, and CT, what percentage of sodium is reabsorbed? The maximum is reabsorbed at PCT, which is around 60 to 65%. Ascending limb of loop of Henle will be around 20 to 25%. DCT is around 10 to 15%, and around 5% is at collecting tubule. So basically we have 4 sites. Okay, 4 sites. Now, ideally, the best site for diuretics to act on should be, what is a diuretic? Diuretic is a drug which would lead to natriuresis. Okay, diuresis is excretion of sodium in the urine, and when sodium would be excreted, it would obviously be accompanied by water. Okay, so that is what is diuresis. So diuresis is always done with the help of natriuresis. So sodium excretion will lead to diuresis. So ideally, the best site for diuresis should be PCT, because 60 to 65% of sodium is being reabsorbed here. But this is not so. The best site for diuretics is the loop of Henle. Why? Why not PCT? Because if PCT is not working, this ascending limb of loop of Henle is very active. Okay? If sodium is not being reabsorbed at PCT, ascending limb of loop of Henle can become more and more active, and it can reabsorb more and more of sodium. Okay? But DCT and collecting tubules are not very good sites. So if ascending limb of loop of Henle is not working, they will not work well. Okay? So the best site for diuretics to act on is the site 2, ascending limb of loop of Henle. So we have these 4 sites. We would discuss now the sodium reabsorption at all these sites. So first we would discuss the proximal convoluted tubule, PCT. Reabsorption of sodium at PCT. Now before that, what all is reabsorbed at PCT? Some other important stuffs which are reabsorbed at PCT along with sodium. So if from the Bauman's capsule, yes, if this is your proximal convoluted tubule. So what all is being reabsorbed here? At PCT there is 60 to 65% reabsorption of sodium, 60 to 65% reabsorption of chloride, and similarly 60 to 65% of your water. What about bicarbs? Another very important stuff which is being reabsorbed here is bicarb. Okay, and what percentage of bicarb is reabsorbed? Around 85 to 90%. So 85 to 90% of bicarb is being reabsorbed at PCT, and then is your amino acid and glucose reabsorption, which is roughly around, which is almost 100%. Okay, so the glucose reabsorption and amino acids, they are completely reabsorbed at the level of PCT. Sodium, chloride, bicarbs, amino acid, glucose, calcium, phosphate, many other things are also reabsorbed, but these are the most important ones. Okay, now before I move on further, if I ask you the water permeability of various parts of the nephron, okay, this was the sodium reabsorption. What about the water permeability? Yes, water permeability. Which parts of the nephron are freely permeable to water? If this is your nephron, this initial part, the PCT, descending limb of loop of Henle, the hairpin turn, they're always freely permeable to water. Okay, now this part, the ascending limb of loop of Henle, the DCT, they're always impermeable to water. Okay, and the last part of DCT and the collecting tubules, the permeability would depend on ADH. If ADH is there, it is permeable to water, otherwise it is impermeable to water. Fine. So PCT, descending limb of loop of Henle, hairpin turn, they are all freely permeable to water. Ascending limb of loop of Henle, DCT, they are always impermeable to water, and the last part, the permeability would depend on ADH. How ADH works, I would tell you later on, but this is something very important. Because of the water reabsorption along with the solutes, the reabsorption at PCT is considered to be isoosmolar. What do I mean by that? The osmolarity of blood is roughly around 300, and by the time the fluid exits PCT, still the osmolarity is 300. It is isoosmolar reabsorption at PCT. Isoosmolar reabsorption at proximal convoluted tubule. Okay, at PCT there is isoosmolar reabsorption. Isoosmolar means osmolarity of the fluid remains same even after reabsorption, and this is because the solutes which are reabsorbed, they are accompanied by same amount of water reabsorption. So since water is also reabsorbed along with the solutes, the osmolarity remains same. So 300 was being filtered, so 300 was what is exiting from your proximal convoluted tubule. Okay, I hope this is clear to all of you. So the osmolarity remains same after the PCT reabsorption. PCT reabsorption. Okay. Now before I move on further to PCT, how does ADH increases the water permeability of this last part? How does ADH increases the water permeability of this last part of the nephron? Yes. So if this is one of these cell in the last part of the nephron, okay. Now ADH is a peptide group of hormone or steroid group. It is a peptide group of hormone. And peptide group of hormones, they always have the receptors where what is the site of the receptors for peptide group of hormones? Yes. Where do they have receptors? The peptide group of hormones. What is the site of receptor for peptide group of hormones? Yes, peptide group of hormones. It is on the cell surface. So similarly, this ADH, which is a peptide group of hormone, will have its receptor. And the receptor that is there in the kidney is the V2 receptor. V2 receptor. It is present on the basolateral surface. This is the basolateral surface of the principal cells. The cells which are present in this last part, they are known as principal cells. Okay. So it is present on the basolateral surface. On basolateral surface. Fine. Now whenever this ADH comes and bind to its receptors, ADH would come and bind to its receptor, what happens? It increases cyclic AMP production within this cell. It increases cyclic AMP production in this cell. And whenever there's increase in cyclic AMP production, what happens is, this is a water channel known as aquaporin 2. Water channel known as aquaporin 2. Now this is translocated onto the luminal surface. Okay. Aquaporin 2 is a water channel. Okay. Aquaporin 2 is a water channel which is normally lying within the cell. But when cyclic AMP production increases because of the ADH, this aquaporin 2 is translocated onto the surface. And in this part of the nephron, we have two more water channels which are aquaporin 3 and 4. Aquaporin 3 and 4. Aquaporin 3 and 4, they are always present on the basolateral surface. They are always present on the basolateral surface, but only when your aquaporin 2 would go to the luminal surface, now both these sides will have water channels so that now they can reabsorb water. Okay. Is this clear to all of you or not? I would just repeat this. I think the diagram is slightly messy. So if this is one of these cell, so this aquaporin 2 is normally lying within the cell, but when there is increase in cyclic AMP production because of the ADH, which is binding to its receptor, there's increase in cyclic AMP production which leads to translocation of aquaporin 2 to the luminal surface, So now this aquaporin 2 would move on to the luminal surface. Okay. And already on the basolateral surface there are two other kinds of aquaporins known as aquaporin 3 and aquaporin 4. They're already present. They are constitutively present. Constitutively means always present on basolateral surface. Okay, they're always present on the basolateral surface. Fine. So now when both the channel surface will have aquaporins, now the cell would become permeable to water. Now they can have water reabsorption from all these channels, and now the cell would become permeable to water. So is this clear to all of you? How ADH increases the water permeability of the last part of the nephron, that is the collecting tubules. Okay? Yes or no? And this part of the nephron is the one which is also responsive to your aldosterone. ADH and aldosterone, they both act on the principal cells. Okay, they both act on the principal cells. Is this clear to all of you? Yes or no? Fine. So should I move forward? Okay. Now after this, Yes, now coming to the reabsorption of sodium. So the first would be the sodium reabsorption at proximal convoluted tubule. Proximal convoluted tubule, okay? So if this is your proximal convoluted tubule, this is one of the cells in your proximal convoluted tubule. Fine. Now all the cells in your kidney, okay, all the cells in the kidney, on their basolateral surface, they always have active sodium potassium ATPases. Active sodium potassium ATPases, which are dependent on ATP and independent of the gradient, they're continuously pumping out 3 sodium in exchange of 2 potassium. They send out 3 sodium in exchange of 2 potassium. This is continuously happening in all these cells. Why? Because we need to have a gradient for sodium reabsorption. Sodium is the most important stuff, and the reabsorption of other things are usually linked with sodium. Fine. So we have to reabsorb sodium, and there should always be a concentration gradient for sodium within the cell. So you must always have reabsorption of sodium from the lumen, and for that gradient to be there, sodium is always being pumped out from the cell into the interstitium by the active sodium potassium ATPases. Okay. Now in this part of the nephron, in the initial part of this PCT, there is absorption of the sodium in exchange of hydrogen. Okay, so sodium is reabsorbed in exchange with hydrogen ion. In exchange with hydrogen ion. Now another important thing which is reabsorbed in this part is the bicarbonates. So this bicarbonate which is coming from above, okay, this combines with this hydrogen ion to form H2CO3. H2CO3. Now this part of the nephron will always also have carbonic anhydrase enzyme. Carbonic anhydrase enzyme which dissociates it into CO2 and water. Carbon dioxide and water. This water is excreted, but this carbon dioxide diffuses into the cell. The carbon dioxide diffuses into the cell, and within this cell it would combine with the intracellular water again to form H2CO3, which is again broken down into H plus and HCO3 negative. H plus is sent out in exchange of sodium, and this HCO3 is sent out into the interstitium in exchange of chloride. Okay, so this is how sodium reabsorption along with bicarb reabsorption is occurring. Is this clear to all of you, or should I repeat this? So sodium is being exchanged with hydrogen. This hydrogen is combining with the bicarb which is coming from above, and combining with the bicarb it would form H2CO3. This H2CO3 is broken down by the carbonic anhydrase into CO2 and water. This water is excreted, but CO2 diffuses back into the cell. Okay. Carbon dioxide diffuses back into the cell, and within the cell it would again combine with the intracellular water to form H2CO3, which would again be broken down by the intracellular carbonic anhydrase into H plus and HCO3. H plus is again secreted out, but HCO3 is reabsorbed. Okay, I would just repeat this again. I would draw this again. Okay. So this was the cell. So sodium is being exchanged. Sodium is reabsorbed in exchange of hydrogen. It is being reabsorbed in exchange of hydrogen. Now this hydrogen combines with the bicarb. Which is coming from above. Okay, this bicarb is also being filtered like your sodium. So this bicarb which is coming from above will combine with H plus ions to form carbonic acid, H2CO3. Now this H2CO3 is broken down into CO2 plus water by the luminal carbonic anhydrase. Okay, luminal carbonic anhydrase. Yes, in PCT we have both luminal and intracellular carbonic anhydrase. Okay, both in the lumen and in the intracellular. Okay, so carbonic anhydrase. Now after this, water is excreted as such in the urine, but this carbon dioxide is reabsorbed. Wherever we have bicarb reabsorption, it is always indirect via the CO2. Okay, the CO2 would enter the cell, and the CO2 within the cell would combine with the intracellular water, and both of them would again form H2CO3. H2CO3. Okay. Now this intracellular H2 carbonic anhydrase would again break down this into H plus and HCO3 negative, and this HCO3 negative is now exchanged or sent out in exchange with chloride. Okay, this bicarb is exchanged from the interstitial side into the interstitium in exchange with chloride. Okay, now this site is very important for bicarb reabsorption. What about hydrogen ion secretion? Hydrogen is being secreted, but whatever is being secreted is being reabsorbed also. Okay, so there is no net H plus secretion, no net H plus secretion here. Okay, though we have bicarb reabsorption which is around 85 to 90%. Okay, in kidney we have two sites for for acid base balance, that is bicarb and H plus ion secretion and reabsorption, that is the PCT and the last part of DCT, or the initial part of collecting tubules where we have the intercalated cells. These are the two places where we have bicarbs and H plus ion secretion, but here there is no net H plus secretion because whatever is being secreted is again being regenerated, so no net H plus secretion, whereas 85 to 90% of the bicarb is being reabsorbed. Okay, fine. Okay, I think now it is clear to all of you. Yes or no? So after this we would move on to the next one, which is the ascending limb of loop of Henle. The next site, which is the ascending limb of loop of Henle. Yes. So if we talk of one cell in the ascending limb of loop of Henle, again even in ascending limb of loop of Henle, on the basolateral surface they will have sodium potassium ATPases which are continuously working to send 3 sodium out in exchange of 2 potassium. So 3 sodium out in exchange of 2 potassium. This is continuously happening in all the tubular cells. Okay. Now how is sodium reabsorbed in this part? In this part they have a transporter known as sodium potassium 2 chloride channel. Sodium potassium 2 chloride channel. So 1 sodium, 1 potassium plus 2 chlorides are reabsorbed in this part. Okay. Now whatever sodium was reabsorbed was being sent into the interstitium. Similarly, for chloride also there is a basolateral channel. Okay, for chlorides also they have a channel with the help of which it is excreted into the interstitium. Okay, they have a basolateral chloride channel, so whatever chloride is reabsorbed, that is also sent into the interstitium. But what about potassium? The potassium is coming in from all these sides. Okay, so potassium is entering this cell from all these sides. So what would happen? This cell would become loaded with potassium. They do not have any potassium channel on the basolateral surface, rather they have a channel on the luminal surface via which there is potassium recycling. Okay, so whatever potassium was reabsorbed from there, it is recycled back. Okay, so since this potassium is being loaded from all these sides and they do not have a channel on the basolateral surface for potassium secretion, so reabsorption. So they recycle potassium via the luminal channel. Okay. Now this potassium is a positive ion. So when this potassium is recycled, it creates a positive charge in this part of the nephron. Okay. The reabsorption in the upper part, the sodium potassium 2 chloride was electroneutral because 2 positive ions, 2 negative ions. But when potassium is recycled, it is not electroneutral. It would rather create a positivity in this part. And because of this positivity, when other positive ions like calcium and magnesium, they reach this part, they are repelled by this. And in the kidneys, we have two kind of reabsorption, either through the cells or via the paracellular route. Paracellular route is in between the cells. Okay. So when this calcium magnesium, they're not reabsorbed via the cell, rather through the paracellular route they are reabsorbed via paracellular route. Okay. Yes or no? Is this clear to all of you, or should I repeat this? This is clear, or should I repeat? The reabsorption in this part is occurring because of the sodium potassium 2 chloride channel. Okay, now potassium is being loaded from all these sides. Sodium and chloride are being sent into the interstitium, but potassium is being sent in from all these sides. So what happens is there is a recycling of potassium, and when there is recycling, it creates a positive charge, and when there's a positive charge, it would repel the positive ions like calcium and magnesium, so they are reabsorbed via the paracellular route. Okay, so the calcium and magnesium are reabsorbed via the paracellular route. Okay, because of this positive charge in this part. Okay, fine, Jammu. So now when we give drugs like furosemide, the loop diuretics, so what would they do? They would block this sodium potassium 2 chloride channel. Okay, so they would block the sodium potassium 2 chloride channel. Now when this channel is blocked, what would happen to this recycling? What would happen to the recycling of potassium? This would also go off, so there would not be any positive charge, so there would not be any calcium magnesium reabsorption. Okay, so when we give furosemide, there is increase in calcium and magnesium excretion. Okay, so furosemide can be used as a treatment for hypercalcemia. Yes or no? Is this concept clear to all of you? So furosemide will decrease the blood levels of calcium as well as magnesium. Rather, it can be used as a treatment for hypercalcemia because it would increase the excretion of calcium magnesium. Why? Because the positive charge in this part of the nephron would go off if there's no potassium recycling. So it would lead to decrease in calcium and magnesium reabsorption. Fine to all of you? Similarly, there are some syndrome. There's a syndrome, genetic syndrome, which resembles the use of furosemide. Yes? Which syndrome resembles the use of furosemide? Anyone? The Bartter syndrome. Okay, the Bartter syndrome is something like your use of furosemide. It is a defect at this part of the nephron. Okay, the most common defect is at sodium potassium 2 chloride channel. We have various types of Bartter syndrome. We have 5 types, and the most common defect is at sodium potassium 2 chloride channel. It is something like giving a furosemide. Fine. Rest we will discuss later. Bartter syndrome. Fine. So after this, the third part is the DCT. So if we take one cell in enlargement in this part, okay, so which channel do we have here? Obviously on the basolateral surface they will have sodium potassium ATPases, which are always pumping out 3 sodium in exchange of 2 potassium. So they're always working. Okay, now the channel for sodium reabsorption in this part is sodium chloride transporter. Sodium chloride transporter, sodium and chloride transporter. Okay, now when this sodium chloride transporter is inhibited, which drugs inhibit this transporter, sodium chloride? Yes, this would be inhibited by which group of diuretics? The thiazide group. The thiazide group of diuretics would act on this part and inhibit the sodium chloride channel. Okay, sodium chloride channel. Now there was decrease in the serum calcium levels when we give furosemide. But what about thiazides? When we give thiazide, there is increase in the serum calcium levels. Thiazide leads to hypercalcemia. Okay, thiazide is a cause of hypercalcemia. Furosemide is a treatment for hypercalcemia. Now, how will thiazide cause hypercalcemia? How does thiazide cause hypercalcemia? Now, when this channel is blocked, so sodium is not entering inside, but it is always being pumped out because of the sodium potassium exchangers. Now, to compensate for this, they have a compensatory mechanism, which is they start now exchanging sodium with calcium ions. Okay, this is sodium calcium exchanger in this part, so they now start exchanging sodium with calcium, and so this cell would now become deficient in calcium, which would increase the calcium reabsorption from the luminal surface. Okay, so they have a calcium channel in this part, so when this channel is blocked, exchange of sodium and calcium increases. When the exchange of sodium calcium is more, so more and more of calcium is sent out, and when more and more of calcium is sent out, so more and more is reabsorbed from the lumen, and from here it would go into the blood vessels via the capillaries. Okay, so via the capillaries from interstitium it would go into the blood. So thiazide is a cause for hypercalcemia, and furosemide is a treatment for hypercalcemia. Okay, is this clear to all of you? Now what about magnesium? Magnesium, when we give thiazide, what happens to magnesium? Yes, thiazide decreases magnesium in the blood. Okay, why? Actually, there's a very small magnesium channel in this part, okay, which is just below the sodium chloride channel. Okay, there's a magnesium channel here in this part, just below the sodium chloride channel. Okay, now when the sodium chloride channel is blocked, this magnesium channel also gets blocked. When sodium chloride channel is blocked, the magnesium channel is also blocked. So the magnesium reabsorption decreases. So both of them will cause decrease in magnesium levels, but calcium levels are different. Hypercalcemia is seen with thiazide, rather furosemide is a treatment for hypercalcemia. Yes or no? Is this now clear to all of you? Okay. Fine. Yes, so now lastly, sodium reabsorption in the last part. Hypercalcemia, thiazide will cause hypercalcemia because when sodium is not reabsorbed from the luminal surface, they have a sodium calcium exchanger on the basolateral surface which becomes active. So now sodium is sent inside the cell in exchange of calcium. So calcium is sent out, sodium is sent in, and when calcium is sent out, finally it would go into the lumen, into the blood because of the capillaries which are present in the interstitium. And when the cell becomes deficient in calcium, so reabsorption of calcium increases from the... No, furosemide does not block the magnesium channel. It decreases magnesium reabsorption because of the inhibition of paracellular transport. Okay, calcium and magnesium in that part were being reabsorbed via the paracellular route because of the positive charge which was being created by potassium recycling. When that is not there, so the reabsorption in the paracellular route is not occurring. So both calcium and magnesium are not reabsorbed and they are rather excreted in the urine. So that was the reason for hypercalcemia, increase in the calcium and magnesium excretion by furosemide. Yes or no, Allahabad? Okay, they do not act on the magnesium channel. They act on the paracellular transport because of the inhibition of potassium recycling. Okay, no positive charge, so no paracellular transport. Allahabad, fine. Now lastly, would be your collecting tubules. Now what are the sodium reabsorbing cells in this part known as? They are known as the principal cells. They are known as the principal cells. Okay, you have principal cells in this part and they reabsorb sodium via the epithelial sodium channels. Epithelial sodium channel. So via the epithelial sodium channel they reabsorb sodium, and whenever sodium is reabsorbed, accompanied by it is water reabsorption. Okay, some amount of water reabsorption is there. Then whenever sodium is reabsorbed, now sodium is a positive ion. So when positive ion is reabsorbed, it creates a electron negativity in this part, and when there's negativity in this part, it would direct the secretion of potassium ion, which is a positive charge. Okay, so when sodium is reabsorbed via the epithelial sodium channels, it leads to electron negativity. Sodium is a positive charge ion, so positive charge is reabsorbed, so there's a negative charge in the lumen which attracts the positive ion, which is potassium. So potassium is secreted in this part. Okay, not only sodium reabsorption, whenever we have electron negativity in this part, it would lead to potassium secretion. It would lead to potassium secretion. It may be because of the bicarb ion or any negative ion. So whenever we have increase in the electron negativity in this part, there is more and more of potassium secretion. Okay, so potassium is not directly being exchanged with sodium. Okay, sodium and potassium are not being exchanged directly. It is because of the electron negativity which is produced, which leads to potassium secretion. Okay, now the sodium channels, they are basically under the influence of aldosterone. So basically in this part, aldosterone as well as ADH, they have a role of ADH I've already told you. So now what about aldosterone? How does aldosterone act on this part of the nephron? Now aldosterone is a steroid group of hormone. So where do we have the receptors for? If this is the nucleus, this is the receptor for ADH. This is the receptor for, aldosterone. So aldosterone would come, this is aldosterone, it would come and bind to its receptors. And they would form homodimers. And this homodimer would then enter the nucleus. And in this nucleus, what do they have? They have the genome or DNA, and the DNA will have a part known as, okay, I will draw it separately. If this was the nucleus, in the nucleus, this is the nucleus. They have the DNA, which has a part known as MRE. MRE is mineralocorticoid response element. Okay, mineralocorticoid response element. Okay, so this homodimer which was formed would come and bind to the promoter region of this promoter region of MRE. Okay. So uh, yes, so in the nucleus there is DNA which has mineralocorticoid response element, the homodimer of aldosterone with its receptors. Okay, they will form a dimer with each other. So that would enter the nucleus, and in the nucleus it would bind to the promoter region, bind to the promoter region of this MRE. Okay, and when it binds to the promoter region of MRE, it starts the transcription of proteins. It increases the transcription, starts the transcription of various genes, and as a result of the transcription there is production of various proteins. And what proteins are synthesized when this part of the genome is transcribed? Two important proteins are synthesized. One is epithelial sodium channel and another is SGK1. So epithelial sodium channel with SGK1 are synthesized whenever aldosterone comes and act on this part. Okay. Now, what is the channels are obvious. Sodium channels are synthesized, that is fine. But what about this SGK1? SGK1. What is this SGK1? SGK1 stands for serum glucocorticoid kinase 1. Okay. So SGK1, serum glucocorticoid kinase 1. Is Jammu actually aldosterone is a steroid group of hormone. So steroids are lipid soluble, so they directly enter the cell. They need not have a receptor on the surface. The receptors are mostly in the cytoplasm, and they combine with the receptors and they finally enter the nucleus and they act on nucleus. The receptors may be in the cytoplasm. Okay, these receptors are not always in the nucleus. The receptors are most of the times in the cytoplasm. They form dimers, homodimers with each other when they bind to the receptor, and then they enter the nucleus. And when they enter the nucleus, they would bind to the promoter region of mineralocorticoid response element. Okay, I would repeat it from the beginning. This aldosterone is a steroid group of hormone. Okay, steroids are lipid soluble, so they can easily diffuse through the cell membrane. So their receptors are always within the cell, either in the cytoplasm or in the nucleus. Most of the times it is in the cytoplasm. So when the aldosterone would enter the cell, it would combine with its receptors. They would form homodimer with each other. One receptor with its ligand, that is aldosterone, another receptor with its ligand, they would form homodimer with each others. Okay, so various receptors are lying, they would form homodimers with each other, and this homodimer would then enter the nucleus. And in the nucleus you have the DNA. In DNA you have various genomes, various parts of the DNA, and one part of the DNA is mineralocorticoid response element, MRE, mineralocorticoid response element. So aldosterone is the mineralocorticoid, so it comes and binds to the promoter region of this MRE, and whenever this promoter region is stimulated, it would lead to transcription of genes. Okay, whenever this DNA is stimulated, it would ultimately lead to transcription of some genes, and as a result of transcription some proteins would be formed. Okay, when transcription is there, so some proteins are formed as a result of it, and the proteins which are formed as a result of aldosterone stimulation are epithelial sodium channel, and the another protein is SGK1. Sodium channels are being formed, that is obvious, because aldosterone, the function is sodium reabsorption. But what about this SGK1, serum glucocorticoid kinase 1? Sodium channels are being formed, that is obvious. But what about this SGK1? Why is there synthesis of SGK1 along with sodium channels? Yes, what is the function of the serum glucocorticoid kinase 1? Actually, the sodium channel, the sodium channel epithelial sodium channel. Now, after its function, after its action, the channel is internalized. by a protein known as is internalized by a protein known as NED4. NED4. Now, NED4 is a protein which internalizes the sodium channels after their function or their action, and then so this internalizes epithelial sodium channel and degrades them by the ubiquitin pathway. Okay, so the sodium channels, they are internalized by NED4 and then they are degraded by the ubiquitin pathway. They are degraded by the ubiquitin pathway. Okay, now whenever we need to have more and more of sodium channel function, we have to inhibit this NED4 so that the channels are not internalized and they remain on the surface. And the SGK1 does this function. So SGK1 inhibits NED4. So the function of SGK1 is to inhibit NED4. So this SGK1 inhibits NED4 which increases cell surface expression of sodium channels. Okay, so because of the inhibition of this NED4, SGK1 increases the surface expression of sodium channels. Okay, by inhibiting NED4, it increases cell surface expression of the sodium channels. Yes or no? So the function of aldosterone is to increase both. Aldosterone would increase both sodium channels and it would also ensure their cell surface expression by the production of SGK1. Okay, yes or no? I hope now this is clear to all of you. Yes or no? Now, what is Liddle syndrome? Have you heard of this? Liddle syndrome? Actually, in Liddle syndrome, there's a defective interaction of epithelial sodium channel with NED4. Okay, so there defective interaction of sodium channels with NED4. So what would happen? The channels are expressed on the surface for a long time. Okay, so even if aldosterone is normal in levels, but the channels which are there, they are not internalized, they are not degraded, they would always remain on the surface and they would keep on having sodium reabsorption. So it would look like as if aldosterone is more. So they would give features which would look like hyperaldosteronism. So it would appear as if there's more of aldosterone, but aldosterone is normal. There's only increase in the cell surface expression of sodium channels. Fine, so Liddle syndrome is because of the defective interaction of sodium channels with NED4. So they are not internalized, they are there on the surface for a long time. They would keep on having sodium reabsorption. Fine, and how do you treat them? Which drug is used for treatment of Liddle syndrome? Treatment of Liddle syndrome is which drug? Yes, amiloride. What is amiloride? Amiloride is a sodium channel blocker. Okay, so by blocking this channel, it would inhibit their function. So that is how it would help in treatment of Liddle's. Yes, triamtrene is also a blocker of sodium channel, but the treatment that is used is amiloride. Okay, triamtrene is not usually used. Amiloride is the one which is used. So amiloride is a treatment for sodium channel blockage and it is the treatment for Liddle syndrome. Okay. So this was Liddle syndrome. What is Gitelman syndrome? So defect at this site, the ascending limb of loop of Henle, was associated with Bartter syndrome, which is of various types. We will discuss slightly later. And here is the Gitelman syndrome. The Gitelman syndrome is defect of the thiazide like defect at thiazide sensitive sodium chloride channel. Okay, it is a defect of thiazide sensitive sodium chloride channel. Okay, so Bartter's is at the site 2 and Gitelman is at site 3. Liddle syndrome was defective interaction of sodium channels and Nephro. Now, so there were 4 sites where you can have your diuretics. Okay, diuretics can act at 4 sites, but whichever diuretic would act proximal to this last site 1, 2 or 3. So the diuretics acting on site 1, 2 and 3, they're all associated with hypokalemia, whereas those acting on site 4, they are potassium sparing. Why? Diuretics acting on site 1, 2 and 3, they are all associated with hypokalemia, whereas those acting on site 4, they are known as potassium sparing diuretics. Why so? Because whenever there is a diuretic acting on site proximal to this site, so what would happen? More and more of sodium would reach this part. When more and more of sodium would reach this part, this principal cell would try to reabsorb more and more of sodium. And when more sodium is reabsorbed, the electron negativity of this part would be more. So more and more of potassium secretion. So because of this more and more of sodium reabsorption which is occurring in this part, it leads to more and more of potassium secretion. So there's hypokalemia in all the diuretics which are acting on site proximal to the last site. Whereas when you block this channel directly, then no sodium reabsorption, no electron negativity here, so no potassium secretion. So drugs acting on this part, site 4, are sodium channel blockers like amiloride, triamtrene, then aldosterone antagonist like spironolactone, aplerenone. Fine. Fine, so is this concept clear to all of you? So all the diuretics acting proximal to this, and examples of diuretics which act on site 1 are, the carbonic anhydrase inhibitors. Like yes, diazoxide and acetazolamide. Okay. Aplerenone is a aldosterone antagonist. We do not use it as a diuretic. It is a aldosterone antagonist. It can cause potassium sparing. Okay. It is not used as a diuretic, but it is a aldosterone antagonist. Okay. Spironolactone, aplerenone. Fine. Aplerenone. Fine. So can I move further? What do you want? Repetition of site 4. Yes. Site 4 is simply sodium reabsorption via the epithelial sodium channels. epithelial sodium channel. Okay, epithelial sodium channel is under the influence of aldosterone. Whenever we have more of aldosterone, so more of channels, so more of sodium reabsorption. Okay, now at site 4, when you block the sodium channel, there's no sodium reabsorption, and if sodium reabsorption is not occurring, so no electron negativity, so no potassium secretion. Okay, so the diuretics which act on site 4, they are said to be potassium sparing, and all the other diuretics which act on proximal sites, site 1, 2 or 3, they're all associated with hypokalemia. Okay, the drugs acting on site 4 are sodium channel blockers like amiloride, triamterene, and aldosterone antagonists like spironolactone and apalerenone. Spironolactone and apalerenone. Fine. Is this clear to you all of? Yeah, clear to all of you? Yes. Can I move further now? Yes. So now, so this was your potassium. This was your potassium. Sorry, sodium reabsorption. The most important thing which is reabsorbed in the kidney. Now, one more important concept in kidney is the acid base balance. The acid base balance. Okay, the acid base balance is happening at two places in your kidney. Okay, in the nephron, we have two sites for acid base balance: the proximal convoluted tubule and this last part of the DCT, or the initial part of collecting tubules. Okay, in this site, it was more importantly this bicarb reabsorption. What percentage of bicarb was being reabsorbed here? 85 to 90%. And here, the last part where you have the intercalated cells, around 10 to 15% of bicarb is reabsorbed. But most importantly, it is the site of H plus secretion. Okay, in site 1, there's no net H plus secretion, but here it is the site for hydrogen ion secretion. So acid secretion is most importantly occurring at the distal site, and bicarb reabsorption is occurring most importantly at proximal site. And based on these two sites, we have two types of renal tubular acidosis. Renal tubular acidosis, which can be type 1, which can be type 2. Okay, we can have renal tubular acidosis type 1, which is the defect at distal site, distal RTA, or you can have type 2, which is the proximal site or the proximal RTA. Okay, so we can have distal and the proximal renal tubular acidosis. Now, what are the intercalated cells? Intercalated cells. Yes, intercalated cells are of two types, type A, type B, or alpha or beta. Some say it alpha or beta. Type A cells are basically acid secreting, and they reabsorb bicarb. Okay, and beta cells are bicarb secreting plus acid reabsorbing. Normally, they are in the type A configuration, or otherwise they say when we have acidosis and we need to secrete acid, it is usually in the type A configuration, and when there is alkalosis, there's more and more of type B configuration, because then we have to secrete the bicarb. Okay, actually this is the same cell, it just rotates and changes its direction so as to become either type A cell or type B cell, and this adaptation of the cell so that it can become type A or type B is dependent on a extra cellular protein known as hensin. Okay, This is the adapter. Okay, extracellular protein known as hensin adapts these cells either into type A or type B. Okay, so when there's more of acid secretion which is needed, they would become type A cell. Normally, also they're usually in type A configuration. Normally, cells are in type A configuration, but when there's more of alkalosis, they would become type B cells, or there's more of acidosis, so more and more of type A cells. Okay, so type A cells, type B cells. So basically, we would talk of type A cells. Type A cells. So what is happening in type A cells? Okay, so if this is one of your cell in this junction of the DCT and the collecting tubule, the intercalated cell type A, okay, type A or alpha type of intercalated cells. Okay, now in this part also there is carbonic anhydrase. Okay, this part of the of the lumen, the kidney, also has carbonic anhydrase. Okay, because of the help of which the CO2 is sent in, and this carbon dioxide combines with water to form H2CO3, which is converted into H plus and HCO3. This H plus is sent out, and this bicarb is sent into the interstitium again in exchange of chloride. Okay, so this part of the nephron, the intercalated cell, will do H plus secretion and bicarb reabsorption. Okay, so bicarb is reabsorbed, H plus is secreted. Water and CO2, they combine to form H2CO3, which is broken down into H plus and H2CO3. H plus is secreted out, and bicarb is reabsorbed with the exchange with chloride. Okay, it is exchanged with chloride. Chloride is sent out, and bicarb is reabsorbed. So this is what is happening in intercalated cell type A. And when this cell would slightly change its direction, then H plus secretion would become on the interstitial side, and bicarb reabsorption or secretion would come into the luminal side. So it would become type B cell. So the same cell, just by changing its direction, it can either become type A or type B. Fine. Now, what is happening in renal tubular acidosis? Renal tubular acidosis. So renal tubular acidosis can be type 1, type, the most important, type 3, 4 I would tell you later on. So type 1, type 2. So what is happening in RTA type 1 and type 2? So basically, renal tubular acidosis, they are a group of disorders where you have normal anion gap systemic acidosis. This type 1 is a defect at the distal site. Distal site means the intercalated cells. And type 2 is a defect of the proximal site, that is your PCT. Okay, PCT. Now distal site is the most important site for H plus secretion. So H plus secretion plus 10 to 15% of bicarb reabsorption. And here it is 85 to 90% of bicarb reabsorption which is occurring. Fine. So now the most important thing is the systemic acidosis, which is normal anion gap. Now why is the systemic acidosis normal anion gap? First we will discuss renal tubular acidosis type 1. So the first point is bicarbonaturia. Bicarbonaturia, the bicarb is not being reabsorbed, it is being excreted in the urine. Normally there is no bicarb in the urine. 85% to 90% is reabsorbed in the PCT, and 10 to 15% is reabsorbed in DCT. So normally there is no bicarb. Previous slide. Don't worry, I'm repeating everything which I told you in the previous slide. Don't worry. Okay. Scroll back. previous slide. Yes, this is the previous slide. So the intercalated cells are there, and renal tubular acidosis. So renal tubular acidosis, most importantly, the two types, the distal and the proximal. So in distal type, there's defect of the intercalated cell. In type 2, there's a defect of the proximal cell, the PCT cells. Okay. And renal tubular acidosis, the most important feature which is common to all of them is normal anion gap systemic acidosis. Normal anion gap systemic acidosis. Yes, so coming to renal tubular acidosis type 1. Okay, renal tubular acidosis type 1. Yes. So bicarbonaturia. So the first thing is bicarbonaturia. Okay. So when this bicarbonaturia, that would lead to acidosis, yes or no? So bicarbonaturia would lead to acidosis, but this acidosis is normal anion gap. Why? What is anion gap? Okay, fine. Anion gap, the formula is sodium minus bicarb plus chlorides. Sodium minus bicarb plus chlorides. Okay. Now what is happening here? This bicarbonaturia, so less of bicarb is there, but what is happening to chloride? The chloride is increasing. Why? Because in the intercalated cells as well as the PCT, this bicarb was being exchanged with chloride. So when bicarb is not reabsorbed, the chloride is not being secreted. Okay, so when bicarb is less, the chloride increases. So the anion gap remains normal. Okay, so in these kinds of acidosis, the anion gap remains normal. Okay, because bicarb is not being secreted, is not being reabsorbed, so chloride is not being secreted. So chloride in the blood increases. That is why they're all known as hyperchloremic acidosis. Okay, so the chloride increases, so this is hyperchloremic acidosis, and that is why the anion gap remains normal. So first is bicarbonaturia, the next point is systemic acidosis, which is normal anion gap. So normal anion gap systemic acidosis, bicarbonaturia. Then third thing is hypokalemia. This is very important. Normally, systemic acidosis is associated with hyperkalemia. Okay, whenever there's acidosis, there's exchange of hydrogen ions with potassium leading to hyperkalemia. Okay, but renal tubular acidosis, they are an exception. Despite systemic acidosis, they are associated with hypokalemia. And why should they have hypokalemia? What is the reason for hypokalemia? The reason for hypokalemia is the bicarbonaturia. Okay, this bicarb is negative ion. So when more and more of negative ion reach your principal cells, so what would happen when more of negative ion is there? So what would happen? It would attract the potassium, and potassium would get secreted in this part. So the potassium secretion is occurring in this part because of the electron negativity. Okay, so in this part of the tubule from the principal cells, you will have the secretion of potassium. So that is the reason for hypokalemia. Okay, is this clear to all of you? Electronegativity. When bicarb, which is a negative ion, so more and more of negative ion reaches this part of the nephron. So negative ions would lead to more and more of potassium secretion. Okay, yes or no? Yes, Masoor. Okay, so negative ions reaching this last part will attract potassium secretion. So all of them are associated with hypokalemia. Okay. Now next point is hypercalcemia and hypercalciuria. Now whenever there is systemic acidosis, there is hypercalciuria. Now why is this there? Whenever there is systemic acidosis, it would lead to increase in the calcium reabsorption from bones. Okay, so whenever we have acidosis, calcium resorption from the bones increases. So there is increase in the calcium levels in the blood. So more and more of calcium is excreted in the urine. Okay, so there's hypercalcemia as well as hypercalciuria in patients with systemic acidosis. So this is also happening in patients with renal tubular acidosis. Now what about citrate levels? There's increase in the citrate reabsorption at PCT whenever there is acidosis. Why? Whenever there is acidosis, the citrate reabsorption at proximal convoluted tubule increases. Why? Because citrate in the body is metabolized into bicarb ions. It produces 3 bicarb ions. Okay. So citrate in the body, when it is metabolized, fine. So when citrate in the body is metabolized, it is converted into bicarb ions. Okay, each citrate would produce 3 bicarbs. Okay, so whenever there is acidosis, to compensate for that, we need to have more and more of bicarb production, and for that there is increase in the citrate reabsorption at the proximal convoluted tubule. Citrate reabsorption is increased at the proximal convoluted tubule. Fine. Now what would happen to the urinary citrate levels? When citrate reabsorption is increased, it would be associated with decrease in the urinary citrate levels. Okay, so citrate in the urine will be less because its reabsorption has increased. Okay, now there's more of calcium in the urine plus less of citrate. When calcium and citrate, they combine with each other, they form water soluble calcium citrate. So calcium is more but citrate is less. So what would happen? This calcium would start combining with other things like phosphate and oxalates, and this would then lead to production of the calcium stones, nephrocalcinosis and nephrolithiasis. So this nephrolithiasis, the calcium stones, is a very important presentation of renal tubular acidosis type 1. Okay, renal tubular acidosis type 1, a very important presentation is the kidney stones. A very common presentation is the kidney stones. Okay, so when there's more of calcium, less of citrate is not there to combine with calcium, it would lead to more and more of calcium combining with phosphate, oxalate, and that would form stones in the kidney. So kidney stones are a very important presentation of renal tubular acidosis type 1. Okay, so is this clear to all of you? Should I move further? Yes. Now lastly, what is the minimum urinary pH? The minimum urinary pH that can be achieved in the patient of renal tubular acidosis even after acid loading, yes, is always more than 5.5. Okay, even after giving acid loading, you cannot bring down the pH to less than 5.5. In normal human beings, what is the limiting pH? The limiting pH or the minimum pH that can be achieved is 4.5. But here, even after giving acid loading, you cannot bring down the pH. Why? Because site of acid secretion is defective. Okay, the site for acid secretion is the intercalated cells, the distal site. So when that is not working, even if you give the patient acid load, it cannot produce the urine acidic. Okay, so even after acid loading, the minimum pH would always remain more than 5.5 because the acid producing site is defective. Okay, fine. So now I will tell you renal tubular acidosis type 2. Renal tubular acidosis type 2. Okay, in renal tubular acidosis type 2, causes we would discuss. Causes of renal tubular acidosis type 1. Okay, causes. The most important cause, very important, is your jogren. Jogren is a very important cause for renal tubular acidosis type 1. Mostly systemic diseases, they affect the proximal tubule, but Jogren's is a condition which affects your last part, that is the distal part. So Jogren's, one important drug is amphotericin B. Amphotericin B is also a very important cause for renal tubular acidosis type 2. Okay. And this is the cause for hypokalemia that we have, which is seen in patient with amphotericin B. Okay, hypokalemia is a very important side effect of giving amphotericin B. Okay, you have to be very careful when you give amphotericin B. No, amphotericin B is a cause for it. Amphotericin B basically affects your distal tubules. Okay, it has more and more of effect on distal tubule. It also causes nephrogenic diabetes insipidus, which is more importantly a cause for renal tubular acidosis type 1. Okay, it affects the distal tubules more. It is associated with nephrogenic diabetes insipidus. Just a second. And other than this, there are some genetic causes. Genetic causes are there. Fine. Which can be autosomal recessive type, autosomal dominant type. The autosomal recessive type would be associated with a mutation of H plus ATPase, the hydrogen pump. And in autosomal dominant type, there's a mutation of the chloride bicarb exchanger. The chloride bicarb exchanger on the basolateral membrane. Yes, aminoglycosides, they also affect the distal part. They are also a cause for both nephrogenic diabetes insipidus as well as your RTA type 1. But amphotericin B is more important. Glycosides can also cause, because they also affect the distal part. Nephrogenic diabetes insipidus is also common to both of them. Nephrogenic diabetes insipidus is also common to both of them. Okay, I would teach you nephrogenic diabetes insipidus, so don't worry about that. Okay. Yes, fine. So what about proximal renal tubular acidosis or RTA type 1? Renal tubular acidosis type 1. Sorry, renal tubular acidosis type 2 or the proximal renal tubular acidosis. So first point here would also be bicarbonaturia. Second would obviously be whenever there's bicarb loss, so they will have systemic acidosis, and again it would be normal anion gap, because when bicarb is less, then chloride increases. So anion gap remains normal. So when this is less, it increases. So again, anion gap remains normal. Third thing is hypokalemia. So these are all same to both of them, type 1 as well as type 2. Now what about hypercalciuria? This is also common to both of them. So calcium excretion is more in both of them. What about citrate? Citrate reabsorption at PCT. This would increase, yes or no? Cirrhosis as a cause of RTA type 1. I'm not sure for that. Cirrhosis, I'm not sure if that is a cause for renal tubular acidosis type 1. I have not read that. Okay. Aminoglycosides is a cause. Amphotericin B is a cause. Sjogren's is a cause. Cirrhosis, I'm not sure. Okay. Yes. Now, what about citrate reabsorption at PCT? It is not increased since proximal convoluted tubule is itself defective. Now when PCT is itself defective, how can you have increase in the citrate reabsorption? So citrate reabsorption does not increase in the proximal convoluted tubule. So now what would happen to the urinary citrate levels? They are high. So increase in the urinary citrate levels. So now we can have citrate combining with the calcium, and when they combine with each other, they form water soluble calcium citrate, which is excreted in the urine. Okay, which is excreted in the urine. So there's no renal stone. So this is something very important. There are no renal stones in a patient with renal tubular acidosis type 2. Okay, in proximal there's no renal stone. Okay. Now what about the minimum urinary pH? The minimum urinary pH after acid loading can be less than 5.5. So this is important. In proximal you can bring down the pH to less than 5.5. नीचे इसमें नीचे स्क्रॉल कर दो इसको. Fine. Fine. Okay, let me just finish this off. So the minimum urinary pH after acid loading here is less than 5.5. Fine. Because the distal site is normal. Normal. Okay. Indanavir, I need to check that. Drugs, I'm not sure. I would just check that. Just give me some 5 more minutes. I would just check and then let you know. Okay. The drugs which are associated with renal tubular acidosis. Okay. Yes. Now, how do you differentiate between RTA type 1 and RTA type 2? RTA type 1 and RTA type 2. In RTA type 1, the first thing is the fractional excretion of bicarb. The fractional excretion of bicarb is only 10 to 15%, is always less than 15% in patients with renal tubular acidosis type 1, and it is always more than 15% in RTA type 2. Okay, so the fractional excretion of bicarb is less than 15% with RTA type 1 and more than 15% with RTA type 2. Then what is Fanconi syndrome? Fanconi syndrome is a generalized defect of the PCT. Generalized defect of PCT. So not only there would be bicarbonaturya, it would also be associated with amino aciduria, phosphaturia, glycosuria, etc. Okay, so this Fanconi syndrome is a feature of renal tubular acidosis type 2. It is not seen in patients with renal tubular acidosis type 1. So Fanconi syndrome is a feature associated with RTA type 2, not in patients with RTA type 1. So Fanconi syndrome is associated with renal tubular acidosis type 2, not with type 1. Then minimum urinary pH. Minimum urinary pH for RTA type 1 is always more than 5.5 even after acid load, and for type 2 you can bring it down to less than 5.5. Dent's disease is a cause of Fanconi syndrome. Okay, Dent's disease is a cause of Fanconi syndrome. There are various causes for Fanconi syndrome. Okay, Hartnup syndrome, cystinosis, Law syndrome, there are various syndromes. So Dent syndrome is one of the causes for Fanconi syndrome. Okay, Dent is a cause for Fanconi. It is a cause for Fanconi syndrome. Then fourth would be renal stones. They are present in type 1, not in type 2. Then lastly, the urinary citrate levels. The urinary citrate levels urinary citrate levels, they are less in type 1, in type 2 they are more. Now what are the causes for RTA type 2? Causes for RTA type 2, it is mostly the minimum urinary pH for type 1 is always more than 5.5 and for type 2 it is less than 5.5. Okay, type 1 the minimum pH is always more than 5.5 and here it is less than 5.5. Okay, yes. So renal tubular acidosis type 2, what are the causes? It is mostly secondary to autoimmune diseases, secondary to autoimmune diseases, some infiltrative diseases like Wilson's, cystinosis, glycogen disorders, storage disorders, some inherited diseases like Dent's syndrome, Law syndrome. Okay, they are X-linked recessive. FeNA we would discuss. Don't worry. FeNA we would discuss when we discuss renal failure. Okay. FeNA is not associated with. We would discuss FeNA when we discuss renal failure. Okay. AKI, pre-renal, post-renal. Okay. Hubly. Yes. What are the various types of your Bartter syndrome? Types of Bartter syndrome. How many types of Bartter syndrome are there? There are 5 variants of Bartter syndrome. Okay. Five variants, type 1, 2, 3, 4 and 5. In type 1 there is a defect of sodium potassium 2 chloride channel. In type 2 there is defect of the apical potassium channel. Just apical potassium channel is defective, via which the recycling was occurring. Okay. In type 3 there is type 3 and type 4 of RT. Don't worry, I will tell you that. Basolateral chloride channels are defective. Type 4 is a defect of the Barton. This Barton is a subunit of chloride channel. Subunit of chloride channel. And type 5 is a defect of the calcium sensing receptor. Is a defect of the calcium sensing receptor. So we have 5 variants of Barters. The most oftenly it is the defect of the sodium potassium 2 chloride channel, which is something like giving them furosemide. Something like giving them furosemide. And which of these forms is associated with sensory neural hearing loss? Sensory neural hearing loss is a feature of type 4 Barter. Type 4 Barter, because this Barton is also associated with some, it is also present in the cochlea. Okay, this Barton, this chloride channel is also present there. So this sensory neural hearing loss is also a feature of type 4 Barters. Type 4 Barters. Okay. Yes. What about renal tubular acidosis type 3 and type 4? What is renal tubular acidosis type 3? RTA type 3 is usually not seen nowadays. Calcium sensitive receptor. Calcium sensitive receptor. Okay, it is a calcium sensing receptor. It senses your calcium. It is present on the basolateral surface of the cells in the ascending limb of loop of Henle. Calcium sensing receptor. Calcium sensing receptor. Okay. Now what is RTA type 3? Renal tubular acidosis type 3. RTA type 3 is not seen nowadays. Okay, it is a mixture of type 1 and type 2. It is a kind of transient condition. It was a transient disease seen for some time, and the cause of this was usually some kind of toxicity. Some toxicities. It is a transient phenomenon. Okay, and usually seen in infants and children. Usually because of the exogenous factors like high salt intake were associated with it. Okay. So in renal tubular acidosis type 3, there's a mixture of type 1, type 2. It is a transient phenomena. It was seen in earlier days in some infants or children because of high exogenous salt intake, but it is not seen nowadays. Not observed nowadays. Okay. Now what is type 4 RTA? Type 4 RTA. This type 4 RTA is totally different from rest of the renal tubular acidosis. The only feature which is common is normal anion gap systemic acidosis. Rest, it is all different. It is associated with hyperkalemia. It is associated with hyperkalemia, which is in contrast with the other kinds of renal tubular acidosis, and it is not actually a tubular disease at all. It is not a tubular defect at all. But it is included in the classification because it is also associated with normal anion gap systemic acidosis. But it has hyperkalemia, and it is said to be because of the defect or decrease in the aldosterone production. Hypoaldosteronism is thought to be the cause of this renal tubular acidosis type 4. So because of less of aldosterone production, there is renal tubular acidosis type 4 associated with normal anion gap systemic acidosis and hyperkalemia. And this systemic acidosis here is because of the impaired ammonia genesis. It is because of the impaired ammonia production. Ammonia production. Okay. In the PCT. Okay. The acidosis here is because of the impaired ammonia production, and otherwise this is totally different from the rest of the tubular acidosis. It is not at all a defect of the tubules. It is just because of the decrease in aldosterone. Okay, hypoaldosteronism is thought to be the cause of renal tubular acidosis type 4. Okay, now I guess we would take a break for 5 minutes or 10 minutes. Okay, so 10 minutes of break now. You want the lunch break right now? Yes or no? How many centers want lunch break right now? No? Lunch break we would have after the next break. Okay, around 2 o'clock we would have the lunch break. Okay, now 10 minutes of break only. Fine? 10 minutes of break. First slide दिखा दो, first slide. Yes, so is everyone there? So I would just summarize the causes of renal tubular acidosis type 1 and type 2. Renal tubular acidosis type 1 and type 2. For RTA type 1, it is conditions like Sjogren's, then nephrocalcinosis, medullary sponge kidney, cirrhosis, sickle cell anemia, cirrhosis, sickle cell anemia, then drugs like amphotericin B, ifosfamide, lithium, aminoglycoside maybe. Yes, so all of you have written it down, yes or no? Causes for renal tubular acidosis type 1. Is everyone there? Fine. Now, what about renal tubular acidosis type 2? Renal tubular acidosis, most of them I have told you already. Few more other than that are multiple myeloma, and drugs associated with type 2 are acetazolamide, sulfonamide, tetracycline, ifosfamide, streptozocin. Acetazolamide, sulfonamide, tetracycline, ifosfamide. So ifosfamide has been associated with both, okay? Type 1 as well as type 2. Indinavir, I could not find it in the list, okay? Indinavir I could not find. Yes, I would scroll it up. First you write down all these things. I would then scroll it up, okay? Renal transplant is with RTA type 1. Renal transplant is with RTA type 1. Renal transplant is with RTA type 1. Okay. Fine. So now this is clear to all of you. The causes for RTA type 1 and 2. Now just few words about cyclosporin causes interstitial nephritis. Okay, it causes interstitial nephritis, but they are not mentioned in the causes for RTA type 1 or type 2. NSAID, cyclosporin, they are all causes for interstitial nephritis. Okay, specifically RTA type 1, type 2, I could not find specifically to which one of them they are associated. Okay, they are causes for interstitial nephritis. Now, what about nephrogenic diabetes insipidus? What is nephrogenic diabetes insipidus? What is diabetes insipidus? How do you define diabetes insipidus? Yes? Diabetes insipidus, how do you define that? You want me to do diabetes insipidus, yes or no? Or have you all done it before? Yes or no? So, what is diabetes insipidus? Diabetes insipidus is polyuria, that is, how do you define polyuria? More than 50 ml per kg body weight per day of urine output. So for a 50 kg individuals, more than 250 ml per day. For a 70 kg individuals, more than 3.5 liters per day. So polyuria plus urinary osmolarity of less than 300 milliosmoles. What if there is polyuria with osmolarity of the urine more than 300? Will it still be diabetes insipidus? No. Then it would be diabetes mellitus. Okay. So urine osmolarity is also important. Not only polyuria, urinary osmolarity less than 300. Fine. Now this diabetes insipidus can be of basically of two types, primary and secondary. Okay. Primary and secondary. This primary diabetes insipidus is further divided into cranial, nephrogenic and gestational. Okay, cranial means when there is decrease in production of ADH from the neurons of hypothalamus. From where is ADH produced? Yes, what is the site of ADH production? ADH is produced by, yes, where is the ADH produced? It is produced by the supraoptic nucleus of the hypothalamus, and it is stored in the posterior pituitary. Okay, it is produced by the supraoptic nucleus of hypothalamus and it is stored in the posterior pituitary. So when there's a defect of the hypothalamus or pituitary, it goes down. Then what about nephrogenic? Nephrogenic is ADH production is normal, but there's decrease in the action of, here it was less of production, here it is less of action, less production, less of action of ADH at V2 receptors. Now, what are the causes for this nephrogenic diabetes insipidus? Causes for nephrogenic diabetes insipidus. Yes, it can either be because of some inherited mutation of the V2 receptor, it can be because of some drugs and drugs associated with nephrogenic diabetes insipidus. Various times it has been asked in the exams. Drugs causing nephrogenic diabetes insipidus. Yes, aminoglycosides, amphotericin B, lithium, phoscarnate, cisplatin, methoxyflurane, etc. Demiclocycline. Okay, this is very important. All these drugs, they are causes for nephrogenic diabetes insipidus. Aminoglycosides, amphotericin B, lithium, phoscarnate, cisplatin. Okay, vincristine is a cause for SIADH. Cisplatin is a cause for nephrogenic diabetes insipidus. They are both totally opposite of each other. Then methoxyflurane and demiclocycline. Demiclocycline is a cause of nephrogenic diabetes insipidus. It is a treatment for SIADH. Okay. Now, other than drugs, some metabolic conditions like hypercalcemia, hypercalciuria, and hypokalemia. More of calcium and less of potassium. They are also causes for nephrogenic diabetes insipidus. Hypokalemia, hypercalcemia, and hypercalciuria. So more of calcium, less of potassium, they are also causes for nephrogenic diabetes insipidus. Causes for nephrogenic diabetes insipidus. And lastly, gestational. Gestational diabetes is because of the production of an enzyme known as N-terminal peptidase or vasopressinase, which is produced by the placenta. Okay, the placenta produces this enzyme N-terminal peptidase or vasopressinase. Okay, and this causes degradation of ADH. This causes degradation of ADH. Fine. So this is gestational. So we have cranial, nephrogenic, or gestational, which are types of primary diabetes insipidus. So all of you have written it down. Yes. Can I scroll it up? Fine. This is clear to all of you. Yes or no? So in cranial, the other name for cranial is, what is not clear? Yes, Vishakhapatnam. What is not clear? Yes, Vishakhapatnam. What is not clear? Audio is not clear? Audio. Yes. The other names for cranial is neurogenic, central, neurogenic, cranial. This is all one and the same. Okay. Central, neurogenic, cranial. It is all one and the same. Okay. Cranial, neurogenic, central. It is all one and the same. Okay. Neurogenic, cranial, central. This is all one and the same. When there is a problem with the production of ADH from the hypothalamus or the pituitary, if there is a disorder in the brain, it is cranial, neurogenic, central. Okay. Yes or no? Vishakhapatnam. Now is it clear to all of you? Okay. Yes, Vishakhapatnam also? Fine. Now what about secondary diabetes insipidus? Yes, Vishakhapatnam. It is visible to you or not? Notepad. What is the problem with the notepad? Okay, fine. So now what do you mean by secondary? So all three of them are types of primary diabetes insipidus. Okay, so they are primary diabetes insipidus. Now what about secondary? What do you mean by secondary diabetes insipidus? What is the secondary diabetes insipidus? Now what is the most important stimulus for ADH release? When is there a release of ADH? The function of ADH is to retain water from the collecting tubules, the last part. Okay, so ADH is released whenever there's hemoconcentration. So whenever we have hemoconcentration, ADH is released. So opposite of this would be whenever there is a decrease in the serum osmolarity, there's dilution. So that would be associated with less of ADH production. So whenever there is decrease in the serum osmolarity, it would be associated with less of ADH production. Okay, so this would be physiological. That is why whenever we have too much of water, we produce a dilute polyuria. That is because of the decrease in ADH production. So whenever serum osmolarity goes down, there is decrease in ADH production, and this is what is primary polydipsia. So for any reason, when there is increase in the water intake, it would lead to dilution of the blood, and that would lead to suppression of the ADH. So the secondary diabetes insipidus is basically primary polydipsia, which can further be divided based on its types. It can be psychogenic, it can be iatrogenic, it can be dipsogenic. Okay, so I'm not going in those details. So this is basically primary polydipsia. Now how do you differentiate various types of diabetes insipidus? How do you differentiate various types of diabetes insipidus? What tests do we do to differentiate various diabetes insipidus? Yes, the diabetes insipidus are differentiated based most importantly on water deprivation test. Now what are the steps of this water deprivation? Water deprivation test. Okay, so this test has 3 steps. The first is at the baseline you have to look for 3 things. Then second step is after water deprivation you repeat those 3 things. And what are these 3 things? Serum sodium osmolarity, serum osmolarity, urinary osmolarity, and the ADH levels, the serum ADH levels. Then after water deprivation you repeat these things: serum osmolarity, urinary osmolarity, and serum ADH levels. Fine. And last step is after exogenous vasopressin or ADH, you repeat urinary osmolarity. Okay, so these are the 3 steps of water deprivation test. 3 steps of water deprivation test. At the baseline you have to look for serum sodium. Third type of secondary is dipsogenic. When there's an alteration of the thirst stimulus. Okay, dipsogenic, dipso. Okay, when there's alteration of the thirst threshold. Okay, yes. So baseline, so serum osmolarity, urinary osmolarity, serum ADH, then water deprivation is done. All these 3 things are repeated, and last is exogenous vasopressin followed by repetition of urinary osmolarity. Okay, so now what if there's a normal individual? There's a normal individual. So in normal individual, at baseline everything is normal. Everything is normal, fine. Now if you give water deprivation to a normal individual, any one of us can be deprived of water for 8 to 10 hours, yes. So what would happen? Whenever there's water deprivation, what would start happening? There would be hemoconcentration. Serum osmolarity would start increasing, but this should not happen in a normal individual. Normal individual would try to maintain a normal serum osmolarity even after water deprivation. Okay, so after water deprivation, normal individual tries to maintain a normal serum osmolarity. Why? Because otherwise there would be shrinking of the brain cells. If there's hyperosmolarity in this blood, it would lead to shrinking of the brain cells and death of the patient. So you have to maintain a normal serum osmolarity if you are normal. Okay, and this is done by increasing the ADH production so that the patient produces a concentrated urine. Okay, so normal individual on water deprivation starts producing a concentrated urine so that they can have normalization of the serum osmolarity. Yes or no? So after water deprivation, normal individual would produce ADH so that their urine is concentrated. Okay? Yes? Notepad. Notepad. Fine. Now what about cranial diabetes insipidus? What about cranial diabetes insipidus? Now, what if there's a patient with cranial diabetes insipidus? In cranial diabetes insipidus, central or neurogenic diabetes insipidus, what is happening? There's less of ADH production at the baseline. Since ADH is always less, what is happening? They're always producing a dilute urine, polyuria, and therefore their serum is slightly concentrated. It can be normal to higher side. Okay, so serum osmolarity even at the baseline is slightly on the higher side. Now, when you give them water deprivation, their serum osmolarity would further increase, and since the ADH is not being produced, so even after water deprivation, the ADH levels would remain low in the blood, and even after water deprivation, they would continue to produce a dilute urine. So this is something very important. The normal individual concentrates urine after giving water deprivation, but here after water deprivation also there is production of a dilute urine. There is production of a dilute urine. Okay, yes or no? And lastly, when you give them exogenous vasopressin, since the defect is with decrease in ADH production, so when you give them exogenous vasopressin and repeat urinary osmolarity, so now their urine would become concentrated. So after water deprivation, they would continue to produce a dilute urine, but when you give them exogenous vasopressin and then repeat the osmolarity of urine, now it would increase. So they would concentrate the urine after giving exogenous vasopressin. Okay? Then nephrogenic diabetes insipidus. So nephrogenic diabetes insipidus. Here what is happening? ADH is being produced, but it is not able to act. There is resistance to the action of ADH, and whenever there is resistance to a hormone, the blood level of that hormone increases. So even at baseline, there is more and more of ADH, but still it is not able to act, and urine is always dilute, and serum is always on the higher side. Okay, serum is always slightly concentrated, it is on the higher side, normal to high, and ADH levels in the blood are high, but still the urine is dilute. Now, when you give water deprivation to such individuals, their blood would become further concentrated, ADH levels would further increase in the blood, but still the urine would remain dilute. Okay, so even after having water deprivation, they would continue to produce a dilute urine. Okay, even after water deprivation, they would continue to produce a dilute urine, and even if you give them exogenous vasopressin, their urine concentration would remain same. Okay, they would not concentrate the urine even after giving exogenous vasopressin. Yes or no? Is this clear to all of you? Yes? Yes or no, or you are not listening? Yes, so finally, what about primary polydipsia? Now, even at baseline, they are having too much of water, so everything is on the lower side, a dilute blood, dilute urine, less of ADH, and when you give them water deprivation, everything would normalize. So this is how water deprivation is going to differentiate between the various types of diabetes insipidus. Okay, I would just summarize it on the whole. A normal individual after water deprivation would concentrate their urine because of ADH production. They would increase ADH production and concentrate their urine. So if any one of us is deprived of water, we would concentrate our urine because of ADH production. But cranial diabetes insipidus and nephrogenic diabetes insipidus, even after water deprivation, they continue to produce a dilute urine. Why? Because in cranial diabetes insipidus, there's no ADH production. So even after water deprivation, ADH levels remain low and they continue to produce a dilute urine. But when you give them exogenous vasopressin, then their urine would become concentrated because now you have compensated for the ADH deficiency. But in nephrogenic diabetes insipidus, ADH is being secreted, but it is not able to act. It is not able to act. Okay, so even after high values of ADH in the blood, their urine is always dilute and this polyuria. So even after increase in the ADH levels in the blood, their urine is dilute. Okay, and even when you give them exogenous vasopressin, the urine remains dilute because that ADH will also not act. Okay. And lastly, primary polydipsia. Vishakhapatnam, don't worry, I would tell you in the end. Okay, you just listen from here onwards. Okay, I would repeat it in the end. Fine. And primary polydipsia, everything is low at the baseline, and when you give water deprivation, everything normalizes. Okay. So how would you treat them? Any other test which can differentiate between them, all three of them? Another test which can differentiate between them is MRI of the posterior pituitary. MRI of the posterior pituitary. How does posterior pituitary look like in a normal individual on MRI? In a normal individual, it appears like a bright spot. Bright spot. Now, what would happen to this? And this bright spot is because of the neurosecretory vesicles containing ADH, which are stored in pituitary, posterior pituitary. Okay. So because of the neurosecretory vesicles stored in posterior pituitary, it appears like a bright spot on MRI. Now what would happen to this spot if a patient has cranial diabetes insipidus? What would happen to this spot? It would be present, it would be absent, or it would be small. Yes, what would happen to this spot if there's cranial diabetes insipidus? Cranial means there's no production of ADH, so no neurosecretory vesicles are there. So what would happen? It would either be small or it would be absent. What about nephrogenic diabetes insipidus? Nephrogenic diabetes insipidus. In nephrogenic diabetes insipidus also, it is either small or absent. Why? Why in nephrogenic diabetes insipidus it is small or absent? It is not large. Yes, Agroha. Why in nephrogenic also it should be small or absent? It is because whatever is produced, whatever ADH is produced is released in the blood, because always the serum osmolarity is on the higher side, so the blood ADH levels, they're high. Okay, so ADH is more in the blood, it is not stored in the pituitary. It is not stored in the pituitary. So the spot is small or absent even in patients with nephrogenic diabetes insipidus. Okay, so this is clear to all of you. What about primary polydipsia? In primary polydipsia, this is normally present. Normally present. So what is more important for diagnosis? Presence of the spot or absence of the spot? What is more important diagnostically? Presence or absence of the spot? More important diagnostically is presence or absence of the spot. Yes. Okay, my father. It's too loud. Fine. Yes. So what is important? Presence or absence of the spot? Yes. Yes. Presence of the spot is more important. Is more important. Fine. Presence of the spot is more important because even 10% of normal individuals can have 10% of normal individuals can also have absence of the spot. Okay, it can be absent even in 10% of the normal individuals. So it is always better to have presence of the spot, because presence of the spot rules out cranial diabetes insipidus as well as nephrogenic diabetes insipidus. Okay. Yes, so presence of the spot is more important because it rules out cranial or nephrogenic. Now, what is the treatment of various types of diabetes insipidus? For cranial diabetes insipidus, you have to give them vasopressin or ADH. Yes, so now is it fine? The volume? Nephrogenic diabetes insipidus, what do you need to give them? Nephrogenic diabetes insipidus. What is the treatment of choice for nephrogenic diabetes insipidus? Treatment for nephrogenic diabetes insipidus. Yes? Treatment for nephrogenic diabetes insipidus is thiazide group of diuretics plus salt restriction. Okay? So salt restriction plus thiazide is the treatment for nephrogenic diabetes insipidus. Demiclocycline was a cause of diabetes insipidus. Okay? It is a treatment for SIADH. It is not a treatment MESUR. It is not a treatment for nephrogenic diabetes insipidus. Okay? Demiclocycline is rather a cause of it. So thiazide group of diuretics with salt restriction. So how would the thiazide help? Thiazide is a diuretic. The patient is already having polyuria. So how will a thiazide help a patient with polyuria? Yes? How would the thiazides help a patient with polyuria? Now thiazide is a low ceiling diuretic. Okay, low ceiling diuretic, okay, because it has a longer action and the onset of action takes some time. So what would happen after 2 to 3 days? So for initial 2 to 3 days, there is diuresis, okay. Patient is having polyuria. On top of that, you have given them diuretics. So there is diuresis. So after 2 to 3 days of diuresis, and plus salt restriction, what would happen? You have restricted the salt intake and there is diuresis. So after 2 to 3 days, what would happen to the circulatory volume? Circulatory volume would go down. So there is decrease in the circulatory volume, decrease in the plasma circulatory volume. Okay, plasma circulatory volume. So when there's diuresis for 2 to 3 days along with salt restriction, after 2 to 3 days there's decrease in the plasma circulatory volume. So when there's less of plasma volume, what would happen to the GFR, the renal blood flow? There would be decrease in the renal blood flow. There would be decrease in the GFR. So whenever there is decrease in the plasma circulatory volume, there's decrease in the renal blood flow, there's decrease in the GFR. Now when GFR becomes less, what becomes active? Whenever GFR is less, what becomes active? Yes? What becomes active? There's increase in the PCT reabsorption. Yes, RAS becomes active, but that is not important here. So here there's increase in the PCT reabsorption. Why? Because if you have a river, if the river is flowing very fast, you cannot pick up things from it. But if the river is slowly moving, you can pick up many pebbles or stones from the river. Similarly, when GFR is less, the flow of fluid is less. And when fluid is slowly moving, the PCT becomes active and many things can be reabsorbed from the proximal convoluted tubule. Okay, so when GFR is less, there's less of tubular flow. The tubular flow decreases. There's a decrease in the tubular flow, and when tubular flow is less, then PCT reabsorption increases. Okay, is this clear to all of you? So what would happen? The urine would now become concentrated at the level of PCT. Whatever water and sodium is being filtered, more and more is reabsorbed at the level of PCT. So already a concentrated urine reach the distal part. Okay, so this is clear to all of you, yes or no? So because of this increased reabsorption at PCT, the urine is already concentrated before it reaches your distal nephron. Okay, before reaching the distal nephron, it is already concentrated at the level of proximal convoluted tubule. At the level of proximal convoluted tubule. Okay, and along with this, there's one more action of this thiazide, that it increases translocation of aquaporin 2 independent of ADH. Okay, it also increases translocation of aquaporin 2. It also increases translocation of aquaporin 2. Is this clear to all of you? How thiazides are going to help a patient with nephrogenic diabetes insipidus? Yes? It would increase translocation aquaporin 2 in the collecting tubules, and it would increase the sodium water reabsorption at the level of PCT. So already a concentrated urine would reach the distal nephron. Fine? Yes. So yes, now I think after this we would move on to the glomerular diseases. Okay. So after your tubule part, we would move on to the glomerular diseases. Okay. Yes, so now what are the components? Yes, furosemide also can be used. Yes, furosemide can be used, but the problem of furosemide is that action of furosemide is brisk in onset and it is there for a short duration. Okay, furosemides can be used, but they're not the preferred one because they will not produce a sustained action. Their action is quick in onset and it would not be there for a longer duration. Thiazides take some time for their action to start, but they have a longer action. Okay, they are low ceiling diuretics. Furosemide may be used, but it is not that helpful. Yes, chlorpropamide can be used. There are various other drugs also which can be used. Okay, chlorpropamide is a cause for SIADH, so it can be used here. Chlorpropamide. It is, what is chlorpropamide? It is a class 1 sulfonylurea. So it can be used in both diabetes insipidus plus diabetes mellitus. It is used in treatment for both of them. But be careful when you give it to younger children and pregnant females because of the chances of hypoglycemia. Okay, it is usually not used. You can give because it is associated with SIADH. It increases the release of ADH, so it may be helpful, but not always. Then another drug which can be given is indomethacin, can also be given. And which drug do we give in lithium-induced nephrogenic diabetes insipidus? Yes, which drug is given in lithium-induced nephrogenic diabetes insipidus? Lithium-induced nephrogenic diabetes insipidus, which can be used? Yes, carbamazepine is a cause of SIADH. Whichever drug causes SIADH can be used in treating diabetes insipidus, but not all of them are used. They have their own side effects. Okay, so they may not be very much effective for treatment. Okay, they can be used, but they are not usually used. So is amiloride. Amiloride is the drug which is also used for treating lithium-induced nephrogenic diabetes insipidus because it blocks the sodium channel and lithium enters this part of the cell by using the sodium channels. Okay, lithium would enter the cell using the sodium channels. So when you block the sodium channels, the lithium would not enter, and this would help in treating lithium-induced nephrogenic diabetes insipidus. Okay, fine. So I think that would be all for nephrogenic diabetes insipidus. Now what about the glomerular diseases? Yes, so for glomerular diseases, so what are the components of your glomerulus? You have the capillaries, okay? Then below the capillary is your basement membrane, and below that is your Bowman's capsule. This Bowman's capsule is made up of two layers. The visceral layer, which is made up of the podocytes. So visceral epithelium made up of podocytes, and this is the parietal epithelium. This is the basement membrane. This is the capillary. You have 8 to 10 loops of capillary in the Bowman's capsule. Okay, in one glomerulus there are 8 to 10 loops of capillary. And here is your mesangial cells. These are your mesangial cells. Mesangia. Okay. So this is the glomerulus. So filter, and the endothelium is made up of only one layer. That is your, the capillary is made up of only one layer. That is the endothelial cells, the tunica intima. Okay, it is made up of only the endothelial cells, the tunica intima. So this is capillary endothelium. Okay, so for filtration to occur, the substances must pass through this filtration barrier. And the filtration depends on two things. The filtration depends on two things. What are those two things? The size of the particle and the charge of the particle. Okay, so filtration of a substance through this filtration barrier depends on two things: the size and the charge. Now, what is the size of this filtration pore? Yes, what is the size of this filtration pore? Yes, 4 to 8 angstroms. Okay, so this is 4 to 8 angstroms. Now, substances like albumin. Albumin has a size around 8 angstroms, so it is just adequate for filtration. But normally albumin does not filter. Why? It is because of the charge. Because albumin is a negatively charged substance. Okay, negatively charged substances, they are repelled by negative charge of the basement membrane. The basement membrane is made up of some proteins which are negatively charged. Okay, the proteins which are present in the basement membrane, they are negatively charged, and because of which there is repulsion of the negatively charged particles. Okay, so what are the negatively charged proteins present here? Polyanionic proteoglycan, laminin, fibronectin, and tectin. Okay. Okay, so because of these negatively charged proteins which are present in the basement membrane, the negatively charged substances are repelled. So albumin, even though the size is adequate for filtration, it does not filter because of its negative charge. And if for some reason this neutralization of this negative charge, then albumin would start filtering down. And this is seen in patients with minimal change disease. In minimal change disease, the filtration barrier is normal otherwise, but still there is massive proteinuria in nephrotic range because of the neutralization of the charge. Okay, so now when we talk of the proteinuria, how many types of proteinuria do we have? The proteinuria can be of two types: the glomerular and the tubular proteinuria. The proteinurias can be of two types. And tectin, they are all proteins present in the basement membrane: laminin, entectin, etc. Okay. Yeah, so proteinuria can be divided into glomerular and tubular. So glomerular is because of the defect of this filtration barrier. Defect in filtration barrier. Defect in the filtration barrier, that there is glomerular proteinuria, whereas tubular is because of the damage of renal tubular cells. Okay, so when there is damage of the tubular cells, it leads to tubular proteinuria. Otherwise, If there's damage of the filtration barrier with increase in filtration of the proteins, it is glomerular proteinuria. So now this glomerular proteinuria will always be more in quantity compared to the tubular, and usually it is more than 2 grams per day, but tubular would always be less than 2 grams per day. So tubular proteinuria is always less than your glomerular proteinuria, and sometimes it can be even more than 3.5 grams per day when it is known as nephrotic range proteinuria. Nephrotic range proteinuria. Okay, so proteinuria is more in glomerular type compared to the tubular type. When we talk of the type of protein which is present here, in glomerular proteinuria, the most important is the albumin. Albuminuria is the most important protein. Albumin is the most important protein which constitutes the glomerular proteinuria. But in tubular, it is mostly constituted by the low molecular weight proteins like alpha 2, beta 2 microglobulin, some small weight molecular proteins which are filtered from above, and along with the proteins produced by the tubules like the TAM horsefall protein. The TAM horsefall proteins. Okay. So TAM horsefall protein are increased in patients with tubular proteinuria, not in patients with glomerular proteinuria. Then when we compare the albumin to beta 2 microglobulin ratio, albumin to beta 2 microglobulin ratio, it is more than 1000:1 in patients with glomerular proteinuria, and it is less than 10:1 in patients with tubular proteinuria. Okay, so the ratio is decreased because there's an increase in the beta 2 microglobulin here. Okay, then N-acetyl glucosamine, N-acetyl glucosamine. Now this N-acetyl glucosamine is a large molecular weight protein. Large molecular weight protein which cannot filter through glomerulus. Okay, which cannot filter through glomerulus. Okay, so this N-acetyl glucosamine will never be a part of, yes, TAM horsefall protein I would tell you, don't worry. So this N-acetyl glucosamine will never be a part of glomerular proteinuria. Okay, this N-acetyl glucosamine is normal here, but it is increased in patients with tubular proteinuria. Okay, because N-acetyl glucosamine in the urine will never come from the glomerulus because it is a very large protein, it cannot filter through the glomerulus. Whatever N-acetyl glucosamine is there is because of the destruction of cells, especially in the proximal convoluted tubule, because the cells in the PCT, they have an enzyme known as N-acetyl glucosaminidase. Okay, and this enzyme would cleave the N-acetyl glucosamine. So when there is injury to these cells, you can have N-acetyl glucosamine in the urine. Okay, otherwise it does not get filtered from the glomerulus. So it is normal in the glomerular proteinuria, but it is more in patients with tubular proteinuria. Yes or no? So can I change over to the next page? Yes. So now what is TAM horsefall protein? What is TAM horsefall protein? From where is this produced? TAM horsefall protein. TAM horsefall protein is produced by the ascending limb of loop of Henle. Okay, TAM horsefall protein is produced, it is not produced by DCT. Okay, Bilaspur, it is not produced by DCT. It is produced by the ascending limbs of loop of Henle. Okay, it is produced from the excuse me, ascending limbs of loop of Henle. Okay, ascending limb of loop of Henle is the site of production of TAM horseful protein. In the collecting tubules, these proteins can get coagulated to form cast. Okay, cast are formed in the last part because the proteins get coagulated there. And why the proteins get coagulated? Because of the acid secretion which is happening in this part, plus the urine is maximally concentrated in the last part. Okay, so because of the increase in concentration, because of the acid secretion, the proteins, they tend to get coagulated in the last part and they form the cast. Okay. N-acetyl glucosamine is a very large protein. Okay, albumin has a size which is just appropriate for filtration, but normally it is not filtered because of its negative charge. Some little bit amount of albumin can be there, up till 150 milligrams of protein we can have in a normal urine also. So some little bit albumin can be there along with some other low molecular weight proteins. Okay, but N-acetyl glucosamine is a very large protein, so it does not get filtered. Okay, it does not get filtered. It is only through the tubular damage that you can have N-acetyl glucosamine in the blood. Yes. So this TAM horsefall protein is produced by the ascending limb of loop of Henle. Now what is the other name for this N-acetyl glucosamine, this TAM horsefall protein? Yes. The other name for this is uromodulin. This is also known as uromodulin. Okay. And it is, what is the function of this TAM horsefall protein in normal urine? It basically inhibits calcium crystallization in renal tubules. Okay, it inhibits calcium crystallization. It inhibits calcium crystallization and it protects against development of UTI. Though the role is not very well understood, what is the function of TAM horsefall protein, why is it produced, but some say it helps in inhibiting calcium crystallization in the normal urine, and also it is a protective protein against the UTI. Okay, it protects against infections and prevents calcium crystallization. Exactly the role is not very much clear why it is produced. Okay, so TAM horsefall protein is clear to all of you. Now what are these cast? So what is cast? Let me just explain you the cast also. Yes. So what are cast? What is a cast? So cast is a collection of coagulated proteins. Collection of coagulated proteins. And where is the coagulation of proteins occurring? The coagulation of proteins would occur in the collecting tubule, because it is the part where urine is most concentrated and there is acidification of urine which has occurred. So this is coagulated protein and other organic matter, which can be anything. So when there is coagulation of protein, some other organic matter can also add on to it, and they would take up the shape of the nephron. They would take up the shape of the nephron. Coagulated protein and other organic matter, they solidify, take the shape of the nephron, and this is what is known as cast. This is what is known as cast. Okay. Now what is hyaline cast? So coagulated protein along with other organic matters which solidify, take up the shape of the nephron, which is the cylindrical shape. Cylindrical shape is your cast, and they are formed in the collecting tubules. In the collecting tubules. Okay, so the cast are always formed in the collecting tubules. Now what is hyaline cast? Hyaline cast is when only proteins are coagulated. When only proteins are coagulated. Only proteins are there. Only proteins they coagulate. It is a hyaline cast. And this hyaline cast can even be seen in some normal individuals. Some normal individuals can have this hyaline cast, especially when they exercise, or when there is fever, or people who have orthostatic proteinuria. Okay, so hyaline cast is when simply these proteins they coagulate, they form hyaline cast. It can be seen in some normal individuals when they exercise or when they have fever, or people who have orthostatic proteinuria, they can be having hyaline cast in their urine. So this is hyaline cast. Now what is RBC cast? When along with the proteins you have RBCs on top of that, it would form RBC cast. When there's hematuria along with proteinuria, both of them would get coagulated, and that would lead to formation of RBC cast. So RBC cast basically indicate glomerulonephritis. Glomerulonephritis. So when there's inflammation of the glomerulus, there's hematuria, RBCs are coming from above, and along with the proteins, they get coagulated in the last part. It leads to formation of RBC cast. Then what are granular cast? Granular cast. They can be further divided into fine and the coarse granules. Granular cast are the ones in which the cells are so much disintegrated that you cannot identify the initial cell. So when there is breakdown of cellular cast to the extent that you cannot identify the original cell, you call it granular cast. Okay, so when you cannot identify the original cell, it is only the debris along with proteins which you see, you call it granular cast. They can be fine or coarse granules. They are also known as pigmented muddy, they are brown cast, muddy brown cast. Okay, they are pigmented muddy brown in color, pigmented muddy brown in color, and the causes for them are pyelonephritis, they're always pathogenic, pyelonephritis, tubular necrosis, any renal parenchymal disease. Okay, so the granular cast are basically pigmented muddy brown cast because of the breakdown of the cellular cast to the extent that you cannot identify the original cell. The causes are pyelonephritis, tubular necrosis. Basically they indicate renal parenchymal disease. Some say even in chronic lead poisoning you can have them. Chronic lead poisoning. Chronic lead poisoning is also a cause for granular cast, is also a cause for granular cast. What are broad cast? Yes, can I change the page? Okay, can I move further? Yes, so what are broadcast? What are broadcast? Broadcast are basically seen in patients with chronic renal failure. In chronic renal failure, there are dilated tubules. There are dilated tubules. Okay, some say because of the fibrosis which has occurred, there's dilatation of the tubules in chronic renal failure. So basically broadcast is because of the dilated tubules. So when the size of the cast is more, we call it broadcast. Okay, yes, what coarse granular cast are basically the pathological. The granular cast which I have told you is basically the coarse granular cast. Fine granules can even be seen in exercise. Okay, coarse granular cast are what I just told you: pyelonephritis, lead poisoning, then this tubular necrosis, renal parenchymal disease. So coarse granular cast is same. Okay, coarse granular cast is granular cast. Okay, fine granules are usually seen in exercise. Okay, then waxy cast. Waxy cast, they are the final stages of fine granular cast. of the granular cast, their final stages of granular cast, to the extent that granules have degraded. Granules degraded. So basically they indicate nephron obstruction. They basically indicate nephron obstruction. So since there's obstruction of the nephron, there's a long standing period, okay, for these casts to form, and there's destruction of the granules. So nephron obstruction is indicated by the waxy cast. You can also have them in diabetic nephropathy, malignant hypertension, okay. So waxy casts are usually the final stages of granular cast, to the extent that even the granules are not appreciated. So usually they indicate obstruction to the nephron, okay. When there's obstruction, there's long standing urine is there, so granules are degraded. They can also be seen in diabetic nephropathy, malignant hypertension, okay. Then epithelial cast. Epithelial casts, they are quite rare, and they are formed of fused, desquamated epithelial cells, okay. When the epithelial cells, they desquamate and they form a cast, it is known as epithelial cast. It is seen in patients with, causes for this is amyloidosis, heavy metal poisoning, ethylene glycol intoxication, also in patient with eclampsia, okay. So epithelial casts are because of the fused desquamated epithelial cells, as we have in amyloidosis, heavy metal poisoning, ethylene glycol intoxication, eclampsia, okay. Fine. So the cast, I hope they are clear to all of you. So now what is nephrotic syndrome and what is nephritic syndrome? Nephrotic or nephritic syndrome. What is nephrotic syndrome? Nephrotic syndrome, yes. If I ask you what are the essential criteria for nephrotic syndrome, what are the essential criteria of nephrotic syndrome? Yes, essential criteria for nephrotic syndrome. The essential criteria for nephrotic syndrome are proteinuria, hypoalbuminemia, and peripheral edema, okay. So these are the important essential criteria. Lipiduria, other things, they are not the essential criteria. These three are the essential criteria. The essential criteria are these three things, okay. So this is the triangle. There's proteinuria because of the damage to your glomerulus, which is leading to hypoalbuminemia, which is then leading to peripheral edema, and this peripheral edema then has a vicious cycle. So these are the essential criteria, okay. Proteinuria, hypoalbuminemia, and peripheral edema. On top of that, we have some other criteria which I would discuss slightly later. First, what about this peripheral edema? So what is the cause for this peripheral edema? The cause for this peripheral edema, there's a primary cause, and superimposed in that there are secondary factors which further increase this peripheral edema. The primary cause for this edema is hypoalbuminemia. So when there's less of albumin, the colloid oncotic pressure of the blood is less, it cannot hold water, so the water exudes and leads to edema formation. Albuminemia, okay. So primary cause for this is hypoalbuminemia. Now because of this hypoalbuminemia and edema formation, what happens to the circulatory volume? This decrease in the plasma circulatory volume. Okay, and this leads to initiation of secondary factors. This leads to stimulation of secondary factors. Okay, what is the first secondary factor? The first secondary factor is the stimulation of renin and renin angiotensin aldosterone system. Whenever the circulatory volume is less, what happens? There is decrease in the stimulation of increase in the stimulation of renin angiotensin aldosterone system, and then this aldosterone is more. This is known as secondary hyperaldosteronism. Okay, so there's secondary hyperaldosteronism because of the stimulation of renin angiotensin aldosterone system, and this would lead to sodium retention, which would be accompanied by some water retention and potassium secretion. And then second thing is whenever there is exudation of fluids, so what is happening? There's hemoconcentration, and because of that hemoconcentration, another thing is stimulated, and that is the ADH. Okay, ADH is released whenever there's increase in serum osmolarity. So there's increase in ADH because of the increase in serum osmolarity, because whatever fluid is there is exudating into the interstitial compartment, and the circulatory volume is less. There's concentration, hemoconcentration, so ADH is also released more, and this would also lead to water retention. But whatever water is being retained by the kidney is not being held in the circulatory compartment. Okay, from the kidney it is retained in, it is brought to the blood, but from the blood it would go on into the interstitial compartment. It would not be retained in the blood. So more and more of edema would lead to more and more of edema. So because of the hypoalbuminemia, whatever is being retained is not being held in the circulatory compartment. It would all go out in the interstitium. Okay, so primary mechanisms and secondary mechanisms of peripheral edema. Now on top of these three things, okay, so this is clear, primary and secondary mechanisms of peripheral edema. So peripheral edema is self-perpetuating. There are some other things like when there is hypoalbuminemia, there's less of albumin in the blood. Now albumin is being produced by the liver. So this leads to stimulation of synthetic capacity of liver. So not only albumin, other proteins are also synthesized more. So this leads to increase in albumin production, which tries to compensate, but it is not able to compensate for the albumin loss. And along with that, there's also increase in other plasma protein, which is fibrinogen. Okay, plasma proteins are albumin, fibrinogen, and globulin. Globulin is coming from the plasma cells, and albumin and fibrinogen, they come from the liver. So not only albumin is more, there's also increase in fibrinogen. But when we see in the blood, albumin still remains low because it is excreted in the urine. It is excreted in the urine. But fibrinogen levels, they increase in the blood because fibrinogen is a large molecule. So it is not usually excreted in the urine. Okay, so usually fibrinogen is not excreted in the urine. Even if there is damage to the filtration barrier, fibrinogen is so large that it does not usually come in the urine. Okay, so fibrinogen levels in the blood, they are more because synthesis has increased, but it is not being excreted in the urine. Not that much excretion is occurring from the urine. Okay, on the other hand, albumin is still less because too much of albumin is being excreted in the urine. So there's increase in the fibrinogen level. Now another thing which increases from the liver is the lipoproteins. There's increase in the lipoprotein synthesis by the liver, and this leads to the dyslipidemias. That we have in patients with nephrotic syndrome. Okay, so because of the increase in lipoprotein synthesis by the liver, there is dyslipidemia, increase in the bad cholesterol is there, increase in VLDL, LDL, and there is decrease in the HDL. Okay, so all the bad cholesterol they increase, HDL is less. Okay, not only this, there would be increase in the excretion of other small weight molecular proteins, excretion of other small weight molecular proteins like transferrin. When excretion of transferrin increases, it leads to iron deficiency anemia, There's increase in the excretion of vitamin D binding protein, that is called calciferol binding protein. There's increase in the excretion of called calciferol binding protein, so that leads to decrease in the calcium, hypocalcemia, rickets, etc. Then there's also increase in the excretion of ceruloplasmin. Okay, so transferrin, vitamin D binding proteins, ceruloplasmin, they're all excreted in the urine, and also antithrombin 3. Okay, they are also excreted in the urine. Antithrombin 3 is also excreted in the urine. Okay, so antithrombin 3, which is an anticoagulant, is excreted in the urine. On the other hand, fibrinogen levels, they are more in the blood. Okay, so this would lead to a procoagulant stage. This hypercoagulation in patients with nephrotic syndrome. They have hypercoagulability because of the increase in the fibrinogen and decrease in the antithrombin 3. There's decrease in antithrombin 3 and there is increase in the fibrinogen level. Yes, lithium utilizes the sodium channels in the last part, distal part, epithelial sodium channel. Not all the places, but in last part, lithium uses the sodium channels only to get reabsorbed. Okay, let's just continue. Yes, Agartala, previously I just told you the other small weight molecular proteins like transferrin, ceruloplasmin, cholecalciferol binding protein, they are also lost in the urine along with antithrombin 3. So this hypercoagulability here because of the increase in fibrinogen levels in the blood and decrease in the antithrombin 3. So they have high chances of thrombosis. And what is the most common site for thrombosis that we have in nephrotic syndrome? Most common site of thrombosis in nephrotic syndrome. Yes, you can have it at various places, but the most common site is the renal vein. And which type of nephrotic syndrome is most often associated with renal vein thrombosis? Yes, which type of nephrotic syndrome? Which nephrotic syndrome? It is most often associated with membranous glomerulonephritis. So membranous glomerulonephritis is the most important cause for renal vein thrombosis. Most common type of nephrotic syndrome associated with renal vein thrombosis. Fine. Yes. So now, what about hematuria? Can a patient with nephrotic syndrome have hematuria? Yes or no? Hematuria. Yes. Minimal hematuria can be present and usually it is microscopic. Okay. Microscopic hematuria can also be present in patients with nephrotic syndrome. Okay. Nephrotic syndrome can also be associated with minimal hematuria. Fine. Now, what about nephritic syndrome? Nephritic syndrome is usually a, is a nephrotic syndrome superimposed with components like hematuria, hypertension, and oliguria azotemia. Oliguria azotemia. This is very important. So, oliguria azotemia with hematuria, hypertension. So, on top of this triangle of nephrotic syndrome, you have a hot triangle made up of hematuria, hypertension, and oliguria oblique azotemia. Okay. Now, treatment of nephrotic syndrome we would discuss with individual types of nephrotic syndrome. Nephrotic syndrome we would discuss the individual types. Okay. Otherwise, the pediatric teacher they would teach you better the treatment of nephrotic syndrome. Okay, because nephrotic syndrome is most often seen in your, especially the minimal change where they have to look for steroid responses, steroid resistant patients, and other things. Okay, rest we would discuss with the individual types when we discuss. Yes. So now, yes, why do they have less of proteinuria? Proteinuria is more in nephrotic or nephritic. Yes, proteinuria is more in nephrotic or nephritic. Is more in nephrotic or nephritic. Proteinuria. Yes, is more in nephrotic compared to nephritic. Now, why there is a decrease in proteinuria in patients with nephritic syndrome? Nephritic is more severe than nephrotic, but still the proteinuria is less. One, how much decrease is there? We would talk of that in renal failure. Okay, when we define renal failure, then we would talk of the decrease or increase in the blood urea nitrogen, the creatinine. Okay. Nephrotic, nephritic, yes. So why is proteinuria less in nephritic syndrome? Proteinuria is less in nephritic syndrome because of the oliguria. The filtration is less because the nephrons are so much damaged that there's no filtration which is occurring. So when filtration is less, so proteinuria would also be less. That is why nephritic is associated with less of proteinuria. Is associated with less of proteinuria. What do you mean by selective or non-selective proteinuria, which is bad for the patient? Selective or non-selective proteinuria, which is bad for the patient? Selective or non-selective proteinuria, which is bad for the patient? The answer is non-selective. Okay, Jammu, Agroha, Shimla, Haldwani, it is non-selective. What do you mean by selective or non-selective? Selective proteinuria means only albumin is being filtered. Okay, only albumin is being filtered. And non-selective would mean albumin with other plasma proteins like globulins, some fibrinogen also. Okay, so non-selective proteinuria is poor, is bad, okay. This means the damage is more. Okay, so non-selective proteinuria means the damage is more. Okay, selective is only albumin. Even in minimal change you will have albuminuria. Okay, okay, so selective proteinuria is better than having non-selective proteinuria. Fine. Now, what are the mechanisms for glomerular diseases, glomerulonephritis? What are the various mechanisms for glomerulonephritis? how do you identify the glomerulonephritis? The patient will have some mechanism of injury, usually some primary immune mediated injury to the glomerulus, which would be followed by some secondary mechanisms which would produce some morphological changes. Lunch break, you want it right now? Yes? Lunch break? Yes? Okay, so let's have half an hour of lunch break. Okay, half an hour of lunch break. Fine. Yes, fine to all of you. Yes, so now the mechanisms of injury. So the patient comes to you with the clinical picture. There are some primary mechanisms of injury, followed by some secondary mechanisms, then the morphological changes are produced in the nephron, and as a result of that, there's a clinical syndrome. Clinical syndrome. The patient would come to you with the clinical picture. This is happening in the patient, okay? No, long-standing nephrotic may not lead to nephritic always, okay? Nephrotic does not lead to nephritic always. Some patients may complicate into nephritic, not always. Some may directly present to you with nephritic, and others may not change into nephritic, okay? I would tell you the causes of nephrotic, the nephritic. The glomerular diseases, they're divided into 6 syndromes, okay? Now we are discussing the mechanisms of pathogenesis for your glomerular diseases. Reason for oliguria is because when the glomerulus is inflamed, when there's damage of glomerulus, okay, so there would not be any filtration, okay, only when the glomerulus is patent you will have filtration. So when no filtration is there, so oliguria, so less of proteinuria, okay. So patient would come to you with clinical pictures. You would do a kidney biopsy, and you would see the morphological changes, and based on that you would decide what mechanism would have happened, okay. Now first, the mechanisms of injury. Mechanisms of injury. The first mechanism is immune complex mediated disease. Immune complex mediated disease. Now, immune complex mediated disease, when we talk of, either you can have in situ immune complexes. In situ means the immune complexes which are formed within the kidney, or you can have immune complexes which were present in the circulation but they come and get deposited in the kidney. So circulating immune complexes or in situ immune complexes. Now the in situ immune complexes, the complexes which form in kidney, can be further divided based on the type of antigen. If the antigen is intrinsic antigen, or if it is a extrinsic antigen. Now a classical example of a condition where you have antibodies against a intrinsic antigen of kidney, yes, we discussed it yesterday. Classical example is anti-GBM syndrome, antibody syndrome, or your good pastures, okay, in which you had antibodies against the NC1 of alpha 3 chain of collagen type 4, okay. So this is one example of antibodies against the in situ intrinsic antigen. Now another example of intrinsic antigen is Hayman's nephritis. This Hayman's nephritis is usually not seen in human beings. It is usually seen in some lower animals. Actually in this there are antibodies against Miguelin. antibody against Migallin. Okay, and where this protein is lying, Migallin, this is lying between the foot processes of the podocyte. Between the foot processes of the podocyte, you have this protein Migallin. Okay, this is sitting in between the foot processes. Okay, in between the foot processes, you have this Migallin. Okay, so in between the foot processes, you have this Migallin. Fine. So Hayman's nephritis. Now some extrinsic antigen which can just come and get deposited in the kidney are various. Most importantly, your toxins, various toxins like some microbes like streptococcus, malaria, hepatitis B, hepatitis C, treponema pallidum. Then drugs. So other than microbes, some drugs. So microbes, then drugs. Drugs like penicillamine, D-penicillamine, heavy metals, then some toxins. Okay. So all these things can also get deposited in the kidney, and when antibodies come and form an immune complex with these antigens which are implanted in the kidney, it would again form a in situ immune complex. And then is your circulatory immune complex. The classical example of disease where you have circulatory immune complexes getting deposited in the kidney is SLE. And when circulating immune complexes they get deposited in the kidney, it is an example of which kind of hypersensitivity reaction? Type 3. And when you have in situ immune complexes, that is type 2. So this is kind of type 3 hypersensitivity reaction, and this would be kind of type 2 hypersensitivity reaction. Type 2 hypersensitivity would be this. Okay. Yes, so now this was the first mechanism. SBP in nephrotic syndrome, it can occur in any kind of ascites. Okay, SBP is because of the transmigration of pathogens. Okay, it has nothing to do with your mechanisms of injury that we are discussing. Okay, that is different. Spontaneous bacterial peritonitis is different. Nephrotic, you have ascites. Okay, yes. Now second mechanism is cytotoxic antibodies which directly damage your cells in the kidney. So cytotoxic antibodies against cells of your kidney like the endothelial cells of the capillaries, the epithelial cells, the visceral or the parietal epithelium, the mesangial cells. So if there are some antibodies which directly just come and damage these cells, it is cytotoxic antibodies. So that can be another examination mechanism. Third would be cell mediated injury due to some cytokines which are produced, and this is classically seen in conditions like minimal change disease. In minimal change disease, some cytokines are produced which lead to alteration of the charge of the membrane. Okay, so because of the alteration of the charge, there's no negative charge and there can be filtration of albumin. So minimal change disease, there is massive proteinuria even though they may not have any morphological change in the kidney. Fine, due to neutralization of charge by cytokines. Okay, and lastly we can have the stimulation of alternate pathway of complement. Okay, so these are various primary mechanisms. On top of this, we have some secondary mechanisms like stimulation of macrophages, stimulation of neutrophils, then macrophages, the classical pathway of complement, etc. Okay, some growth factors which are produced. So all these contribute to the secondary mechanisms. Okay, so after this injury, the end result is morphological change in the nephron. So because of all these processes, there would be a morphological change in the nephron, and the morphological changes in the nephron are seen on various kinds of microscopy. So to look for the morphological changes, we have to do the kidney biopsy, and then you have to do various kinds of microscopy. For example, light microscopy, which would show you the cellularity, which would tell you about the thickness of the basement membrane, etc. Then second would be immunofluorescence, which would show you immune deposits. Previous slide I just told you the mechanisms of secondary mechanisms of injury. The macrophages, neutrophils, the complement, they are also activated when you have some primary mechanism going on. Okay, so top of that, the secondary mechanisms also start acting. And lastly, the electron microscopy. Electron microscopy. This electron microscopy would show you immune deposits. It would tell you about the foot processes of the podocytes, their effacement, fusion, cellularity, everything. Okay, so based on the morphological changes that we have, we give different names to different kinds of glomerulonephritis. Okay, now to understand the different types of glomerulonephritis, you must understand some terminologies. Okay, this all of you have written it down. Can I move further? Immunofluorescence, electron microscopy, light microscopy. Okay, yes. So if this is your capillary, then this is your basement membrane, and then is your Bowman's capsule. Okay, now, and this is your mesangium. So when we talk of the immune deposits can occur at various places. They can be there in the mesangium, they can be there between the basement membrane and the endothelium, they can be there between the basement membrane and the visceral podocytes. So you can have mesangial deposits, you can have subendothelial between the endothelium and the basement membrane, and you can have subepithelial deposits. Okay, you can have mesangial, subendothelial, subepithelial deposits. Now whenever we have the subepithelial deposits, they are lying immediately below your visceral podocytes. So whenever we have subepithelial deposits, they start irritating the visceral podocytes. And when visceral podocytes, they are irritated, they increase the production of basement membrane material. Okay, so whenever the podocytes are irritated, okay, whenever the podocytes are irritated, okay, by these subepithelial deposits, what would happen? The podocytes would increase the production of basement membrane material. They would increase the production of basement membrane material. And when there's more and more of basement membrane material, how would your basement membrane look like on light microscopy? It would appear to be thick. So, subepithelial deposits would increase the thickness of the basement membrane by irritating the visceral podocytes. And whenever we have a thick basement membrane, the name given to that kind of glomerulonephritis is membranous. So, membranous glomerulonephritis is any glomerulonephritis associated with a thick basement membrane. So, whenever we see a thick basement membrane on microscopy, light microscopy, then we call it membranous glomerulonephritis. Membranous glomerulonephritis. Is this clear to all of you? Yes or no? So, subepithelial deposits would always lead to thick basement membrane, and that would be known as membranous glomerulonephritis. Okay. Now, there are some terms which are used to describe the morphological changes like focal, diffuse. What do you mean by focal? What do you mean by diffuse? Focal means less than 50% of all the glomeruli are involved. And diffuse means more than 50% of all the glomeruli are involved. So focal, diffuse. So when we talk of the total number of nephrons in the entire kidney, we describe it either as focal disease or a diffuse disease. The next is to see if it is segmental or global. The terms like segmental and global, they are used when we talk with respect to individual nephron. Okay, if the nephron is totally involved, we call it global. If it is partially involved, we call it segmental. For example, this focal segmental glomerulosclerosis, FSGS. Now, focal segmental. Focal, so less than 50% of all the glomeruli. What do you mean by segmental? This glomerulosclerosis, that means there is fibrosis of the vessels. Now, in each glomerulus, there are 9 to 10, 8 to 9, some say 8 to 10 loops of capillaries. Okay. Usually we show only one, but there are 8 to 10 loops of capillary. This is the basement membrane and this is your Bowman's capsule. Okay. So now in focal segmental, since it is segmental, so only some loops will have fibrosis, other loops will not have fibrosis. So only when some loops will have fibrosis, it would be a partial involvement, it would be known as segmental. Okay. So focal means less than 50% of the total glomeruli are involved, and individually when we look at the glomerulus, the nephron, it is only partially involved. Partially involved since some loops have fibrosis, some do not have. Okay. And in those which are involved also, some have complete fibrosis, some have partial fibrosis. Okay. Fine. Okay. Now is it better? Now am I audible, Ludhiana? Yes. So focal, diffuse, segmental, global. Is this clear to all of you? Now when we use the term proliferative, whenever we use the term proliferative means, whenever we use the term proliferative, it means there's increase in cellularity. Okay. This can be membranoproliferative. Membranoproliferative would be increase in thickness of basement membrane as well as increase in the cellularity. You can have mesangial-proliferative when the mesangial cells start proliferating. So whenever the cellularity increases, we add on the term proliferative. Okay. So based on the morphological changes, we have different types of glomerulonephritis. So the first syndrome that I would tell you is minimal change disease. Okay, before that, the glomerular diseases are divided into 6 kind of syndromes. Glomerular disease is divided into 6 kinds of syndrome. What are these 6 syndromes? Either they can present as acute nephritic syndrome, some can present as nephrotic syndrome, some would present as the basement membrane syndrome, some would present as glomerular vascular syndrome, some are pulmonary renal syndrome, and last is infectious disease associated with infections associated with glomerulonephritis. Okay, so the glomerular disease can be of various types: nephritic syndrome, nephrotic syndrome, basement membrane syndromes, vascular syndromes, pulmonary renal syndromes, and those associated with infections. Okay, so examples of nephritic syndrome, conditions which are present with nephritic syndrome are post streptococcal glomerulonephritis, subacute bacterial endocarditis, lupus nephritis, SLE, ANCA associated, as we had seen in Wegener's microscopic polyangiitis, Churg-Strauss, then Henoch-Schönlein purpura, membranoproliferative glomerulonephritis, mesangioproliferative, essential cryoglobulinemia. All these conditions would present as nephritic syndrome. Okay, all these would present as nephritic syndrome. Yes or no? So causes for nephritic syndrome are all these: post streptococcal glomerulonephritis, subacute bacterial endocarditis, lupus nephritis, ANCA associated like Wegener's microscopic polyangiitis, Churg-Strauss, Henoch-Schönlein purpura, membranoproliferative, mesangioproliferative, and essential cryoglobulinemia. They are all causes for nephritic syndrome. Nephritic. Now when we talk of the nephrotic conditions which usually present with nephrotic syndrome are minimal change disease, FSGS, focal segmental glomerulosclerosis, membranous glomerulonephritis, diabetic nephropathy, amyloidosis, Fabry's disease. Okay, light chain deposition disease, multiple myeloma. Okay, so minimal change, FSGS, membranous glomerulonephritis, they would present with mostly nephrotic syndrome. PSGN, then membranoproliferative, mesangioproliferative, they would usually present with nephritic. So proliferative syndromes are more severe, they usually present with nephritic syndrome. Basement membrane syndromes would be anti-GBM antibody syndrome, or the good pastures, then Alport syndrome, thin basement membrane disease, nail patella syndrome. Okay. Now the glomerular vascular diseases. The glomerular vascular diseases examples are atherosclerotic nephritis, hypertensive nephropathy, hypertensive nephropathy, sickle cell disease, sickle cell disease, antiphospholipid antibody syndrome, Henoch-Schönlein purpura. Then ANCA associated. The ANCA associated one have the ANCA associated diseases, they're associated with both. Okay, they can present with nephritic syndrome, they can present as glomerular vascular syndrome. Okay, ANCA associated like Wegener's, MPA, Churg-Strauss. Then thrombotic microangiopathies. Examples of thrombotic microangiopathies: atherosclerotic nephropathy. Okay, because of atherosclerosis also you can have involvement of the vessels of the glomerulus, which can lead to glomerulopathy. Okay, so that can also be associated. Thrombotic microangiopathy diseases, microangiopathic diseases. Examples are hemolytic uremic syndrome and TTP, thrombotic thrombocytopenic purpura. Okay, so HUS, TTP, etc. Then infectious diseases, infections associated. All of you, have you written it down? Can I change it over to next page? Yes or no? Fine. Yes, infections. Infections associated with glomerular disease are PSGN, subacute bacterial endocarditis, HIV, hepatitis B, hepatitis C, syphilis, leprosy, malaria, schistosomiasis. Okay. Then pulmonary renal syndromes. They include good pastures, then ANCA associated like Wegener's, microscopic polyangiitis, Churg-Strauss, all of them. Then even Henoch-Schönlein purpura and essential cryoglobulinemia. They are said to be associated with both kidney and the pulmonary involvement. Okay, HSP and essential cryoglobulinemia. Okay, fine. So now the individual types of glomerular nephritis, the first would be minimal change disease. What is the other name for this condition? Minimal change disease. Minimal change disease. What are the other names for this condition? Minimal change disease other than this? Yes. The Nils disease or lipoid nephrosis. Okay, so Nil's disease and lipoid nephrosis are other name for this. Nil disease or minimal change is known as because light microscopy is absolutely normal. When you do light microscopy, it is absolutely normal. When you do electron microscopy, when you do immunofluorescence, that is also absolutely normal. And when you do electron microscopy, then the only thing that you see is effacement of the food processes, which was earlier given the name of fusion of food processes. So there's effacement of food processes. Okay, effacement of the food processes on electron microscopy. This is the only morphological change that you see. Otherwise the basement membrane, the mesangium is absolutely normal. Now, lipoid nephrosis. It is also known as lipoid nephrosis. Why? Because when too much of lipids, they are filtered. There's lipiduria. It is a nephrotic syndrome. So there's lipiduria because of the hyperlipidemia. So when too much of lipids are filtered, now PCT is always very greedy. Okay, so when too much of lipids are filtered, more and more of PCT reabsorption of lipids would occur. And these cells of proximal convoluted tubule, they appear loaded with lipids. They are loaded with lipids. Okay, so they appear loaded with lipids. So the proximal convoluted tubule cells, they appear loaded with lipids. So it is also known as lipoid nephrosis. Now, what are the causes for minimal change disease? What are the causes for minimal change disease? This minimal change disease would present to you with nephrotic syndrome, and this is the most common cause of nephrotic syndrome in children, where it accounts for approximately 80% of the patients. And what about adults? In adults, it accounts for 20% of the nephrotic syndrome. Okay, so 20% of the nephrotic syndrome that we have in adults is because of the minimal change disease. Now, what are the causes for minimal change disease? This minimal change disease can be primary. Mostly it is primary, that means idiopathic, you don't know why it is occurring. Mostly it is idiopathic, but in some it is associated with some secondary causes. And examples of secondary causes of this minimal change disease is Hodgkin's lymphoma, HIV, drugs like NSAIDs, rifampicin, interferons. Okay, so secondary causes are Hodgkin's lymphoma, HIV, drugs like NSAIDs, rifampicin, interferons. Okay. Now, second syndrome is membranous glomerulonephritis. Now, membranous glomerulonephritis is now considered to be the most common cause of nephrotic syndrome in adults. Earlier it was FSGS, but now the incidence of membranous is increasing. And why do we call it membranous? Because on light microscopy, this is a thick basement membrane. So most likely what kind of deposit would be seen? There would be subepithelial deposits. Okay, so whenever there's thick basement membrane, so deposits would be subepithelial. Okay, now nowadays it is considered to be the most common cause of nephrotic in adults. Earlier it was FSGS. Yes, so now what are the causes for membranous glomerulonephritis? Causes for membranous glomerulonephritis. It can again be sorry, age group. Yes, so what are the causes? Causes are, again, it can be primary or secondary. There's a long list of causes which are associated with membranous glomerulonephritis. For example, various infections which can lead to membranous glomerulonephritis are hepatitis B, hepatitis C, syphilis, malaria, schistosomiasis, leprosy, filariasis. Okay, so all these infections, most common cause of adults, young adults, not elderly. Okay, it is most commonly in young adults. Okay, membranous is most common cause of nephrotic in adults, not elderly. Okay, elderly still they consider it to be FSGS. Okay, adults, when we talk of adults, it is membranous. So first thing is infections, hepatitis B, C, syphilis, malaria, schistosomiasis, leprosy, filariasis. Then comes your various cancers like those of bronchial cancers or the lung cancers, colon cancers, stomach cancer. Okay, stomach cancer, then renal carcinoma, esophageal carcinoma, etc. So GI tract and lung. So GI cancers and lung cancers are important causes for then drugs associated with drugs associated with nephrotic syndrome, membranous type are gold, mercury, D-penicillamine, NSAID, probenecid. Okay. After that, autoimmune conditions. Autoimmune conditions like SLE, RA, primary biliary cirrhosis, Sjogren's, myasthenia gravis, dermatitis herpetiformis, then other conditions which are associated are sickle cell anemia, diabetes mellitus, Crohn's, GBS. Okay. Yes, so all of you have written it down. Yes. Okay, yes. So next is focal segmental glomerulosclerosis. FSGS. Okay. So FSGS was earlier considered to be the most common cause of Now is it fine? Am I audible now? Yes? Okay. So FSGS is now considered to be the most common cause of nephrotic syndrome in adults of African ancestry. Adults of African ancestry. Okay. Otherwise, if they only say adults, it is clear. Okay. Adults of African ancestry. Adults of African ancestry. Fine. Adults of African ancestry or in elderly individuals. Okay. Now, what are the causes for FSGS? What is FSGS? That you all understand, I hope. It is because of the sclerosis of some of the blood vessels. Okay. So, some blood vessels would undergo sclerosis. So, focal, segmental, glomerulosclerosis. So, there is glomerulosclerosis in some of the capillary loops. Okay. Some capillary loops will have sclerosis or fibrosis. Fine. Now, what are the causes for this? The causes can be, again, it can be primary or it can be primary or it can be secondary. Now, causes for secondary are various. For example, some infections, infections like HIV, hepatitis B, parvovirus infection. Okay, parvovirus, HIV, hepatitis B. Fine. Then one very important thing is reflux nephropathy. Okay, reflux nephropathy is associated with focal segmental glomerulosclerosis. So reflux nephropathy, then hypertensive nephropathy. Hypertensive nephropathy is also associated with FSGS. Is also associated with FSGS. Then drugs. Examples of drugs associated with FSGS are heroin, analgesics, bisphosphonate like pamidronate. Okay, heroin, analgesics, pamidronate, which is a bisphosphonate. Then Alports, other conditions like Alports, sickle cell disease, lymphomas, Alports, lymphoma, radiation nephritis. Then storage diseases like alpha galactosidase deficiency. That is your Fabry's disease. Fabry's disease. Okay, so these are all causes for FSGS. Chronic pyelonephritis I could not find. Okay, so I'm not sure if it is a cause of FSGS. Okay, so all these are causes for focal segmental glomerulosclerosis. Then membranoproliferative. Then is membranoproliferative. Now this membranoproliferative means there is thickness of the basal membrane which is increased. So diabetes is not mentioned as a cause. Diabetes was a cause of membranous. It was not associated as a, it is not mentioned as a cause of FSGS. This is from the Harrison. Okay. No, diabetes is not mentioned. Okay, diabetes is not there. Yes, so membranoproliferative. So membranoproliferative glomerulonephritis is further divided into three types: type 1, type 2, and type 3. And which of these is most common? Okay, don't worry. Questions I would tell you. Okay, let us just complete this part, then I will tell you some questions. Fine. MCQs I would tell you. Don't worry. Fine. Let us just complete off this theory part, then we will discuss the questions. So the most common type of membranoproliferative disease is type 1. It is not type 3. In RPGN it is type 3, but here it is type 1. Now, what are the causes for this type 1 membranoproliferative? In type 1 membranoproliferative, the causes are, it can be idiopathic, it can be because of the subacute bacterial endocarditis, it can be because of SLE, it can be because of hepatitis B, hepatitis C, and some cancers like lung cancer, ovarian cancer, and breast cancer. Okay. So idiopathic, SABE, SLE, hepatitis B, hepatitis C, and cancers like lung, ovarian, and breast cancer. Now type 2 is said to be associated with dense deposits, and the cause is either idiopathic or C3 nephritic factor associated. Then lastly, type 3 is again either idiopathic or that associated with complement receptor deficiency. Okay. Now in this type 1, what do you see on microscopy? What characteristic appearance is seen in type 1? In type 1, what characteristic appearance is described on microscopy? Yes, the tram track appearance. Okay. Now what is this tram track appearance? This tram track appearance is a double contour appearance. due to interpositioning of mesangial cells between basement membrane and endothelial cells. What do I mean by this? This is your capillary endothelium and here is your basement membrane. So some mesangial cells, they just come and get interposited in between. Okay, so some mesangial cells would get interposited in between and this would give the appearance of a railway track. So tram track appearance is a double contour appearance because of the interposition of mesangial cells between the basement membrane and the capillary endothelium. Okay, and this is the mesangial cell. Yes or no? Okay, so this is clear to all of you. So tram track appearance is because of the interposition of mesangial cells in between your endothelial cells and the basement membrane. Fine. Now next, what is RPGN? Yeah, so next we would do rapidly progressive glomerulonephritis. So am I audible to all of you? RPGN or crescentic glomerulonephritis. Now, when do we say it crescentic glomerulonephritis? What is the crescent? What are the diagnostic criteria for crescentic glomerulonephritis? Yes? Type 2, type 3, I've already told you. Type 2 is associated with dense deposit. The cause can be idiopathic or it can be associated with C3 nephritic factor associated disease, or type 3 is again idiopathic or that associated with complement receptor deficiency. Fine, only the causes are required. Now RPGN. Now RPGN or crescentic glomerulonephritis, it is basically because of formation of crescents in the Bowman's capsule. And from where is this crescent formed? The crescent is formed because of the proliferation of the parietal epithelial cells. The parietal epithelial cells, they start proliferating. The parietal epithelial cells, they start proliferating and this gives the appearance of a half moon. That is why we call it crescent. Okay, so the crescent is because of the proliferation of parietal epithelial cells. Parietal epithelial cells. Now the question is asked, what is important? Shape of crescent, number of crescent, size of crescent. Yes, what is important diagnostically? Shape of the crescent, number or size of the crescent. The number of the crescents are important prognostically. Number of crescents. Number of crescents. So more than 70% of the Bowman's capsule should be occupied by crescent. More than 70% of the Bowman's capsules would be occupied by crescents before we labeled it as crescentic glomerulonephritis, and it should be accompanied by a rapidly progressive renal failure. Okay, so number of crescents are more than 70% of Bowman's capsule is occupied by the crescents or rapidly progressive renal failure is associated, then we call it crescentic glomerulonephritis or rapidly progressive glomerulonephritis. Now, how many types of RPGN are there? Types of RPGN. Types of RPGN are again three types. It is again three types: type 1, type 2, and type 3. Now, this time the most common type is type 3. Okay, in membranoproliferative, the most common was type 1, but here it is most common type is type 3. Now, this type 1 is basically that associated with anti-GBM antibodies, so either anti-GBM antibody syndrome or good pastures or good pastures. Then type 2, or it can be idiopathic, Type 2 is associated with immune complex mediated disease. So examples of immune complex mediated disease are, as we discussed yesterday, Henoch-Schönlein purpura, polyarthritis nodosa, essential cryoglobulinemia. Others are SLE, IgA nephropathy, post-streptococcal glomerulonephritis, and lastly it can also be idiopathic. So first is anti-GBM antibody mediated, either anti-GBM antibody syndrome or good pastures or idiopathic. Second is immune complex mediated, as we have in Henoch-Schönlein purpura, PAN, essential cryoglobulinemia, SLE, IgA nephropathy, PSGN, idiopathic. And lastly, the most common type, which is actually the posse-immune RPGN. As I told you yesterday also, the posse-immune RPGN is the one which is usually associated with ANCA-mediated injury. So this is seen in conditions with ANCA-mediated injury, as we have in Wegener's microscopic polyangiitis, Churg-Strauss, and idiopathic crescentic glomerulonephritis. Okay, so these are all causes for posse-immune type 3 RPGN. Okay. Now, once a question was asked, all of the following except is associated with RPGN. Options were SLE, PAN, diabetes mellitus, Wegener's. So the answer was, yes, all of the following except is associated with RPGN. Options were SLE, PAN, diabetes mellitus, Wegener's, granulomatosis. So the answer was diabetes. So diabetes and Alport's. Even Alport's was asked once in the exam, the question. So the diabetes as well as Alport's, they are not associated with RPGN. They're not associated with RPGN. Okay, Alport's as well as diabetes. Now next is post-streptococcal glomerulonephritis. Now, I told you yesterday also, this is one of the DDs of hematuria following respiratory tract infection. Okay, so following respiratory tract infection, you can have hematuria, which can either be synpharyngitic, that is within 1 week, or it can be delayed, that is after 1 week. And for synpharyngitic, it is IgA nephropathy or Burgess disease, and for delayed, it is either Henoch-Schönlein purpura or post-streptococcal glomerulonephritis. Fine. So when we talk of post-streptococcal glomerulonephritis, you can have two kinds of streptococcal infection. You have two kinds of streptococcal infection, either upper respiratory tract infection or skin infections. The strains of streptococcus associated with upper respiratory tract infection are URTI. This is URTI. Are M1, 2, 4, 3, 25, 49. Okay. And usually after 1 to 3 weeks, after 1 to 3 weeks of upper respiratory tract infection, you get hematuria. As skin infection are associated with the higher strains like M47, 49, 55, 260, 57. Okay. And usually hematuria associated with skin infections starts after second week. Okay. Even after second week. Usually 2 to 6 weeks. Between 2 to 6. 2 to 6 weeks. Between 2 to 6 weeks. Here it is 1 to 3 weeks. Here it is 2 to 6 weeks. Now, upper respiratory tract infections, they are more common in winters. They are more common in winters, and skin infections, they are more common during summers. Okay, a question was asked once even on this. So, skin infections more often associated with hematuria during summers, and upper respiratory tract more often during winters. And this PSGN is classically due to molecular mimicry, which is because of the antibodies which are produced against antigens of these streptococcus. And which antigens of these streptococcus? SBEB, which is streptococcal pyrogenic exotoxin B, streptococcal pyrogenic exotoxin B, or another one is NAPLR, that is nephritis associated plasmin receptor. Okay, so the antibodies which are formed against these antigens of streptococcus, they cross react with your kidney and they are associated with glomerulonephritis leading to hematuria. Now, will a patient of PSGN have strep recurrence? No, the answer is no. Recurrence is rare. Usually they impart lifelong immunity. Lifelong immunity is usually seen. Okay, so usually the recurrence is not seen. On the other hand, membranoproliferative is mostly associated with nephritic. It is mostly associated with nephritic. Okay. Okay, and 50% would develop end-stage renal disease after 10 years, and 90% would develop renal failure after 20 years in membranoproliferative. RPGN is seen in 25%. Okay, RPGN in membranoproliferative is seen in 25%, but mostly they're present with nephritic syndrome. Okay, proteinuria, hematuria, etc. Okay. Yes, Masoor, they can present with RPGN in 25%, but nephritic is what is seen in most of them. Yes, so recurrence is rare and they're usually associated with lifelong immunity. On the other hand, patients with IgA nephropathy, they classically have as well as high chances of recurrence. Very often associated with recurrence. In Henoch-Schönlein purpura also, recurrence is there, but less than IgA nephropathy. Okay, here it is roughly around 40%. Okay, so 40% patients will have recurrence. Membranoproliferative would develop into end stage renal disease in 50% of the individuals after 10 years and in 90% of the individuals after 20 years. Okay, so they are associated with end stage renal disease. It is associated with a very poor outcome. Okay, membranoproliferative is associated with a bad outcome. Fine. So Hinong-Schollen purpura and IgA nephropathy, they're both associated with recurrence, which is much more common in IgA nephropathy, and HSP associated with recurrence only in 40%. Now lupus nephritis. Lupus nephritis, we did not do that. Delayed is associated with HSP and post-streptococcal glomerulonephritis. IgA nephropathy, it would start off with hematuria within 2 to 3 days of our upper respiratory tract infection. Okay. Yes, lupus nephritis. Lupus nephritis, the most important complication of SLE. Okay. Now, how many grades of lupus nephritis are there? How many grades of lupus nephritis are there? Grades. There are 6 grades. So, what is grade 1? Grade 1 is mesangial lupus nephritis. So, mesangial lupus nephritis is something like your minimal change. So, on light microscopy, everything is normal, but on immunofluorescence and electron microscopy, you have immune deposits in the mesangium. Okay, so immune deposits are present in the mesangium in type 1. Okay, in type 1. Now next is type 2, which is mesangioproliferative. So mesangioproliferative means when proliferation word is added, that means there is an increase in cellularity also. So here when you do light microscopy, there's increase in cellularity of mesangial cells, and on immunofluorescence and electron microscopy you would see immune deposits in mesangium plus the increased cellularity. Okay, so in type 1 only some immune deposits, in type 2 along with immune deposits some proliferation of mesangial cells is also occurring. Then is type 3, which is focal lupus nephritis. So since the term focal is there, so less than 50% of all the glomeruli are involved, but here in addition to the mesangial deposits they have subendothelial deposits. Subendothelial deposits. Okay, so subendothelial deposits are also there in addition to your mesangial deposits. Similarly, stage 4, which is diffuse lupus nephritis. So diffuse more than 50% of all the glomeruli, and again the kind of deposit that they have is subendothelial. Stage 5 is membranous lupus nephritis. Now since the term is membranous, even on light microscopy there is thick basement membrane, and what kind of deposits are appreciated? Subepithelial. Since it is membranous, the kind of deposit would be subepithelial. So there are subepithelial deposits with a thick basement membrane. Okay, subepithelial deposits. So here you have subepithelial deposit. Subepithelial. And stage 6 is stage of advanced fibrosis, end stage kidney disease. Advanced sclerosis or fibrosis. Okay. stage of advanced fibrosis. So these are the 6 stages. Is it clear to all of you? Yes? No confusion in these stages? Mesangial, mesangioproliferative, focal, diffuse, membranous, and advanced fibrosis. Fine? Yes, Shimla? Okay. Now next, if I ask you, which stages of lupus nephritis are associated with poor outcome or bad prognosis? Okay. Bad prognosis is associated with which stages of lupus nephritis? Stage 1, 2, 3, 4, 5, 6. Which of these stages are associated with bad prognosis? Bad prognosis, yes? Yes, mesangial deposits would be seen in all of them. Okay, mesangial would always remain. But in addition, they will have subendothelial, subepithelial also. Okay. The answer is third, fourth, and fifth. Third, fourth, and fifth are associated with active nephritic syndrome, acute nephritic syndrome, proteinuria, hematuria, etc. Okay, so they are all associated with bad prognosis, and if you do not start them on treatment, that is your steroids, then they would develop acute renal failure. What about stage 6? Stage 6 is stage of advanced fibrosis. Okay, it is not associated with any prognostic value. The patient has already developed end stage kidney disease, so even if you give them steroids or treatment, they will not come out of this renal failure. But in stage 3, 4, and 5, if you give them treatment, you can prevent end stage kidney disease from developing. Okay, so this is not associated with prognostic value. Okay, already end stage renal disease has developed. Okay, so stage 6 is not associated with prognostic value, so be very careful about it. Stage 1 and 2, they are both associated with poor prognostic value. Okay. Now, if I ask you what is the characteristic lesion of lupus nephritis, characteristic lesion of lupus nephritis on kidney biopsy, what is the characteristic lesion of lupus nephritis on kidney biopsy? Yes, wire loop lesion. What is this wire loop lesion? The wire loop lesions are basically indicative of subendothelial deposits. Okay. Yes. So wire loop lesions are basically indicative of the subendothelial deposits. Fine. Now, what is actually seen is, if this is your capillary endothelium, and when you do PAS positive staining, when you do the PAS staining, the subendothelial deposits which are immediately below your subendothelium, okay, in 3D it would be there on all these sides. So subendothelial deposits would appear as PAS positive thickening of the capillary endothelium. Okay, so wire loop lesions are basically PAS positive thickening of capillary endothelium. Okay, it would appear as if a wire is wound around a cord. Okay, so that is why the name given is the wire loop lesion. The wire loop lesion. Okay, see the past positive thickenings of the capillary endothelium wall is wire loop lesions. Now, question is asked. Sorry. Question is asked. Which stages of lupus nephritis are associated with wire loop lesions? Yes, wire loop lesions are most characteristic of which stage of lupus nephritis? Wherever we have subendothelial deposits. So they are most characteristic of stage 4, but you can also have them in stage 3. And whenever you have subepithelial deposits, they are also coming from above. So it might also be associated with some patients of stage 5. So whenever we have subepithelial deposits, they may also be associated with some little bit amount of subendothelial deposits. So you can also have them in patients with stage 5. So this is most characteristic of stage 4, then 3 and then 5. Yes, Jammu. Basically I just told you the wire loop lesions after this prognosis. Wire loop lesions. Okay, so wire loop lesions basically indicate subendothelial deposits. So they appear like past positive thickenings of capillary endothelium. They are just below the endothelium. It appears as if a wire is wounded around a capillary loop. Fine. And question is often asked, they are most characteristic of which stage? So most characteristically they are seen in stage 4, because there more than 50% will have subendothelial deposits. Then also in stage 3, and also some patients of stage 5 can have wire loop lesions. Okay, but also remember, wire loop lesions are associated with all these stages of bad prognosis. So all these stages have bad prognosis. So wire loop lesion, if it is present on kidney biopsy, that means it is associated with bad prognosis. So wire loop lesion, if it is present, that means it is associated with a bad prognosis. Okay, so lupus nephritis, is this clear to all of you? Okay. Yes. Now next, IgA nephropathy is same as your Henoch-Schönlein purpura. So there would be IgA containing aggregates in the mesangium. Okay. When we talk of IgA nephropathy or the Berger's disease, this is considered to be the most common glomerulonephritis worldwide. Okay, so the most common type of glomerulonephritis worldwide, we would discuss that, which recur after renal transplant and which do not recur. Okay, so most common glomerulonephritis worldwide is IgA nephropathy or Berger's disease. Fine. And the patient with Berger's disease, they classically present with recurrent hematuria in childhood. Okay, and on pathology, what do you have? You have IgA containing aggregates, IgA containing immune complexes, in mesangium. Okay, in the mesangium. So when you see on the microscopy, you have IgA containing aggregates in the mesangium. Fine. Now classical causes of recurrent hematuria in childhood. Recurrent hematuria in childhood. What are the causes or DDs of recurrent hematuria in childhood? What are the differential diagnoses other than IgA nephropathy? Yes. Causes of recurrent hematuria in childhood. Another is Alport's disease, thin basement membrane disease, then nephrolithiasis, Henoch-Schönlein purpura can also be there. Okay, so Alport's thin basement membrane disease, nephrolithiasis are the classical examples of recurrent hematuria. and hypercalcemia, which would also be associated with nephrolithiasis. Hypercalcemia. Okay. Now, what is Alport's disease? What is Alport's disease? Now, how many alpha chains are there in? How many alpha chains are there in collagen type 4? How many alpha chains are there in collagen type 4? 6 alpha chains. Okay. We have alpha 1, alpha 2, alpha 3, alpha 4, alpha 5, alpha 6. The gene for alpha 1, alpha 2 lies on chromosome number 6. Gene for alpha 3, alpha 4 lies on chromosome number 2, and the gene for alpha 5 and 6 lies on the X chromosome. Okay. So now this Alport syndrome is a defect of alpha 5, which is present on X chromosome, the gene for which is present on X chromosome. So it is a inherited X-linked recessive disease Since it is X-linked recessive disease, it would be more common in males or females. Yes, X-linked recessive disease. So more common in males or females? Yes, so it would be more commonly seen in males. So X-linked recessive disease with a defect of alpha 5 chain of collagen type 4. Okay, so alpha 5 chain of collagen 4 is defective in patients with Alport disease. Most common form is this inheritance. There are some other patterns of inheritance also. Some other defects are also there in alpha 3, alpha 4, etc. But most common is alpha 5. Now, what are the manifestations of Alport syndrome? The Alport syndrome can have they have a classical triad, which is some kidney involvement along with some extrarenal involvement. So kidney involvement is most often manifested by nephritic syndrome, is manifested by hematuria. Classical thing that we have here is hematuria. The classical symptom is that of hematuria, that too recurrent hematuria, and it occurs early in life. The patient would complain of hematuria. Proteinuria can be there, but that is usually a late feature. Proteinuria is there, but it is usually a late feature. Proteinuria develops later on. First hematuria is there, and then chronic renal failure is developed in most of them by the age of 20 years. Okay, by 20 years of age, most of them would develop chronic renal failure. Okay, now what about extra renal manifestations? The most common extra renal manifestation is sensory neural hearing loss. Most common out of this, overall the most common is kidney involvement. Most common is kidney involvement. And most common extra-renal manifestation is hearing loss. And this hearing loss would classically involve the higher tones. The higher tones are not required for day-to-day living. Okay, so the patient may not appreciate any hearing loss. Only when you do a audiogram, the patient would be detected of this hearing loss. So this higher tone sensory neural hearing loss is basically detected on audiogram. And according to Harrison, this is non-progressive. The hearing loss is non-progressive. Some say it is progressive, but Harrison says it is non-progressive. Okay. Now, lastly, the ocular manifestations, which include things like bilateral, often this is bilateral, lenticonus anterior, lenticonus. Then they can have a retinopathy, which is given the name of dot and fleck retinopathy. Dot and fleck retinopathy. Dot and fleck retinopathy. Dot and fleck retinopathy. And some other features that can be seen are mental retardation, leiomyomatosis. Fine. Dot and fleck retinopathy, anterior lenticonus. And then some can also have mental retardation, leiomyomatosis. Leiomyomatosis. Fine. And finally, what is the treatment for this Alport syndrome? Ultimate treatment would be to do renal transplant. Now, there are some set of diseases which recur after renal transplant. So, diseases associated with, questions have been asked on this, significant recurrence after renal transplant. Yes. So those which are associated with renal transplant, you would avoid doing a renal transplant in them. Okay. So diseases associated with recurrence after renal transplant are some kinds of glomerulonephritis and some metabolic diseases. So the glomerulonephritis which recur after renal transplant are membranoproliferative glomerulonephritis, especially type 2. That seen associated with dense deposits. Then IgA nephropathy. IgA nephropathy. Then anti-GBM antibody syndrome. Or good pastures. Then FSGS, hemolytic uremic syndrome. They are all associated with recurrence after renal transplant. Metabolic conditions like type 1 diabetes mellitus, amyloidosis, oxalosis, cystinosis. Okay. Fine. Now, what is thin basement membrane disease? So, this is clear to all of you. Fine. So, what is thin basement membrane disease? Where is the defect in patients with thin basement membrane disease? In thin basement membrane disease, there is again persistent or recurrent hematuria not associated with proteinuria, hypertension, or loss of renal function. Okay. So this thin basement membrane is a recurrent persistent hematuria not associated with proteinuria, hypertension or loss of renal functions. And it is usually a benign familial condition. Family history is there. It is a benign condition and the pattern of inheritance is autosomal dominant. Autosomal dominant, okay? And the defect is said to be there at both collagen 3 alpha 3 as well as alpha 4 chain, okay? So both alpha 3 alpha 4 chain of collagen type 4 are defective, okay? So if alpha 3 is asked, it is thin basement membrane disease. Alpha 4 is asked, then it is alpha 3 and alpha 4 both are defective. So alpha 3 is seen in both anti-GBM antibody syndrome or good pastures as well as thin basement membrane disease. Alpha 4 is with thin basement membrane disease. Alpha 5 is with Alport's disease. Okay. So if alpha 3, alpha 4, alpha 5, they ask you alpha 3 is with good pastures or anti-GBM antibody syndrome. Alpha 4 defect is in thin basement membrane disease and alpha 5 is Alport's. Okay. And when you did the biopsy on kidney biopsy, there is diffuse thinning of basement membrane. Diffuse thinning of basement membrane. What do you see in the kidney biopsy of this Alport syndrome? What do you see in the kidney biopsy of Alport syndrome? Yes? Kidney biopsy of basement membrane, thin basement membrane would show you diffuse thinning of basement membrane. In Alport syndrome, on kidney biopsy, you will have in Alport's on kidney biopsy, basket weave, I'm not sure, but Harrison says there is irregular thickening and thinning. Okay, so thickening and thinning both are seen. Thickening and thinning of basement membrane. Okay, the splitting of lamina, and microgranulations. Okay, irregular thickening and thinning of the basement membrane is seen. Okay, basket weave, I'm not sure if that is there. Now some vascular diseases of your kidney. Vascular disease of kidney like renal vein thrombosis. I would just tell you few points regarding renal vein thrombosis, some MCQs. Renal vein thrombosis. Okay, renal vein thrombosis. What is the most common cause of renal vein thrombosis? Yes, this renal vein thrombosis can be primary when there's no cause other, or it can be secondary because of some secondary diseases. Yes, primary when we talk of renal vein thrombosis. The most common cause of primary renal vein thrombosis. Most common cause. It is different for children and adults. Thin basement membrane disease usually does not require any treatment. Okay, usually it is a benign condition, they do not require any treatment. Okay, in children and in adults. Yes, so most common cause of renal vein thrombosis in children and adults. Yes, in children and adults. In children it is dehydration, and in adults it is nephrotic syndrome. And which kind of nephrotic syndrome is most often associated with renal vein thrombosis? The membranous glomerulonephritis. Okay, so dehydration in children and in adults, nephrotic syndrome. Some secondary causes associated with renal vein thrombosis are trauma, external compression like that due to lymph node, some tumor, some aortic aneurysm. Then involvement by the renal cell carcinoma, RCC, pregnancy, etc. or use of some OCPs can also be associated. Okay, so they are all causes for secondary renal vein thrombosis. Now, how do they present patients with renal vein thrombosis? Clinical features would be flank pain, tenderness, hematuria, rapid decrease in renal functions, proteinuria. Which sided renal vein thrombosis is more often? Right or left? Which side is more often associated with renal vein thrombosis? Left is more common than right. Okay, left is more common than right. So left is more common than right. Some other causes for renal vein thrombosis are, other than membranous was the most common. Others are membranoproliferative glomerulonephritis, amyloidosis, etc. Okay. Why left? Even I don't know why left sided, but left is more often involved than the right side. And diagnosis, how do you diagnose renal vein thrombosis? When we talk of the diagnosis, the best screening test is Doppler ultrasound, and the most sensitive is CT angiography. Okay. Okay. So left sided renal vein has a 90-degree attachment. Fine. So best screening test is Doppler, most sensitive is CT angio. Now what is microangiopathic anemia? What are the types of microangiopathic anemias? Types of microangiopathic anemias. What do you mean by microangiopathic anemias? Okay. Examples of microangiopathic anemias are hemolytic uremic syndrome and TTP, micro angiopathic anemias. Okay, the example of these are hemolytic uremic syndrome and TTP, HUS and TTP. Fine. Now, HUS and HUS and TTP, they are both diseases of the same spectrum, but there are some differences between them, and sometimes we are not able to differentiate between them. Then we call it HUS TTP overlap syndrome. Okay. So first, let's discuss what is HUS, hemolytic uremic syndrome. Now, HUS is a disease which is classically characterized by the triad of anemia, bleeding because of thrombocytopenia and renal failure or decreased urinary output. Okay, so the classical triad is anemia, bleeding with renal failure. Anemia, bleeding, and renal failure. Treatment of renal vein thrombosis would obviously be, you have to treat the underlying cause. If it is dehydration, treat that. If it is glomerulonephritis, treat that. And otherwise, you have to give them anticoagulants. And you may also need to give them vena cava filters. Okay, vena cava filters are also required so that you don't have any pulmonary embolism. Okay, so it would depend on the cause. Along with anticoagulation, you have to treat for the cause, dehydration or the underlying cause. If it is trauma, if it is pregnancy, so depending on the cause, you have to treat renal vein thrombosis other than anticoagulants and filters of the vena cava, so as to prevent the migration of the thrombus. Fine. So the classical triad in HUS is anemia, bleeding and renal failure. Anemia, bleeding and renal failure. Now, why is there HUS? What happens in HUS? What is the pathogenesis of hemolytic uremic syndrome? Pathogenesis of hemolytic uremic syndrome. This HUS usually occurs after a diarrheal episode, diarrhea caused by which strains? Diarrhea caused by which strain? Yes, diarrhea caused by diarrhea due to infection of E. coli O157H7 or Shigella Okay, so E. coli O157H7 or Shigella. Now what happens is, these organisms, they produce a toxin which is known as Shiga toxin or virotoxin. They produce a toxin known as Shiga toxin or the virotoxin. And this toxin, what it does is, it damages the endothelium. The Shiga toxin, it damages the endothelial cells, and when endothelial cells, they are damaged, what happens is, they would attract their phospholipids, their basal membrane would be exposed, and they would attract the platelets to get deposited. So this would lead to platelet aggregation. Okay, and when more and more of platelet aggregates are there, so what would happen? More and more of platelets would be used in formation of this thrombus. Okay, so what would happen? The platelets in the blood would go down. So this would be associated with thrombocytopenia. Less of platelets in the blood, because more and more of platelets are being used in platelet aggregation to form thrombus at the site of endothelial damage. And the most important organ where you have this endothelial damage is the kidney. This endothelial damage is most commonly seen in renal capillaries. So in renal capillaries, they are the most common site of this endothelial damage with platelet aggregation. Fine. Can I change this page? Yes or no? This is clear to all of you? The first thing is infection by diarrheal agents like E. coli, Shigella, which produce this toxin which damage the endothelial cells and leads to platelet aggregation. So, when platelets aggregate, when platelets aggregate, they attach even with fibrin, this formation of thrombus, and this is most commonly seen in renal vessels, most commonly seen in renal vessels. And when there are thrombi at various places, the thrombus, they will have a mechanical destruction, they would do the mechanical destruction of your RBCs, okay? They would lead to mechanical obstruction to the flow of RBCs, so mechanical destruction of RBCs is seen, which is given the name of microangiopathic. Microangiopathic means when there is some obstruction to the flow of RBCs which is leading to their mechanical destruction. So microangiopathic hemolytic anemia. Hemolytic anemia. Sometimes hemolytic anemias are due to antibody mediated when it is Coombs positive. Okay, here it is microangiopathic hemolytic anemia. Here it is microangiopathic because of the mechanical destruction we have hemolytic anemia. So what would happen to the Coombs test here? Coombs test. Coombs test would be positive or negative here? Yes or no? What about Coombs test? Coombs test. Yes, Coombs test will be negative. So this is Coombs negative hemolytic anemia. Okay, this is Coombs negative because it is not an antibody mediated destruction of RBCs. It is simply because of the mechanical destruction because of the thrombus in various vessels that we are having a hemolytic anemia, given the name of microangiopathic hemolytic anemia. Okay. Now since it is hemolysis, which enzyme in the blood would be more? LDH. Okay, so LDH is increased in the bloods. What about prothrombin time and APTT? PT and APTT. PT and APTT. Is there any effect on PT and APTT? The coagulation profile, yes or no? It is normal. PT and APTT, they are both normal because we have not affected the coagulation pathway. Okay, they are not affected. Only the platelets are affected. So PT and APTT, they remain normal. They remain normal. So what about DIC? Is it associated with any DIC? No. There's no DIC. Okay. There's no association with DIC. The coagulation pathway is not stimulated. No DIC. PT, APTT are normal. LDH is raised and Coombs test is negative. Is this feature clear to all of you? Because all these features are asked in the exam. So negative Coombs test, raised LDH, PT, APTT normal, no DIC. Okay. Now, how do you treat them? How do you treat them? What is the treatment for HUS? Bleeding time can be increased because the platelets are involved. Okay, bleeding time is a function of the platelets. So when there's thrombocytopenia, bleeding time can be increased. Okay, Jabalpur, fine. Because bleeding time is a function of platelets. So when there's thrombocytopenia, bleeding time would be increased. Fine. So treatment is usually supportive. You have to manage the renal failure and usually it is considered to be self-limiting disease. Okay, self-limiting disease. Okay, so treatment is supportive. If anemia is there, give blood. If thrombocytopenia is too much, give them platelets, manage the renal failure. What about antibiotics? What about antibiotics? Do we give them antibiotics? Yes, role of antibiotics in, it is because of the infection. So do we give antibiotics? Yes or no? If no, why? Bareilly. Yes. Why do we give them antibiotics? We should give them. Yes or no? The answer is no, because no role of antibiotics. Because if you give them antibiotics, there would be increase in the release of toxins. Yes, they would increase the release of toxins, so it might deteriorate the patient. Okay, it might lead to deterioration of the patient. So antibiotics have no role in the management of HUS. Fine. Now next is TTP, thrombotic thrombocytopenic purpura, TTP. Yes, let's do TTP, then I would take a break. Okay. Thrombocytopenic purpura. Now, in this kind of microangiopathic anemia, along with microangiopathic anemia, thrombocytopenia, and renal failure, the classical triad seen in HUS, what else is there? What else is there? Yes, what else is there? Most common organ involving sepsis in HUS. Sorry, I did not get that question. Most common organ involving sepsis. What do you mean by that? Sepsis is multi-system, okay? Sepsis is in the blood. So, most common organ, renal, kidneys, otherwise most commonly involved organ, but organism most common overall is E. coli. Most common organism is E. coli. And in India, we consider it to be Shigella dysentery type 1, okay? Okay. So for HUS, E. coli is considered to be the most common organism worldwide, and in India it is Shigella dysentery type 1, which is considered to be the most common organism in India. Okay, so E. coli is the most common organism worldwide, and Shigella dysentery type 1 is the most common organism in India. Okay. Fine, Jammu, I could not get that question. Most common organ involving sepsis in HUS. But organisms, you write it down, please. Okay, so E. coli, most common worldwide. Okay. Worldwide, it is most common E. coli. In India, most common is Shigella dysentery type 1. Kidney is the most common organ involving the thrombi formation. Sepsis, I don't know, what do you mean by sepsis involving most common organ involving sepsis? No idea, okay. Yes, so the classical triad of HUS is there in TTP, and along with that, what is the pentad? What else is there? Along with this, there are two other features. There are two other features which are the neurological complications and the fever. Okay, so fever and neurological complications. Steroid in HUS is also not recommended. Okay, you may give steroids in various places. Sepsis, you may give, you may not give. So steroids are controversial, though do not have any proven role. Okay, management of HUS is basically supportive. You have to manage the renal failure, you have to look for the thrombocytopenia, you have to treat for the anemia. Okay, steroids also do not have a proven role. Okay, you may give steroids in n number of places, but they are not a recommended thing in HUS. So neurological complications and fever are the additional components of the pentad that we have in TTP. But to be very true, various times even patients of HUS can have fever and neurological complaints. So sometimes we are not able to differentiate between them. So we give the term HUS TTP overlap. HUS TTP overlap. Okay, sometimes this term is also used. HUS TTP overlap. Now, HUS was because of some infections. What is the neurological complications? Which neurological complications are not mentioned? I don't know which one. Okay. So, what is the cause for this TTP? The cause for TTP is defect of ADAMTS13. What is this? This is a von Willebrand cleaving factor. This is von Willebrand factor cleaving protein. Okay, it disintegrates von Willebrand factor. So when there is a defect of this ADAMTS13, the full form is A disintegrin and metalloproteinase with thrombospondin type 1 motif of member 13. Okay, so this ADAMTS13, which is a von Willebrand factor cleaving protein, is defective, or it is mutated. So this leads to TTP. This leads to TTP. Okay. Now this TTP is classically seen in children, whereas HUS is more often seen in adults. Okay. The full form is A disintegrin and metalloproteinase with a thrombospondin type 1 motif member 13. Okay, so A disintegrin and metalloproteinase with a thrombospondin type 1 motif member 13 is ADAM-TS13. Okay, so when this is defective, we have TTP. This TTP is more often seen in children, whereas HUS is more often seen in adults. Okay. Okay, so this was asked in your NEET exam. A disintegrin and metalloproteinase with a thrombospondin type 1 motif member 13. So just to summarize the differences between HUS and TTP. Here there was the classical triad, here it is the pentad, but sometimes it is overlap. Classically seen in age group of 30 to 40 years. This is classically seen in children less than 5 years. Now one important thing is plasma exchange. Sorry, I'm so sorry. This is opposite. This is seen in less than 5 years and this is seen in 30 to 40 years. Sorry, please make that change. Okay. HUS is classically seen in children and TTP is seen in adults. I'm so sorry for that. HUS is seen in children, so please change that even on the previous page. Okay. So HUS is seen in children, TTP is seen in adults. Then plasma exchange is classically recommended for, plasma exchange is classically recommended for TTP, is classically done in patients with TTP. It is not effective, considered not effective to be patients in patients with HUS. Okay, even I am tired now, I need a break. So HUS TTP, just one question I would ask you, just before we take a break, just one MCQ I would like to ask you. Yes, a 10-year-old child, yes, MCQ, a 10-year-old child comes to you with complaints of hematuria for 2 days, for 2 days after an episode of diarrhea. After an episode of diarrhea. On examination, the blood picture is suggestive of fragmentation of RBCs, and thrombocytopenia. Ultrasound is suggestive of kidney enlargement. Bilateral. What is the most likely diagnosis? Options are DIC, HUS, renal vein thrombosis. Yes. So what is the diagnosis? Yes. The diagnosis is renal vein thrombosis. The diagnosis is renal vein thrombosis. It is not HUS or HSP. Okay. Even if HSP was given here, the answer was renal vein thrombosis. Okay. The patient is coming to you after diarrhea. The blood picture looks like that of HUS, but the clue which indicates renal vein thrombosis is bilateral kidney enlargement. Okay, the kidneys in a patient with HUS, they are not enlarged. Okay, this decrease in the blood flow to your they are not enlarged. Okay, kidney enlargement is classically a feature of renal vein thrombosis. The most common cause of renal vein thrombosis in a child was dehydration. So after diarrhea, because of dehydration, they can develop renal vein thrombosis. So this kidney enlargement indicates renal vein thrombosis, not HUS. In HUS, the kidneys are not increased in size, whereas in renal thrombosis, there is increase in the size of kidneys because of the back pressure. There's no drainage of blood from the kidneys, so because of the back pressure, there's enlargement of the kidneys. So in renal vein thrombosis, there's bilateral kidney enlargement, whereas in HUS or HSP, this is not what is seen. Is this clear to all of you? Because of the backflow. Okay, when renal vein thrombosis is there, outflow is not coming in, so because of the backflow, there is kidney enlargement. Because of the backflow, okay, because of the back pressure of the blood which is not being drained, there is kidney enlargement. Yes, Bikaner, okay. Yes, 10 minutes of break now, fine. Renal vein thrombosis is most often bilateral. 2/3 of the patients will have bilateral. Renal vein thrombosis is bilateral in break. Just a second. Renal vein thrombosis is bilateral in 2/3. So almost 60% to 70% are associated with bilateral renal vein thrombosis. So bilateral. This is bilateral in 2/3 patients. Renal vein thrombosis bilateral in 2/3 patients. Okay. Yes, so 10 minutes of break. Nephrogenic causes are drugs like, most important is mutation of the V2 receptor, then drugs like, aminoglycosides, amphotericin B, lithium, demiclocycline, phoscarnate, methoxyflurane. Yes or no? So drugs associated with nephrogenic diabetes insipidus are all of them. एक बार notepad refresh कर देना. Notepad refresh. Okay. So mutations of the V2 receptor, drugs like aminoglycoside, amphotericin B, lithium, demiclocycline, phoscarnate, methoxyflurane, then metabolic causes like hypercalcemia, hypercalciuria, and hypokalemia. More of calcium, less of potassium, okay, in the blood is associated with nephrogenic diabetes insipidus. So these are all causes for nephrogenic diabetes insipidus. For lithium induced nephrogenic diabetes insipidus, the treatment is amiloride, which is a sodium channel blocker, because lithium uses the sodium channels to enter the cells of the distal tubule. Okay, Vishakhapatnam, what else do you want me to ask? Yes? What else do you want to ask? Yes? Differences of what? Differences of? The primary causes are mostly associated with bilateral, okay, and the extrinsic causes, depending on the site, if it is only RCC of one side, it may be associated with unilateral, it may be otherwise bilateral. Yes, we would see that if we can take a break. Break. एक चाय मंगा दो प्लीज. Here and chronic renal failure. Fine. पेन दूसरा दे लो. HIV associated nephropathy. It is given the name of HIV-AN. HIV associated nephropathy. Fine. Now this is mostly associated with FSGS, focal segmental glomerulosclerosis. But the classical nephropathy that we have is associated with collapsing glomerulopathy. Okay, collapsing glomerulopathy is specifically used in respect to HIV. Okay, in which there is collapse of the glomerular tuft, collapse of glomerular tuft, okay. Yes, with HIV, in HIV we have HIV associated nephropathy. So now is it fine to all of you? Okay. Most common type that we see here is FSGS. HIV is most often associated with focal segmental glomerulosclerosis. The other kind of nephropathy that we have is collapsing glomerulopathy. Collapsing glomerulopathy is sometimes seen in patients with HIV, where there is collapse of the glomerulus, and associated proliferation and hypertrophy, hyperplasia of the visceral epithelial cells. and it is associated with a very poor prognosis, associated with a very bad prognosis. Okay, this is very bad. If collapsing nephropathy is there, it is associated with a very bad prognosis. Okay, HIV associated nephropathy. Fine. Other lesions which you can have in HIV are membranoproliferative glomerulonephritis. You can have minimal change disease. Okay, you can also have HIV associated TTP is also reported. Okay, okay, fine. So now is it fine to all of you? Everyone is connected. So when we talk of HIV, the most common type of glomerulopathy that we have in HIV is FSGS, and one specific type that we see in HIV is collapsing glomerulopathy, in which there is collapse of the glomerulus with proliferation and hyperplasia of the visceral epithelial cells, and this is associated with a very bad prognosis. The other kind of glomerulonephritis which we can have in HIV is membranoproliferative minimal change, HIV associated TTP can be there. Then diffuse proliferative glomerulonephritis is also seen. Okay, so all these kind of glomerulonephritis can be seen in patients with HIV. Okay, now what about hepatitis C, hepatitis B, and hepatitis C? Hepatitis C. In hepatitis C, you have various types of glomerulonephritis, and the most common is in hepatitis C, the most common variant is that of essential cryoglobulinemia, which is followed by membranous glomerulonephritis, then type 1 membranoproliferative glomerulonephritis. Okay, so the most common okay. Jammu, what did you not copy? HIV. HIV was FSGS, collapsing glomerulopathy, membranoproliferative, diffuse proliferative glomerulonephritis, minimal change, and HIV associated TTP. HIV associated TTP. Is that fine, or you want me to show you the slide again? Okay, or we can continue now. We would show you the slide at the end. Okay, in hepatitis C we can have all these things. Most common is essential cryoglobulinemia, then then membranous glomerulonephritis, then type 1 membranoproliferative glomerulonephritis. When we talk of hepatitis B, in hepatitis B you can have membranous glomerulonephritis. You can also have membranoproliferative glomerulonephritis, this is more common in children. With hepatitis B, this is slightly more common in patients who are adults. Okay, fine. And in some patients, PAN can also be associated with hepatitis B and then the glomerulonephritis. Okay, so HIV, hepatitis B, hepatitis C, I hope that is clear to all of you. When we talk of the diabetic nephropathy, what is the earliest manifestation of diabetic nephropathy? Earliest manifestation of diabetic nephropathy, the earliest change that you see in a patient with diabetes in the glomerulus. Yes, the earliest change that we see in diabetes. No, on microscopy, what is the earliest change that we see? Earliest time, yes, thick basement membrane. Thickness of the basement membrane is increased, which is the earliest change. So in diabetic nephropathy, what you need to remember is the first is the earliest change that is seen. The earliest change that is seen is thickening of basement membrane. Okay, thickening of basement membrane. Membranous glomerulonephritis is the most common that is seen here. Membranous glomerulonephritis. Thickening of basement membrane is the earliest, but what is the most common? Most common change which is seen is diffuse glomerulosclerosis. Is diffuse glomerulosclerosis. Okay, diffuse glomerulosclerosis. This is not focal segmental, it is diffuse glomerulosclerosis. This is the most common. Earliest is thickening of basement membrane. And what is most specific? Most specific to diabetes, that is nodular glomerulosclerosis or Kimmelston-Wilson disease. Most specific is nodular glomerulosclerosis or Kimmelston Wilson disease. Okay. So earliest change is thickening of basement membrane. Most common is diffuse glomerulosclerosis, but most specific is Kimmelston-Wilson nodule or nodular glomerulosclerosis. Okay. Kimmelston. Kimmel-Steele. Okay. Kimmel-Steele. Yes, don't worry, I would tell that. When should we screen for? Screening for diabetes should always be done at the time of diagnosis. Okay, first and most important is at the time of diagnosis. At diagnosis in both type 1 and type 2 diabetes. In both of them, first you should do it after, at the time of diagnosis. Then in type 1, that is for nephropathy. Type 1 after every 5 years, and in type 2 every year. This is for retinopathy. I'm slightly confused for this. This type 1 every 5 years, type 2 every year. This is for retinopathy. Nephropathy. At diagnosis, definitely we have to do. And what test do we do for diagnosis? Is microalbuminuria. Microalbuminuria. And how do we look for microalbuminuria? What is the value? The range is 30 to 300 milligram per day of microalbumin in the urine. Okay. Now, what is the better marker other than the urine microalbumin? For this, you have to take a 24-hour urinary protein. This is per day. So, better marker is in the spot urine, you look for albumin to creatinine ratio. Albumin to creatinine ratio. This is known as ACR. Albumin creatinine ratio. Okay. And the value for this is also 30 to 300 microgram per milligram of creatinine. Okay. So 30 to 300 microgram of albumin per milligram of creatinine in the urine. Spot urine. Okay, so this is a better marker because here only one sample is what is being taken. Okay. Now before that, let me just complete this slight query with this. 5 years and 1 year. Type 1, type 2. That is, I guess, for retinopathy. Every 5 years for type 1 and every year for type 2. Okay, just go back and check this. Okay. Yes, now what other things can you have in the microscopy other than thickening of basement membrane, diffuse glomerulosclerosis, nodular glomerulosclerosis? Two other things can be there, which are capsular drop and fibrin caps. Okay, capsular drop and fibrin caps. capsular drop and fibrin caps are also seen in diabetic nephropathy. Capsular drop and fibrin caps. What are capsular drops? They are eosinophilic thickenings of the parietal layer of epithelium. Eosinophilic thickening of parietal epithelium. So if this was your parietal epithelium, there's a eosinophilic red color thickening in the parietal epithelium. So it would appear as if there's a drop in the capsule, Bowman's capsule. So capsular drop is eosinophilic thickening of the parietal epithelium. And what is fibrinous cap? Fibrin cap? This fibrin cap is homogeneous eosinophilic material on wall of capillaries. Okay, this is on the wall of capillaries. So fibrin cap is homogeneous eosinophilic material on the wall of capillaries, and capsular drop is in the parietal epithelium of your Bowman's capsule. Is this clear to all of you? Diabetic nephropathy, nephropathy, the lesion seen in diabetic nephropathy, yes or no? Okay, I hope there's no confusion in this. So spot urine albumin test is the better marker. And why is it important to diagnose diabetic retinopathy at the stage of microalbuminuria? Why should we diagnose it at the stage of microalbuminuria as early as possible? Why? Because we have drugs which can reverse this diabetic retinopathy, and that is your ACE inhibitors. ACE inhibitors can reverse diabetic retinopathy. So you must diagnose it as early as possible and treat them. Because if you diagnose it at later stages, now what are the contraindications of giving ACE inhibitors? One important contraindication of giving ACE inhibitors is at higher values of creatinine, the chances of hyperkalemia. A side effect of ACE inhibitor is hyperkalemia. Okay, so when you give ACE inhibitors at later stages of diabetic retinopathy, chances of hyperkalemia are more, so they are usually not advised for use. Okay, once the creatinine goes above 2.5, chances of hyperkalemia increases, so you do not give ACE inhibitors after that range of creatinine. So it is important for you to diagnose diabetic retinopathy as early as possible and start them on treatment. Fine. Okay, so now can I move further with renal failure? Stepwise is, first is thickening of basement membrane, then a diffuse glomerulosclerosis is there, and on top of this diffuse you have nodular, on top of this you have nodular. Okay, so this is almost stepwise. First thickening of basement membrane, then diffuse glomerulosclerosis, then nodular. On top of diffuse, the nodules would develop. Okay, nodules are not in isolation, they are on top of the diffuse. But remember, focal sclerosis is not a feature of diabetes. Focal sclerosis is not a feature of diabetes. Okay, इस पे question आया हुआ है. They've asked this question: which of the following is not seen in diabetes? Diffuse glomerulosclerosis, nodular glomerulosclerosis, focal sclerosis. So the answer was focal sclerosis. Focal sclerosis is not a feature of diabetes. Okay, fine to all of you. Yes, so now what is acute renal failure? Acute renal failure. This term acute renal failure is not used nowadays. The term used now is AKI, acute kidney injury. Okay, acute kidney injury. So acute kidney injury is the term that we use nowadays, not acute renal failure. Okay, now how do you define acute kidney injury or ARF? How do we define that? The definition of acute kidney injury is rise in the serum creatinine level by at least zero point 0.3 milligram per deciliter or more than 50% higher than the baseline value in 24 to 48 hours. So in a time period of 24 to 48 hours, if the creatinine values rise above by at least zero point 0.3 milligrams per deciliter or more than 50% from the baseline, then it is known as acute kidney injury. Or if there's a decrease in the urine output to less than 0.5 ml per kg body weight per hour for at least more than 6 hours. Okay, so for more than 6 hours, if the urine output is decreased to less than this value or there's a rise in creatinine, this is how we diagnose acute renal failure. This is how we diagnose acute renal failure. Okay, fine. So now what are the types of acute renal failure? This acute renal failure can be divided into 3 types. It can be pre-renal, renal or post-renal. Okay, it can be pre-renal, renal or post-renal. Now what are the causes for pre-renal? Pre-renal, refill criteria in AKI. Sorry, I'm not aware of them. Refill criteria, fine. Now pre-renal failure is basically because of the decrease in the blood flow to your kidneys. Okay, the hypovolemia. So the causes for pre-renal are hypovolemia, which can be because of diarrhea, vomiting or any kind of fluid loss, blood loss. Then stages of low cardiac output, renal failure, cardiac failure, heart failure, low cardiac output. So now is it audible to all of you? Okay. So pre-renal failure can be because of all these things: hypovolemia, low cardiac output, or renal vascular occlusion, as we have in HUS, TTP, vasculitis, malignant hypertension. Okay. Now after this we have renal causes. The renal causes are further divided into ischemic causes, septic causes, sepsis, and toxins. The intrinsic renal failure is further divided into ischemic type, that associated with sepsis, and lastly the toxins. Ischemic type is usually when the pre-renal failure is not treated and it keeps on going on. Okay, so when this pre-renal failure, which is usually considered to be reversible, now when this moves on further, it becomes irreversible, and now it becomes intrinsic renal failure. Okay, by the time it is pre-renal, it is usually considered to be reversible. If you reverse the obstruction to the blood flow, or you correct the hypovolemia, it would reverse. But when this pre-renal becomes irreversible, there's ischemic change to the renal parenchyma, it is known as ischemic type of intrinsic renal failure. Then sepsis can cause intrinsic renal failure, and lastly toxins. The toxins can further be divided into endogenous and exogenous toxins. They can be further divided into endogenous and exogenous toxins. Now, what are the endogenous toxins? Endogenous toxins are the rhabdomyolysis, then in patients with hemolysis, uric acid, then multiple myeloma, the light chains, okay, so they are all examples of endogenous toxins: rhabdomyolysis, hemolysis, uric acid, multiple myeloma associated with deposition of light chains. Then exogenous toxins, examples of exogenous toxins are, most important is contrast. So contrast is a very important cause of nephropathy nowadays, which is given the name of contrast induced nephropathy, CIN. And what is the most important treatment for CIN, or how can you prevent CIN? Contrast are nowadays being given to so many patients for investigations. Okay, so you have to be very careful before you give contrast to a patient, and the most important prevention is hydration. Okay, so once you have once you maintain proper hydration, you prevent development of CIN. Okay, so hydration is very important. Then it can be because of some drugs like cyclosporin, antibiotics like aminoglycosides, amphotericin B, then cisplatin. Preloading with steroids may not help. Hydration is what is most important. Okay, hydration is most important. Can I write it on the next page? Yes. Can I write it on the next page? Yes. So exogenous toxins, when we talk of, the most important is contrast. Exogenous, most important was contrast induced nephropathy, for which you need to have proper hydration. Then drugs like cyclosporin, antibiotics like aminoglycosides, amphotericin B, then drugs like cisplatin, ethylene glycol, paracetamol, and various others. Okay, so they are all associated with NSAIDs, heavy metals. Fine. So they are all exogenous toxins, fine. And lastly is post-renal, which is basically because of the obstructive uropathy. Post-renal is basically your obstructive uropathy. Now, so acute renal failure will have three phases. ARF will have three phases, and they are initiation phase, maintenance phase, and last is recovery phase. Okay, so ARF has three phases. Most common is pre-renal. Yes, most common cause of ARF is considered to be pre-renal failure. So initiation phase, maintenance phase, and recovery phase. In maintenance phase, usually the patient will have oliguria, whereas in the recovery phase, the patient is usually having diuresis. That is the diuretic phase. Okay, so maintenance phase is associated with oliguria, recovery phase is associated with, yes, your diuretic phase. Now, what are the complications of acute renal failure? Complications of acute renal failure. Complications of acute renal failure can be because of the uremia, because of the electrolyte imbalance, because of the metabolic acidosis. Okay, so what kind of electrolyte imbalance do we have? You can have bleeding diathesis. Yes, electrolyte imbalance. What is the most important electrolyte imbalance when we talk of? Yes, the most important is hyperkalemia. Why? Because potassium is a ion which is completely or almost, which is only regulated via the kidney. Okay, the regulation of potassium mainly occurs via the kidney. The only site of potassium secretion is kidney. Sodium can be lost from the skin, by the sweat glands, from the GI tract, but 90%, more than 90% of your potassium is being excreted by your kidneys. So potassium regulation is most importantly occurring from kidneys. So whenever there's renal failure, the first thing and the most important thing to go up is your potassium. Then similarly, other things whose excretion would decrease are phosphate, magnesium, so they would all be increased in the blood. Now what about sodium? What about sodium? Sodium is decreased because there's no sodium reabsorption in the kidneys. Okay, sodium is not being reabsorbed, plus not reabsorbed, plus there is water retention because that is also not being excreted in by the kidneys. So sodium is not reabsorbed, water is not excreted. So because of this, there's volume overload, there's hyponatremia. Volume overload, hyponatremia. Then what about calcium? Calcium is usually on the lower side. Why? Because decrease in the synthesis of active vitamin D in kidneys, plus this calcium is utilized to combine with phosphate because there's more and more of phosphorus. So calcium combines with phosphate to form calcium phosphate stones. So there is decrease in the serum calcium levels. So calcium and calcium and sodium, they are on the lower side. Potassium, phosphate, magnesium are on the higher side. Okay. Now, is there any biomarker for acute renal failure? Biomarker for acute renal failure. Do we have any biomarker for acute renal failure? Yes. The biomarker for acute renal failure. Yes, biomarkers for acute renal failure are KIM1, which is kidney injury molecule, kidney injury molecule. So KIM1, which is produced by the PCT, and similarly NGAL, which is neutrophil gelatin associated lipocalin. Okay, so NGAL and KIM1, they are the biomarkers. This is also produced from the PCT when they're injured. So KIM1, NGL1 can be used as biomarkers for AKI. Now, what are the causes for preserved urinary output in AKI? Actually, water is not being excreted by the kidney. When kidney is not functioning, there's oliguria. Okay, so water retention is happening. Fine, water is being retained back. And similarly, sodium is also not being reabsorbed. Whatever, if it is filtered, it is basically because of the sodium, the water retention, that there's hyponatremia. Okay, hyponatremia, the most important cause is water retention. Okay, moreover, there's no sodium reabsorption. Whatever is being filtered, if at all it is there, it is not being reabsorbed. So sodium is not reabsorbed. On top of that, this is lipocalin, CALIN, lipocalin, CALIN. Sorry, 1,8 also biomarker. I'm not aware of that. 1,8. There are various other biomarkers. The Harrison gives a big list of biomarkers, okay, but they are not practically useful. KIM1 and NGL1 are they have some clinical relevance also. We have used them in we are using them clinically also, even though they are not very useful, but still you can use them. Others are not at all useful clinically, though we have a very big list of biomarkers which are increased in patients with AKI. Okay. Yes. Now, what are the causes for preserved urinary output? Yes, kidney injury molecule and neutrophil gelatin associated lipocalin. Okay. Yes, now what are the causes for preserved urinary output? Preserved urinary output. Causes for preserved urinary output. The most important cause for preserved, yes, clinically most useful are KIM and NGAL. Okay. Preserved urinary output can be seen in patients with associated nephrogenic diabetes insipidus, as we have in patients with, I did not tell you there, long-standing urinary tract obstruction is also a cause for nephrogenic diabetes insipidus, and all other drugs which were associated with nephrogenic diabetes insipidus, like aminoglycosides, amphotericin B, cisplatin, lithium, they can all be associated with preserved urinary outputs. Okay, so when there's associated nephrogenic diabetes insipidus, then you can have preserved urinary output despite acute renal failure. Now, how do you differentiate pre-renal and renal, intrinsic renal failure from each other? So what are the criteria to differentiate them? Pre-renal and intrinsic renal failure. So how do we differentiate them? The most important criteria to differentiate them is FeNA, fractional excretion of sodium. FeNA. FeNA stands for fractional excretion of sodium. Okay, and the formula for this is 100 into sodium in the urine divided by sodium in the plasma multiplied by creatinine in the plasma divided by creatinine in the urine. Okay, so this is FeNA, fractional excretion of sodium. Basically, FeNA indicates the amount of, it indicates the percentage of sodium reabsorbed out of the total filtered sodium. Okay, so the percentage of sodium reabsorbed from the total amount of sodium which is filtered. So in pre-renal, your tubules are normal. So most of the sodium which is filtered will get reabsorbed. Okay, so the FeNA is less than 1%. But in intrinsic renal failure, the sodium which is filtered, it may not be reabsorbed because the tubules are not functioning well. So the amount of sodium which is excreted is increased. Okay, it is not reabsorbed. So FeNA is more than 1 in patients with intrinsic type of renal failure. So fractional excretion of sodium is the most important thing which would help in differentiating between pre-renal and renal type of. Fine, so FeNA is most important. Now, other than FeNA, there are some other criteria also. Okay, so low FeNA indicates sodium retention. Okay, low FeNA indicates sodium retention. Okay, when sodium retention is there, that means the tubules are not the reabsorption at the level of tubules is normal. So FeNA is the most important thing which differentiates pre-renal and renal failure. The other criteria which differentiate them are the urinary sodium. Obviously, in pre-renal, sodium is reabsorbed, so the urinary sodium is less. It is either less than 10 millimole per liter, this is pre-renal, and this would be renal, or less than 20 milliequivalents per liter. Okay, the values are different because the units are different. Here it is more than 20 and more than 40. So urinary sodium will also help in differentiating. Then urinary creatinine to plasma creatinine ratio. This is more than 40 in pre-renal and this is less than 20 in renal. One more thing is the blood urea nitrogen to creatinine ratio. This is more than 20 in pre-renal and less than 10 to 15 in renal. So when blood urea is increased out of proportion to your creatinine, suppose the blood urea is 100, normally it is still 40 and creatinine is only 2.5. So creatinine is slightly increased but blood urea nitrogen is much more increased. So when the ratio is more than 20, then it is more indicative of pre-renal failure compared to renal failure. So blood urea nitrogen to creatinine ratio is more than 20 in pre-renal, less than 10 to 15 in renal. The reason for this is increase in the urea recycling, which is happening in patients with pre-renal failure. Now, can we have FeNA less than 1 in a patient with renal failure? Yes or no? Yes or no? Normal one, yeah. Can we have a FeNA of less than 1 in renal failure. There are some causes for FeNA of less than 1 in renal failure, and these are sepsis, rhabdomyolysis, contrast-induced nephropathy, acute interstitial nephritis, renal graft rejection, okay. So normally in pre-renal failure, ideally FeNA is more than 1%, but there are some exceptions to this rule. In renal failure associated with sepsis, rhabdomyolysis, contrast-induced nephropathy, acute interstitial nephritis, allograft rejection, you can have FeNA of less than 1. Okay, Gorakhpur. Okay, classically pre-renal it is less than 1, renal it is more than 1. But in some types of renal failure, intrinsic renal failure also, you can have a FeNA of less than 1. You can have a FeNA of less than 1. Okay, Gorakhpur. Yes. And can you have a FeNA of more than 1 in pre-renal failure? Yes or no? Can you have a FeNA of more than 1 in pre-renal failure? Yes or no? Yes. Yes, those of you who are saying yes, if yes, then when? We can have a FeNA of more than 1 even in pre-renal failure, and that would be the one associated with diuretic use. The diuretic use can also be associated with hypovolemia. Okay, hypovolemia. So when there is more than one of so when diuretic use is there in a patient with pre-renal failure, it can cause a FeNA of more than 1. Okay, is this clear to all of you? Yes or no? So remember these exceptions. The classical questions have been asked so many times, but now they might ask you the questions on these exceptions. So a patient of pre-renal renal failure, if they have a FeNA of more than 1, that means there might be some associated diuretic use. Okay, they might be on Lasix furosemide. So diuretic use in a patient with pre-renal failure can cause a FeNA of more than 1. Okay, fine to all of you. Best way is still considered to be FeNA. Best way is still considered to be, obviously, anytime your pre-renal failure can convert into renal. Okay, anytime your pre-renal would convert into renal, when there is irreversible change, it can anytime convert into renal failure. So sometimes it may not be a very absolute demarcation. You may not be absolutely able to demarcate between them. But if you are asked the best option, then it is FeNA. The fractional. And one more is renal failure index. Okay, renal failure index is almost similar to your FeNA only. Okay, renal failure index. Renal failure index. Renal failure index, which is, how do you look for renal failure index? Urinary sodium divided by urinary creatinine divided by plasma creatinine. It is less than 1 in pre-renal, more than 1 in renal. Okay, so renal failure index is urinary sodium divided by urinary creatinine divided by plasma creatinine. Okay. And after ARF, CRF, chronic renal failure. Chronic renal failure. Chronic renal failure, CRF. Now, how do you define CRF? Renal failure index is urinary sodium divided by urinary creatinine, and divided by plasma creatinine. renal failure index was urinary sodium divided by urinary creatinine divided by plasma creatinine. Okay. Urinary sodium divided by urinary creatinine multiplied by plasma creatinine. Okay. It is less than 1 in pre-renal, and renal it is again more than 1. Less than 1, more than 1. Yes, CRF, chronic renal failure. Now, how do you define chronic renal failure? Chronic renal failure is abnormal kidney function and progressive decrease in GFR associated with azotemia and sign and symptoms of uremia. Okay, sign and symptoms of uremia. So CRF is abnormal kidney function with progressive decline of the glomerular filtration rate associated with azotemia and sign and symptoms of uremia. Sign and symptoms of uremia. Okay. Then ATN is basically when there is ischemia. Ischemia leads to necrosis of the tubules, so it is a kind of intrinsic pre-renal failure only. Okay, ischemic type. And when there is, okay, so azotemia, sign and symptoms of uremia, and other things which you have is hypertension, which is very important, anemia. Any patient coming to you with anemia, hypertension, always rule out chronic renal failure. Anemia, hypertension, and ultrasound suggestive of bilateral small kidneys. Bilateral small kidneys and renal osteodystrophy. This means the patient is having CRF, and all these symptoms should be seen for at least a duration of more than 3 months. So abnormal kidney function with the progressive decline of GFR associated with azotemia, sign and symptoms of uremia, hypertension, severe anemia, bilateral shrunken kidneys on ultrasound, renal osteodystrophy for more than 3 months, we label it as CRF. We label it as CRF. Now, what are the grades of CRF? How many grades of CRF are there? Grades of CRF, chronic renal failure. How many grades are there? We have 6 grades starting from 0. We have 6 grades starting from 0. Grade 0 is GFR of more than 90 ml per minute, but associated with this there are risk factors. So GFR is normal, but there are risk factors for renal failure. Risk factors for renal failure or chronic kidney disease. Risk factors like diabetes, hypertension, some autoimmune disease, old age, African ancestry, family history of renal disease, then previous history of acute kidney injury, history of AKI. Okay, so all these are risk factors. So GFR of more than 90 ml per minute, anemia and CRF, we will discuss that. Anemia, okay, there are various causes for anemia and CRF. So GFR of more than 90 ml per minute with risk factors of CKD, it is considered to be stage 0. Stage 1 is again GFR more than 90 ml per minute, but associated with a demonstrable kidney damage in the form of either persistent proteinuria or abnormal urinary sediment. Okay, so GFR may be normal, but still there is proteinuria or abnormal urinary sediment. Then stage 2 is, sorry, between 60 to 89 ml per minute. Stage 3 is 30 to 59. Stage 4 is 15 to 29. And stage 5 is less than 15 ml per minute. Less than 15 ml per minute. So these are the various stages, 6 stages of your chronic kidney disease. Okay. Stage 3, 4, 5 are considered to be end stage renal disease. End stage renal disease. 3, 4, 5 is considered to be end stage renal disease. End stage renal disease. Stage 3, 4, 5 of CKD. Now, how do you calculate GFR? A rough idea of GFR is given by the formula known as Cockcroft-Gault equation. Cockcroft-Gault equation. Cockcroft-Gault equation. Okay, so Cockcroft-Gault equation will give you estimated creatinine clearance by the formula of 140 minus age, 140 minus age, last slide, into body weight in kgs, body weight in kgs divided by 72 into plasma creatinine milligrams per deciliter. Okay, last slide you can see later on. Okay, so the estimated creatinine clearance or GFR can be calculated roughly by this formula: 140 minus age into body weight in kgs divided by 72 into plasma creatinine in milligrams per deciliter. And if it is a female, you further multiply it by 0.85. You further multiply it by 0.85. So this is Cockcroft-Gault equation. Cockcroft-Gault equation. Questions have been asked on this. Okay. And why do Africans have a high chance of renal failures? It is because of a gene that they have. Africans, they have a high chance of renal failure because of a gene that is present, that is APOL1 gene. Okay, it is because of this gene that Africans are associated to have a high risk of kidney disease. Now, what are the complications of chronic renal failure? Complications of chronic renal failure. There are various complications of chronic renal failure, and some of them can be treated by dialysis, some of them will not be helped by dialysis, and some of them may even develop after dialysis. So the clinical problems that we have in uremia, first is the fluid and electrolyte imbalance. Fluid and electrolyte imbalance. So most importantly, there is volume expansion, because your sodium and water are the water is being retained. Okay, so volume expansion is there. Less of sodium is the hyponatremia is there because of volume expansion. Then this hyperkalemia is there. This hyperphosphatemia. Okay. Now, when we give them dialysis, all these things will tend to improve. They all improve on giving dialysis. Volume expansion, hyponatremia, hyperkalemia, hyperphosphatemia, they all improve on giving dialysis. They all improve on giving dialysis. Now, can a patient of CRF have hypokalemia? Can they have hypokalemia? Yes or no? Cause of hypokalemia in CRF. Sometimes, rarely, they can have hypokalemia. That might be because of the diuretic use, or decrease dietary intake, which is very rare. Most importantly, it is because of the diuretic use, or sometimes because of the GI loss, vomiting, etc., or diarrhea, that they may have hypokalemia. Okay, so diuretic use, decrease intake, or GI loss may be a reason for hypokalemia in patients with CRF, though it is very rare, though it is rare. And primary renal potassium wasting, as is seen in patients with some Fanconi syndrome or renal tubular acidosis type 1, 2. So they can also be associated with hypokalemia. Okay. So CKD because of Fanconi syndrome, renal tubular acidosis type 1 and 2 can also be associated with hypokalemia. Fine. And lastly, what about calcium? Calcium is less, again because of less of the active vitamin D which is synthesized. What is the site of synthesis of vitamin D? What is the site of 1 alpha hydroxylase in the kidney? Where does 1 alpha hydroxylation occur in kidney? Site of 1 alpha hydroxylation in kidney. Yes, it is PCT. This occurs in PCT. Fine. So 1 alpha hydroxylase is present in PCT, and moreover calcium combines with phosphate. So this leads to again decrease in calcium. So the phosphate which is being retained is combining with the calcium, which again contributes to hypocalcemia. Now next is the hematological complication, the neuromuscular complications. Neuromuscular complications. So neuromuscular complications like fatigue, sleep disorders, altered sensorium or encephalopathy, uremic encephalopathy, peripheral neuropathy can be there, myoclonic jerks can be there, myopathy can be there. They can have restless leg syndrome, they can have seizures. Okay, now if I ask you which of them will improve on doing dialysis, fatigue would improve, altered sensorium would improve, peripheral neuropathy, it may improve or it may persist. Sleep disorders, they persist after doing dialysis. Peripheral neuropathy, it may improve or it may persist after doing dialysis. Myoclonic jerks, if they are there, they improve on doing dialysis. Okay, they improve or persist. Myoclonic jerks, they improve on doing dialysis. Seizures, they may improve or they may persist after doing dialysis. Myopathy may persist or develop after doing dialysis, may persist or even develop. So myopathy persist or develop after doing dialysis. Restless leg syndrome can improve or persist after doing dialysis. Improve or persist after doing dialysis. Questions have been asked on this. Okay, which of these disorders will improve after doing dialysis? So if myopathy is one of the options, so the questions were all of the following except improve after doing dialysis. Options were encephalopathy, peripheral neuropathy, myopathy. So the answer was myopathy. Myopathy does not improve on doing dialysis. Okay, myopathy does not improve on doing dialysis. Does not improve on dialysis. Okay, so myopathy does not improve on dialysis. So this is important. Which improve and which persist or which rather develop after doing dialysis. Fine. Now, when we talk of the dermatological, before that hematological. 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 There is anemia. Okay. There are various causes of anemia in patients with patients on dialysis. First, there is decrease in the erythropoietin synthesis. What is the site of production of erythropoietin? From where is erythropoietin produced? Yes? Erythropoietin is produced from? No, this time the answer is not PCT. From where is erythropoietin produced? Not macular dense epithelium. Actually, this is produced by the peritubular capillary network. Okay, the cells of the peritubular capillary network, they sense the amount of oxygen being presented to them. Okay, so the endothelial cells of the peritubular capillary network would produce erythropoietin. Okay, so there's decrease in erythropoietin production. This is the most important cause. Then, because of the bleeding diathesis, there's loss of blood due to bleeding diathesis. Bleeding diathesis may be because of dysfunctional platelets and other things. There is loss of blood during dialysis. Loss of blood during dialysis. During dialysis also they lose blood. Okay. So there's decrease in production, there's loss of blood because of the bleeding diathesis, because of the dysfunction of the platelet, because of the blood loss during dialysis. So there are various other contributors also, and sometimes because of the decrease of appetite, they have nausea, they have vomiting, so the intake is less, because of which nutritional component like iron deficiency they also superimpose. Okay, because of the decreased intake they may also develop iron deficiency. So there are various causes for anemia in CKD. Anemia in CKD. And when you do dialysis, this anemia would improve. The most important reason would be because of the volume overload which is there, that is taken care of. So anemia tends to improve in patients with CKD when they are put on dialysis. Next is they can have thrombocytopenia. They can have leukopenia. And this thrombocytopenia may even be a complication of dialysis. So it may develop after dialysis. So thrombocytopenia does not improve with dialysis, rather it may develop after doing dialysis. And even leukopenia, it persists after doing dialysis. The Harrison gives this entire table which has mentioned all these things which improve, which develop, which persist after doing dialysis. So if you have any confusion, you can just go back and check from the table there. Okay. Then cardiovascular complications. Cardiovascular complications, the most important is because of the anemia, they can have congestive heart failure, they can have pulmonary edema because of the volume overload, and both of them will improve on doing dialysis. Then they can have uremic pericarditis. They may have uremic pericarditis which would also improve on doing dialysis. Then they can have hypertension because of the chronic renal failure, they develop renal mediated hypertension. Then they can have vascular calcifications. Okay, this hypertension may improve or may persist after doing dialysis. It may improve or it may persist. Vascular calcification, they persist and they may even develop after doing dialysis. They persist or they develop after doing dialysis. Then they can even have cardiomyopathy, dilated cardiomyopathy, DCMP. And this may improve, it may persist, it may develop after doing dialysis. It may improve, persist or develop after doing dialysis. Then in the lungs, they can develop uremic lung. Uremic lung infiltrates in the lung and they usually improve when you do dialysis. Uremic lung would improve when you do dialysis. Uremic lung improves on doing dialysis. Then endocrine abnormalities like the vitamin D deficiency, which would improve on doing dialysis. Then they can have the renal osteodystrophy, which can be further divided into two types. Either it can be a high output, high turnover disease, or it can be a low turnover disease. Okay? High turnover disease is classically associated with hyperparathyroidism. When PTH values are high, it is said to be high turnover bone disease, and when PTH values are less, it is said to be turnover bone disease. And the other name given for this is adynamic bone disease. Okay, so the bone disease in a patient with chronic kidney failure can be of two types. Either it can be high turnover, which is associated with high levels of parathormone, or it can be low turnover disease where we have less of parathormone. Now, why should they have hyperparathyroidism? The hyperparathyroidism that is seen here is given the name of secondary hyperparathyroidism. Yes, why should they have secondary hyperparathyroidism? It is because of the fact that there is less of calcium. Whenever there's less of calcium in the blood, they have less of calcium because there's less of active vitamin D. So when there's less of calcium, what is stimulated? The calcium sensing receptors. The calcium sensing receptors which are present on the parathyroid gland and the kidneys. Okay, and the parathyroid gland, when they're present in the sensors present on the parathyroid gland, whenever this decrease in serum calcium, it increases the parathormone release. It increases the parathormone release, and from kidney it increases the reabsorption of calcium. Okay, so from kidney it increases reabsorption of calcium, and from parathyroid gland it increases the release of parathormone, because this parathormone would increase bone resorption and it would lead to normalization of serum calcium. Okay, it would lead to normalization of serum calcium. So whenever we have hypocalcemia, not only in vitamin D not only in chronic renal failure, for any cause of this hypocalcemia, it would stimulate the calcium sensing receptors, the calcium sensing receptors on parathormone and on the parathyroid gland and the kidney. So when they are stimulated, they would increase PTH release, and when this PTH is more, it would lead to bone resorption, so that the serum calcium levels are normalized. Okay, not much is left, don't worry. Okay, after CRF, I think after CRF would be all that I would teach you in kidney. Okay, then we would just do arthritis. Okay, let me just complete the CRF renal osteodystrophy, then we would take a break. Fine. Okay, so this is your high turnover bone disease. High turnover is because of more of parathormone, and when parathormone is more, it would lead to bone resorption. It would lead to bone resorption. Okay, then low turnover disease, which is also given the name of adynamic bone disease. When do we see this? This is seen in patients who have overzealous 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 calcium and vitamin D supplementation. When too much of calcium and vitamin D is given to a patient with CKD, what happens? It leads to suppression of parathormone. Okay, so when too much of calcium and vitamin D, as we see nowadays, so when there is too much of vitamin D supplementation, it would lead to suppression of the parathormone. Okay, I would see as early as possible what we can finish by 8 or 9. Okay, polycystic kidney disease. Okay, I would tell you some few points about polycystic kidney disease. Fine, don't worry, I would tell that. Most common cause of anemia in CRF. What were the options? What were the options of most common cause of? I need a break for myself also. Okay? I also need to have some rest because I'm speaking since morning, so I need a break for myself if at all I need to have it. Okay? Most clear secondary hyper. Okay. Most common cause blood loss. Actually, this I will also go back and check. Most common. Okay. Usually we say it is because of the erythropoietin, but other things also contribute a lot. The blood loss, the dialysis. Most common. I would go back and check. Okay. Let us just do this. Okay. CRF. Okay, now just one very important thing about CRF is hyperkalemia. So, how do you treat hyperkalemia? How do you treat hyperkalemia? Treatment of hyperkalemia. Treatment of hyperkalemia. Yes. Treatment of hyperkalemia. What is the most important treatment? The first thing that you need to give in patients with hyperkalemia is calcium gluconate. Gluconate. This does not decrease the potassium in the blood, but it helps in stabilization of the cardiac membrane potentials. Okay. So the first thing that you give in treatment of hyperkalemia is calcium gluconate. Otherwise, when we talk about things which decrease serum potassium levels, the first thing is the beta agonist. The beta agonist like salbutamol, salbutamol. Okay, so salbutamol, beta agonist. Then another thing is infusion of glucose plus insulin. Insulin is a thing which internalizes potassium. So insulin is a very important treatment for this. You have already done hyperkalemia. Okay, fine. Then let's leave this. Okay, dermatological features. Dermatological features. Dermatological features that we have is pallor, which improves on doing dialysis. They have pruritus, which usually persist on doing dialysis. They may have uremic frost, pigmentations on the skin, which usually improve on doing dialysis. Then they can have ecchymosis because of the bleeding, which tend to improve after doing dialysis. Okay. Okay, then hyperkalemia you can just all go back and read. Okay. The causes of interstitial nephritis. The causes of interstitial nephritis. I would just mention them. Causes of interstitial nephritis, and before that, causes of renal papillary necrosis. Now, renal papillary necrosis, half and half nail. Okay. Let me do renal papillary necrosis, then we will do half and half nail. Okay. Renal papillary necrosis, the most ischemic area is the medulla, and in medulla the tip of papilla is the most ischemic. Papilla is associated with most precarious blood supply in the kidney. So whenever there is ischemia, there is necrosis of the papilla. Okay, because it is having the least blood supply. It is deep down the medulla where we have minimal blood flow. So papilla are associated with minimal blood flow. So whenever there is ischemia, they undergo necrosis. So during ischemia, they undergo necrosis. Fine. And the renal papillary necrosis is characterized by clinical features like loin pain, hematuria, and variable degree of renal failure. Okay, deterioration of the renal functions. Now, what are the causes for renal papillary necrosis? The most important cause is diabetes mellitus, which predisposes. Another one is analgesic abuse. These are the most common causes for renal papillary necrosis. Otherwise, you have a mnemonic Postcard. Postcard. These are the causes for renal papillary necrosis. P stands for pyelonephritis. O stands for obstruction. S stands for sickle cell anemia. T stands for tuberculosis. C stands for cirrhosis. A stands for analgesic abuse. R stands for renal vein thrombosis. D stands for diabetes mellitus. Okay, so postcard, because analgesic abuse is associated with nephritis, interstitial nephritis. Okay, so that may also lead to sloughing off of papilla. So pyelonephritis, obstruction, sickle cell anemia, tuberculosis, cirrhosis, analgesics, RVT, and diabetes mellitus. Okay, so they are all causes for renal papillary necrosis. Hypertension, I'm not sure. Okay, so learn these causes for renal papillary necrosis. Now, what are the causes for interstitial nephritis? Causes for interstitial nephritis. The causes for interstitial nephritis are mostly your drugs like antibiotics like beta-lactam antibiotic, sulfonamides, quinolones, vancomycin, erythromycin, rifampicin, ethambutol, acyclovir, not antibiotics. So then another drug is NSAIDs. Then is diuretics like thiazide, furosemide. Then anti-epileptics like phenytoin, carbamazepine, valproate, phenobarb, etc. And lastly, other drugs like acyclovir, captopril, H2 receptor antagonist like cimetidine, ranitidine, proton pump inhibitors, mesalamine. Okay, so first is the drugs. Another thing which can cause interstitial nephritis is infections like bacterial or viral infections. Bacterial infections like staphylococcus, streptococcus, salmonella, brucella, yersinia, corynebacterium. Viruses like Epstein-Barr virus, cytomegalovirus, HIV, hantavirus, polymavirus. Okay. Fine. So infections and drugs can lead to interstitial nephritis. Now, one very important feature hantavirus is polymavirus. Okay, polymavirus. Proton pump in mesalamine. Like sulfasalazine, we have mesalamine. Okay, so mesalamine. After HIV, hantavirus, HANTA, hantavirus, polymavirus. hantavirus. Okay, hantavirus polymavirus. Fine, can I change over? Yes, can I change? Okay, let me just tell you one thing, then we would have a short break of 5 minutes. If I would be fast, then many of you would tell me to be slow. So I have to maintain a break because I need a rest for 5 minutes. Okay, that is why I need a break. It's not for you, it's for me. Fine. Isostenuria. Isostenuria means fixed specific gravity of the urine. This is because whenever we have tubular diseases, there's no concentration or dilution of the urine. The specific gravity of the urine remains fixed. This is known as isostenuria. Okay, so when you have tubulointerstitial disease, there would be a fixed specific gravity of the urine, which is known as isostenuria. Okay, so 5 minutes of break, just 5 minutes, don't worry, it would not be more. I need to rest my voice for 5 minutes. Okay, just for 5 minutes. Anemia which is not refractory to your medical management, then volume overload leading to pulmonary edema, uremic encephalopathy, blood urea more than 100. There's no value of creatinine. Okay, blood urea more than 100 is also an indication for doing dialysis. Okay, now the dialysis can be of two types. It can be hemodialysis or peritoneal dialysis. Some important side effects of hemodialysis and peritoneal dialysis. Hemodialysis, some side effects are the most important, the most common side effect is hypotension. Then others can be electrolyte imbalance, leading to cramps. Then anaphylactic reactions can occur. With peritoneal dialysis, the side effects are most important being peritonitis, hypoproteinemia, weight gain, hyperglycemia. Weight gain. Right? So these are the side effects of peto. So you have dialysis of two types, peritoneal and hemodialysis. Yes, when we talk of the cystic disease, last slide we will repeat it in the end. Okay, so polycystic kidney disease is of two types. It can be autosomal dominant. Or it can be autosomal recessive. Autosomal dominant or autosomal recessive. This autosomal dominant type is usually because of the defect of genes, genetic defect of PKD1 gene or PKD2 gene, and these genes, they basically are associated with production of a protein known as polycystin. Okay, this polycystic kidney disease is of two types: autosomal dominant, which is known as autosomal dominant polycystic kidney disease, or you have autosomal recessive polycystic kidney disease. Okay, so you have autosomal dominant and autosomal recessive polycystic kidney disease. This autosomal dominant is associated with defect of genes like PKD1, PKD2, which are associated with production of a protein known as polycystin. This polycystin has various functions. It acts as a flow sensor. It acts as a flow sensor on the luminal surface. It acts as an adhesion molecule on the basolateral surface. Okay, and this adult autosomal dominant polycystic kidney disease, it usually presents in third or the fourth decade, and some say up till fifth decade of life. Okay, it usually present in adulthood, late adulthood, third, fourth, or the fifth decade. The patient would present to you with autosomal dominant polycystic kidney disease. Okay, on the other hand, when we talk of the autosomal recessive, here there's a defect of chromosome number 6 on which there's a gene PKHD1. Okay, there's a gene on chromosome number 6 PKHD1. So this is defective in patients with autosomal recessive type of polycystic kidney disease, and this gene dictates the production of a protein known as fibrocystin or polyductin. Okay, the proteins which are synthesized by this gene are fibrocystin or polyductin. Okay, here it was polycystin, here it is fibrocystin. Here it is fibrocystin. This autosomal recessive it is usually present in children. This was present in adults. This is usually seen in children, and it has three variants. It can be infantile, it can be juvenile, or it can be seen in adolescents. Okay, this can be seen in three forms: infantile form, juvenile form, and adolescent form. So autosomal recessive is seen in children, in infantile form, juvenile form, or adolescent form. Now this autosomal dominant polycystic kidney disease, what is the usual clinical picture? Clinical features. The clinical features of this condition are usually pain abdomen. They present with Okay. So clinical features of autosomal dominant would include pain abdomen, which can be either because of the infection of the cyst, it can be because of the rupture of the cyst, etc. Okay, so most importantly, they present with clinical features like pain abdomen. Other features are hematuria, nephrolithiasis, hypertension. So they can present with pain abdomen, which can be because of infection like pyelonephritis, or because of the rupture of the cyst. They can present with hematuria, they can present with nephrolithiasis, they can have hypertension. Nephrolithiasis is seen in 15 to 20%, hypertension is seen in 75% of the adults. Okay, hypertension is seen in. Now, other than the kidney involvement, patients with autosomal dominant polycystic kidney disease, they also have some extra renal. Yes, most common usually is incidental hypertension. The most common presentation that they come to you with is incidental hypertension. Incidental. The patient may not know, incidentally, they are discovered to have incidentally hypertension. Incidental hypertension. A very important presentation may be incidental hypertension. Okay. Yes, now other than the kidney involvement, they can have some extra renal. If we talk of the most common site, the most common site for extra renal cyst is the liver. You may have hepatic cyst in approximately 83% patients. Okay, other than the cyst in the kidney, they may also have cyst in the liver in 83%. This is the most common. Usually it is asymptomatic. The liver function tests are normal. LFT are usually normal. They are asymptomatic, but sometimes they may be associated with pain abdomen because of the rupture or infection of the cyst. Then other sites where you can have cyst is intracranial. Other sites for cyst are in the abdomen are spleen, pancreas, ovaries. They can all have cyst. Okay, most common is liver. Others are spleen, pancreas, and cyst. Other extrarenal manifestation is intracranial aneurysms. Intracranial aneurysm, there is increase in 2 to 4 times incidence of intracranial aneurysm compared to the normal population. Okay, so they have 2 to 4-fold increase in the intracranial aneurysms in patients with autosomal dominant polycystic kidney disease. Then in the heart, cardiac manifestations can also be there in the form of mitral valve prolapse or aortic regurgitation. They can have mitral valve prolapse or aortic regurgitation. Okay, mitral valve prolapse or aortic regurgitation. Then they can have colonic diverticulae. Okay, they can have colonic diverticulae. So they are all extrarenal manifestations of autosomal dominant polycystic kidney disease. And how do you treat? It is symptomatic. You have to control hypertension. Control hypertension by giving drugs like ACE inhibitors because it is because of the renin-mediated hypertension. If there's UTI, you have to give drugs for the UTI, especially those which can penetrate the cyst. Antibiotics which can penetrate the cyst like trimethoprim, sulfamethoxazole, that is your Septran, or fluoroquinolones. Okay, so you have to give them drugs which can penetrate the cyst. So you have to control hypertension, treat for the complications like UTI if they're there. Now this and always remember this is a autosomal dominant condition, so family history is always there. Family history is important. Family history is always important because it is autosomal dominant. Family history is important because it is autosomal dominant. Now what about autosomal recessive? Autosomal recessive polycystic kidney disease. So I told you this is seen usually in children. It is present in 3 forms: infantile, juvenile, or adolescent. This is less common than the. No, it is not reversible. Polycystic kidney disease is not reversible. It is because of the mutation of which gene? PKHD1 on chromosome number 6, which produces which protein? Fibrocystin or polyductin. Fine. Now when we talk of this infantile form, most commonly is infantile form, and in infantile form more than 50% will die in utero. Because why do they die in utero? Because of the oligohydramnios. When kidney is not functioning well, there's oligohydramnios. which leads to pulmonary hypoplasia. Pulmonary hypoplasia. So most of them die in utero. And if at all they survive, they develop end stage renal disease by the age of 3 years. Most of them die during in utero. The function of fibrocystin, it is a receptor in the primary cilia of tubular cell. It is a receptor in primary cilia of tubular cells. Exactly what function is there, it is not mentioned, but it is a receptor present in the cilia of the tubular cells. Okay. So most of them did die in utero, and if they survive, they develop end stage renal disease by the age of 3 years. Now, one association which is seen in patients with autosomal recessive is it may be associated with hepatic fibrosis or the Carolese disease. Okay, it is associated with Carolese disease or hepatic fibrosis. Okay, it has an association with Carolese disease or hepatic fibrosis. Now, another disease is nephronophthisis. Nephronophthisis. Which organ is not involved? Lung. I guess lung is not involved. Okay, in autosomal dominant polycystic kidney disease, lung is not involved. Lung. Okay. Nephronophthisis. Nephronophthisis is the most common genetic cause of end stage renal disease in childhood and adolescence. Okay, it is the most common genetic cause of end stage renal disease in childhood and adolescent. There are 11 variants based on different kinds of genetic mutation. 11 genetic mutations are associated with this nephronophthisis. Basically, in nephronophthisis, there is impaired tubular function. So when your tubules are not functioning well, what would happen? There will be salt wasting, and when there is salt wasting, and also one more thing would be there, defect in the concentration and acidification of urine. Okay, so basically it is a disease where there is impaired tubular function associated with salt wasting, defect in concentration of the urine, acidification of the urine. Okay, and because of this impaired tubular function, this loss of various electrolytes, which is associated with growth retardation. This growth retardation, this is very important, growth retardation. They will all have polyuria, especially nocturnal enuresis may be a complaint. They may have polydipsia also. Because of the salt loss, they may have a craving for the salt, polyuria, growth retardation. Then there is renal insufficiency, that is increase in the blood urea, increase in the serum creatinine, but without hematuria. Hematuria is not seen in patients with nephronophthisis. Hematuria is not seen in patients with nephronophthisis. Hematuria is not seen in nephronophthisis. Will they have hypertension? Will a patient of Nephronophthisis, can they have hypertension? Yes or no? Hypertension. Will they have hypertension? Yes or no? Hypertension was quite common in patients with polycystic kidney disease, but here they will not have hypertension. Why? Because already there is salt wasting. Salt wasting. So, no hypertension. No hypertension. No hypertension. Similarly, no proteinuria, no edema. These features are not seen in patients with nephronophthisis. Now, there are some extrarenal manifestations also. Extrarenal manifestations also. Extrarenal manifestations like it may be associated with retinitis pigmentosa, retinitis pigmentosa, extra renal manifestations can be retinitis pigmentosa, which is given the name of senior Loken syndrome. Then they can have association with Omorosis fugax, oculomotor apraxia, cerebellar ataxia, which is given the name of Joubert syndrome. Okay. So retinitis pigmentosa, cerebral ataxia, they can all be seen in association with this. What is the pathogenesis and the pattern of inheritance? The pattern of inheritance for nephronophthisis is usually autosomal recessive. It is autosomal recessive, and there's a defect in which protein? There's defect in a protein known as nephrocystin. Nephrocystin, which is again present in the primary cilia. A protein which is present in the primary cilia of the tubular cells, which is defective. Nephrocystin. Now, what about the size of the kidney on ultrasound? On ultrasound, the size of the kidney in autosomal dominant polycystic kidney disease would be more. Okay, there are cysts, so it is usually increased. But patients with nephronophthisis on ultrasound, they either have a normal or small size of the kidney. Okay, normal or small size kidney is there on the ultrasound. Okay, on ultrasound, the kidney size is all either normal or small. It is not increased. This is very important. This is again a difference for nephronophthisis. Nephronophthisis. Fine. Now, what is I did not tell much in dialysis. Okay, so don't worry about dialysis. Okay, okay, don't worry, I would show you the slide at the end. So what is medullary cystic disease? Medullary cystic kidney. Medullary cystic kidney. Medullary cystic kidney is an autosomal dominant condition. Autosomal dominant condition. So there will always be a family history. Okay, and it is usually seen in adults. Usually seen in adults. Usually seen in adults more than 20 years. The defect lies in which gene? MCKD1 or MCKD2. MCKD1 or MCKD2 because of which there are cysts in the medulla. No growth retardation is seen in these individuals, no growth retardation. Extrarenal manifestations are usually absent except increase in the uric acid levels, which may be associated with gout. Okay, except hyperuricemia, which may be associated with gout. Fine. And on ultrasound, the kidney size is small. Here also the kidney size is small. Kidney size is also small. So this is medullary cystic kidney. Now what is medullary sponge kidney? Medullary sponge kidney. Medullary sponge kidney. This medullary sponge kidney is a benign condition. It is common condition, but it is a benign condition characterized by ectasia of the papillae and collecting ducts. What do you mean by ectasia? Ectasia means dilatation. So medullary sponge kidney is a disease characterized by dilatation of the papilla and the collecting tubules. And because of this dilatation, there is urinary stasis, okay, and it may be associated with incomplete renal tubular acidosis of distal type. Okay, so because of this backflow, the back pressure, sometimes they can have incomplete distal RTA, because of which they can have stones, renal stones. Renal stones are a feature of distal renal tubular acidosis, so they may be associated with renal stones. And in some patients, they may also be associated with extrarenal manifestations like anomalies of urinary tract or the hepatic duct, ectasia, that is your Caroli's disease. So Caroli's disease may be seen in patients with both medullary sponge kidney as well as patients with autosomal recessive polycystic kidney disease. Okay, hepatic duct ectasia may also be seen. Now when you do ultrasound, in ultrasound there is, you see hyperdense papillae with clusters and renal stones. And on intravenous pyelography, there is a sign which is seen, which is given the name of paintbrush appearance. On intravenous pyelography, the appearance which is seen is given the name of paintbrush appearance. Okay, on intravenous pyelography, because of the ectatic collecting tubules. And usually treatment is not required. No treatment is required. Just ask them to increase the fluid intake so as to decrease the stones, okay, renal stones. So hyperdense, it is mentioned hyperdense papillae, not hyperechoic, hyperdense papillae with clusters of small stones. Okay, so I think rest you can all do it for yourself. Now just the summary of causes associated with nephrocalcinosis. That is stones in the renal parenchyma. Medullary causes and cortical causes. The medullary causes are more common. Okay, UTI I would tell you, don't worry. Medullary causes, they are common, and cortical causes are uncommon, less than 5%. Medullary causes like renal tubular acidosis type 1 or distal, then medullary, sponge kidney, which may be associated with incomplete distal RTA, papillary necrosis, then Bartter syndrome because of the hypercalciuria which is seen there, furosemide abuse because of the hypercalciuria, increased calcium excretion, oxalosis, then conditions like primary hyperparathyroidism, sarcoidosis, malignancies, hypervitaminosis type D, etc. Okay. Then cortical conditions like renal cortical necrosis, then chronic glomerulonephritis, transplant rejection, tuberculosis, oxalosis. Okay. When we talk of UTI, urinary tract infection, this UTI can be divided into asymptomatic bacteriuria, cystitis, pyelonephritis, or prostatitis. Okay, so they are all types of UTI. Okay, asymptomatic bacteriuria, cystitis, pyelonephritis, prostatitis. Now, when we talk of the most common organism associated with most common organism associated with the UTI, it is E. coli in 75 to 90%, then Staphylococcus saprophyticus in 5 to 15%, then others are Klebsiella, Proteus, Enterococci, Citrobacter. Okay, so most of them are gram negative. So basically it is gram negative bacteria which lead to UTI. One very important feature of pyelonephritis which differentiates it from cystitis is the kind of fever, picket fence fever. Okay, high grade fevers which goes down after 72 hours. Okay, so picket fence fever is seen in patients with pyelonephritis. Picket fence fever. What is the diagnostic criteria for pyelonephritis or cystitis or UTI? The diagnostic criteria is the gold standard for diagnosis of UTI. The gold standard for diagnosis is urine culture, but it would take some time, so most often we rely on urine routine microscopy. And on urine routine microscopy, if you have more than 10 to the power 2 colony forming units per ml in a female, or more than 10 to the power 3 colony forming unit per ml in a male, we diagnose UTI. So more than 10 to the power 2 colony forming unit per ml of urine in female and more than 10 to the power 3 in a male is UTI, is considered to be diagnostic of cystitis or pyelonephritis. When we talk of asymptomatic bacteriuria, it is said to be associated with no sign and symptoms of UTI, but on urine routine microscopy, more than 10 to the power 5 colony forming unit is present per ml of the urine, and if it is a catheter sample, so more than 10 to the power 2 colony forming units per ml. Okay, so this is it is not the other way around. No, this is not the other way around. This is fine. So this is female more than 10 to the power 2 and more than 10 to the power 3 in males. Fine. And finally, how would you treat them? Treatment is ideal is based on the antibiotics, but the drugs which we give them, usually that we give them is nitrofurantoin, trimethoprim sulfamethoxazole, fluoroquinolones, they are the usual ones which are given. Okay, nitrofurantoin. Okay, trimethoprim sulfamethoxazole or fluoroquinolones. What about asymptomatic bacteriuria? You can also give them beta-lactams, phosphomycin. Beta-lactams and phosphomycin can also be given. Okay. After this, we will discuss arthritis. Don't worry. Okay. In asymptomatic bacteriuria, usually there's no treatment is recommended. Asymptomatic bacteriuria, unless in special occasions like pregnant females, otherwise it is not usually recommended. Asymptomatic bacteria treatment is not recommended. Rest, I think you can all just go, and if no organism in culture, ideally you should not treat. Okay, then it means that there's no UTI. Okay. Rest you can go back and read for yourself. I don't know more than this. Okay, so let's just discuss arthritis and finish it off. Okay, now when we discuss arthritis, the first step in arthritis is to see the number of joints which are involved. Number of joints involved: mono, possy or oligoarthritis and polyarthritis. Okay, now mono is one. What do you mean by poly and polyarthritis? What is polyarthritis? What is polyarthritis? Polyarthritis and polyarthritis. Yes, poly is more than equal to 4 joints and possy is only 2 or 3 joints. So when 2 or 3 joints, it is possy, rest is poly. Now the first thing that you see after the number of joints is the type of arthritis. If it is inflammatory or mechanical. Inflammatory or mechanical. In inflammatory arthritis, the causes are autoimmune conditions like rheumatoid arthritis, SLE, then infections like tuberculosis, gonococcal infection, then Reiters or reactive arthritis, reiters or reactive arthritis, then crystal arthropathy, crystal associated, that is gout and pseudo gout. They are type of gout, pseudo gout, and all other your seronegative arthritis, seronegative arthritis, seronegative, which would include other things like ankylosing spondylitis, psoriatic, enteropathic. So they are all examples of inflammatory. What about mechanical? Mechanical includes osteoarthritis, traumatic arthritis, that associated with a tumor within the joint cavity, which is villonodular synovitis. Okay. Now, if I ask you the most common arthritis overall, the overall most common kind of arthritis is osteoarthritis. Overall most common is osteoarthritis, and when we talk of the most common arthritis in inflammatory, it is rheumatoid. Okay, overall most common is osteo, and in inflammatory most common is rheumatoid arthritis. Now, basically, inflammatory arthritis is the domain of physicians, and mechanical is the domain of your orthopedicians. How do you differentiate? What is the most important difference between mechanical and inflammatory arthritis? Inflammatory and mechanical. The most important difference. Most important difference, clinical difference, is regarding early morning stiffness. Early morning stiffness is present at least for 30 to 60 minutes in a patient with inflammatory arthritis, and in mechanical, stiffness is usually seen in latter part of the day. In latter part of the day, and if at all there is any early morning stiffness, it would be for less than half an hour, for less than 30 minutes. Fine. So here it is 30 to 60 minutes, there it is less than 30 minutes. Fine. Then sign and symptoms of inflammation like raised temperature, redness, swelling, tenderness, they're all features of inflammatory, and they are all absent in patients with mechanical. And lastly, crepitus is a feature of mechanical. It is not seen in patients with inflammatory. Crepitus is seen in mechanical, not in inflammatory. Fine. So inflammatory, it improves on movement, improves on movement. That is why we have stiffness in the morning, and it improves when the patient starts moving around. Whereas with mechanical, it get worsened on movement, worsened on movement. So improves on movement and worsening on movement. Now, the most important inflammatory arthritis is rheumatoid arthritis. Is rheumatoid arthritis. Fine. So this rheumatoid arthritis is the most common inflammatory arthritis. It is an autoimmune disease, but the name is a misnomer. It is not a disease limited to the joints. It is rather a multi-system disease. It is rather a multi-system disease characterized by autoantibodies. This is autoimmune, so some autoantibodies are there, and the most common autoantibody that we have is RA factor. This RA factor is autoantibody against the FC portion of IgG class of antibodies, against FC portion of IgG class of antibodies, and itself it is 80% of the times yes, what do I need to scroll down? So 80% of the times it is itself of IgM class. It can also be of IgA, D or E class, but 80% of the times it is itself of IgM class. Now this is the most common antibody seen in 75 to 80% of the patient, but the problem with this is that it is Gorakhpur. What is not clear? Yes? Yes, so this RA factor is the most common antibody, but the problem with this is it is associated with n number of false positive causes, n number of false positive causes. Okay, so false positive causes for RA are various, like your other autoimmune diseases, SLE, Sjogren, sarcoidosis, various kinds of vasculitis, chronic active hepatitis, infections like tuberculosis, malaria, leprosy, syphilis, filariasis, schistosomiasis, leishmaniasis. Okay, then it is against IgG and itself it is of IgM type. Okay, Belgaon. RA factor is against IgG. It is against IgG. It is antibody against IgG and itself it is of IgM class. Is it now clear? Rohtak and Belgaon. Okay, it is against FC portion of IgG. Itself it is of IgM class 80% of the times. Okay, Rohtak also clear. So don't confuse. It is against FC portion of IgG. Itself it is of IgM class. Now question was asked, all of the following except is associated with false positive RA factor. Options were Coombs positive, hemolytic anemia, VDRL positivity, HBV infection, inflammatory bowel disease. Yes, the answer is all of the following except is associated with false positive rheumatoid factor. The answer is inflammatory bowel disease because enteropathic is classically associated with seronegative. Coombs positive anemia can be seen in any autoimmune disease like your SLE also we have Coombs positive hemolytic anemia. Then hepatitis B, VDRL is syphilis positive. VDRL positive means syphilis. So all of them are associated with false positive except inflammatory bowel disease. Now the problem with RA factor was that it is not specific. So now we have a specific antibody and the most specific autoantibody for rheumatoid factor is rheumatoid arthritis is anti-CCP, anti-cyclic citrullinated peptide. So anti-CCP is most specific antibody. Now how do you diagnose rheumatoid arthritis? Earlier the diagnosis was present on 5 criteria. Based on the 5 criteria, first was early morning stiffness for more than 3 months. Second was symmetrical polyarthritis involving the small joints of the hand. Small joints of the hand, which would classically involve which joints? PIP, metacarpophalangeal, metatarsophalangeal and the wrist joint, and they would classically spare DIP, spare distal interphalangeal joint, spare distal interphalangeal joint. Then third was presence of rheumatoid factor. Fourth was presence of rheumatoid nodules. And last was radiographic changes like joint erosion, decrease in the joint space, juxta-articular osteopenia, so earlier if 3 out of 5 were present we used to diagnose the patient as, if 3 out of 5 were present we diagnosed the patient to have rheumatoid arthritis. Okay, so if out of these 5 criteria, if 3 were present we used to diagnose the patient to have rheumatoid arthritis. Okay, but now there's a very big change in this. Now we do not diagnose RA based on these criteria anymore. Now we have a new scoring system which was given by the American College of Rheumatology and European League Against Rheumatism. Okay. Can I change this page? Yes or no? Should I change and move over to the next scoring system? Yes. So now we have a scoring system which was given by ACR ULAR guidelines in the year 2010. 2010. Okay. The American College of Rheumatology and European League Against Rheumatism. In 2010, they gave a scoring system, and if the score is more than equal to 6 out of 10, we diagnose the patient to have early rheumatoid arthritis. Now, what was the need to change this scoring diagnostic system? It was because based on the previous criteria, we used to diagnose RA when the patient would have already developed deformities, joint erosion. So that time giving DMARDs would not be helpful. But when we give DMARDs in a patient with early rheumatoid arthritis, you can actually prevent erosions. You can prevent erosion. So now we aim to diagnose RA as early as possible. And for that, we need to use this scoring system. The first point included in the scoring system is the kind of joint involvement. So in joint involvement, if one large joint is involved, large joint are shoulder, hip, knee, elbow and ankle, knee, elbow and ankle, knee, elbow and ankle, knee, elbow and ankle. Okay. So shoulder, hip, knee, elbow and ankle. So out of these 10 joints, earliest joint involved, small joints of the hand, which one specifically, sorry, I don't know. Okay, which one is the earliest to be involved? So if one large joint is involved, the score given is 0. If 2 to 10 large joints are involved, the score given is 1. If 1 to 3 small joints are involved, small joints like PIP, metacarpophalangeal, metatarsophalangeal and the wrist joint. If 1 to 3 small joint is involved, we give them a 2. If 4 to 10 small joints are involved, we give them a 3. And if more than 10 joints are involved with at least one small joint, we give them a 5. So the maximum number of points which are given out of 10 to the joint involvement is 5. So out of 10, 5 points are given to the kind of joint involvement. Okay, if 1 large 0, 2 to 10 large it is 1, 1 to 3 small is 2, 4 to 10 small is 3 and more than 10 with 1 small is 5. Okay. Second is the serology. In previous criterias, only one thing was included in serology, that was RA factor. Now we have 2 things in the serology, that is, last is more than 10 joints with at least one small. Okay, more than 10 joints with at least one small joint is given 5. Okay, is more than 10 joint with at least one small joint. Agroha. Okay, so that is clear. Now for serology, if it is negative RA factor and negative anti-CCP, you give them a 0. You give them a 0. If one or both they are increased, but quantitatively they are less than 3 times the upper normal limit. Upper normal limit. So one or both of them are increased. Presence of rheumatoid factor is good or bad? Is good or bad? 0, negative. I'm not sure if it is good or bad. Okay. If it is good or bad. So if one or both are increased and quantitatively it is less than 3 times the upper normal limit, we give them a 2. So the minimum that we give here is 2. And if one or both are increased and quantitatively more than 3 times the upper normal limit, we give them a 3. So the maximum points given to this is 3. Okay. So is 3. Then third is acute phase reactants. Acute phase reactants are a new entry. Now we have to diagnose RA at the acute phase, so we include this also. Okay, so acute phase reactants like ESR and CRP. If they are both negative, you give them a 0. If one or both is increased, you give them a 1. So 1 point is given to the acute phase reactants. And last is the duration of the symptoms. If it is less than 6 weeks, you give them a 0. More than 6 weeks, you give them a 1. So the maximum point given to the duration is 1. So according to this, if the score is more than equal to 6 out of 10, you give them, you make the diagnosis of RA. Okay, you make the diagnosis of RA. Now, other than the joint involvement, it is a multi-system disease. Other features can be constitutional features, weight loss, fever. Then they can have rheumatoid nodules. Now, where all do we have nodules? Rheumatoid nodules are firm, non-tender nodules which are attached to the underlying periosteum or the bursa. Okay, they are firm non-tender nodules attached to the underlying periosteum or the bursa, and the most common site is the elbow or the olecranon. Elbow or the olecranon. Fine. Now, not only in the subcutaneous tissue, you can also have them in some tissues like pleura, peritoneum, pericardium, as well as in the lung parenchyma. They can also have the rheumatoid nodules. Okay, not only the subcutaneous tissue, these visceral organs, the serous cavities can also have the nodules. Okay. Then lastly, now what about the lung involvement? If I ask you the lung involvement, what is the most common form of lung involvement in rheumatoid arthritis? The most common form of lung involvement is pleural effusion. And what kind of pleural effusion is this? Transudative or exudative? Yes, it is exudative with the predominance of neutrophils as well as macrophages. Okay, exudative. This is not transudative, it is exudative. Then they can also have fibrosis of the parenchyma, leading to interstitial lung disease. Now, what is Kaplan syndrome? Kaplan syndrome. Yes, Kaplan syndrome is presence of rheumatoid nodules in a patient with pneumoconiosis. Which pneumoconiosis is most often associated with Kaplan syndrome? Yes, which pneumoconiosis is most often associated with Kaplan syndrome? Yes, which kind of pneumoconiosis? Most common is silicosis, and it was first identified in coal workers pneumoconiosis. First it was identified in coal worker pneumoconiosis, but most common overall it is in silicosis. Okay, it is most common in silicosis, that was first identified in coal workers pneumoconiosis. Okay, so be clear, first in coal workers, but most common overall is silicosis. Then yes, when we talk of the cardiac involvement, again in cardiac the most common type of involvement is pericardial effusion, like your heart. Then they can have pericarditis, myocarditis, etc., cardiomyopathy. Now what about the hematological? Hematological. In hematological, they will have anemia, which is anemia of chronic disease. Okay, anemia of chronic disease. Then, since it is an inflammatory condition, they will have thrombocytosis as well as leukocytosis. This is important. This is in contrast with SLE. In SLE, there was decrease in all the bloodlines: anemia, leukopenia, thrombocytopenia. Here, anemia is there, but it is different from that seen in SLE, and there is leukocytosis, there is thrombocytosis. Then, what is Felty syndrome? Felty syndrome is neutropenia plus splenomegaly in a patient with rheumatoid arthritis. Neutropenia plus splenomegaly in a patient with rheumatoid arthritis. Which lymphoma is more often seen in patients with rheumatoid arthritis, especially those who have Felty syndrome? Which lymphoma is more often seen? Diffuse B cell lymphoma is more commonly seen in patients with RA, especially those who have Felty syndrome. Okay, diffuse B cell lymphoma. Now, Sjogren's is also a very important complication of rheumatoid arthritis. It is seen in around 10% patients, and Sjogren's, the most common secondary cause for Sjogren's is rheumatoid arthritis. Most common ocular manifestation of RA is keratoconjunctivitis sicca. Similarly, in SLE, most common ocular manifestation is keratoconjunctivitis sicca. Okay. Now, they are also associated with increase in chances of coronary artery disease, increase in chances of atherosclerosis. Increase in chances of atherosclerosis and coronary artery disease is also there. And they have hypogonadism with less of testosterone production. Okay. Now, how do you treat rheumatoid arthritis? How do you treat rheumatoid arthritis? The most important drugs in the management of RA is the DMARDs. The most important drug is the DMARDs, and the best DMARD that we have is methotrexate. Methotrexate is the best DMARD that we give, usually started with a dose of 7.5 milligram once a week, and along with this you always give folinic acid in rest of the 6 days of the week. Okay, but the problem with this is it is contraindicated in pregnancy. Okay, other DMARDs which are used clinically are hydroxychloroquine, sulfasalazine. Okay, and they are both safe in pregnancy. Gold, penicillamine, they are not used clinically. Okay, KDT mentions all of those, but practically only 4 DMARDs are used: methotrexate, hydroxychloroquine, sulfasalazine. DMARD is drugs modifying activity of rheumatoid disease. Okay, drugs modifying activity of rheumatoid disease. Okay, DMARD. So methotrexate, duration would depend. Okay, duration would depend based on the Rose Valor test. I don't know what is this. Okay, just a second. So hydroxychloroquine, sulfasalazine, and one more is leflunomide. But again, this is contraindicated in pregnancy and lactation. Okay, so only these drugs are used practically: methotrexate, sulfasalazine, hydroxychloroquine, and leflunomide. Fine. Now, if a patient is not controlled on 3 DMARDs, then you give them biologicals. Okay, if a patient is not controlled on 3 DMARDs then you give them biologicals. Fine? Sorry. Yes, DMARD is disease modifying. It is disease modifying anti-rheumatoid drugs. Anti-rheumatoid drugs. Okay, this is disease modifying anti-rheumatoid drugs. Okay, so disease modifying anti-rheumatoid drugs is DMARD. Disease modifying anti-rheumatoid drugs is DMARD. Okay, this is disease modifying anti-rheumatoid drugs. I'm so sorry for that. Okay, so yes, if 3 DMARDs cannot control your rheumatoid arthritis, then you start them on biologicals. Okay, and the best biological that is to be given is anti-TNF. Anti-TNF, which is your infliximab. The best one which is used is infliximab. Okay, there are various others which are used, but most important is infliximab. Most important is infliximab. Now, what is the most important precaution that you need to take before you start a patient on biologicals? Most important. कर्सर आ रहा है कुछ. Okay. Yes, most important precaution that you need to take before you start biologicals is to rule out latent infections like tuberculosis. Fine. Fungal infections and other things. And what is the role of steroids? Steroids are given in low doses for a short duration during flares. Okay, so low dose short duration of steroid is given during flare of the disease. Fine. Now, what are the deformities which are seen in rheumatoid arthritis? Yes. Swan neck deformity, Bottonaire's deformity, piano key movement, Z-line deformity. Okay, so bridge therapy, hammer. Yes, steroids may be used as a bridge therapy. Swan neck deformity. Swan neck deformity is hyperextension of PIP with flexion of DIP. What is Bottonaire's deformity? Bottonaire's deformity is opposite of this, that is flexion of PIP with hyperextension of DIP. Okay, so this is opposite of this. Then we have Z-line deformity, which is subluxation of first metacarpophalangeal joint with hyperextension of first interphalangeal joint. Okay, hyperextension of first interphalangeal joint. I don't know about bridge therapy. I guess steroids are the one which are used for bridge therapy. I think you guys know it better than me. So subluxation of distal ulna, subluxation of distal ulna is given the name of piano key movement. Piano key movement is given the name of piano key movement. So subluxation of distal ulna is given the name of piano key movement. Now what is Jacquard's arthropathy? Jacquard's arthropathy. What is Jacquard's arthropathy? Yes? Yes, Jacquard's arthropathy. Yes. It is a deformity. It is a reversible joint deformity, which comprises feature of ulnar deviation of second to fifth finger, subluxation of metacarpophalangeal joint, swan neck deformity, Bottine's deformity. This is a combination of all these things. Okay, reversible joint deformity, reversible, very important. This is reversible joint deformity characterized by ulnar deviation of second to fifth finger, subluxation of metacarpophalangeal joint, swan neck deformity, Bottine's deformity. Classically, this was described in patients of rheumatic fever. Classically, this was described in patients with rheumatic fever. But now the incidence of rheumatic fever is less and it is more often seen in patients with SLE. Okay, but this deformity is not due to erosions. In patients of RA, the deformities are because of the erosions. In patients of SLE, the deformity like this which is seen is because of the laxicity of the tendon. It is because of the laxicity of the tendon and not because of the deformities. Okay, so SLE, this Jacquard's arthropathy, is a reversible deformity because of the laxicity of the tendon. Okay, is this clear to all of you? So just remember, reversible deformity in SLE because of laxity of tendon. And some say if a patient of SLE has erosion, it is usually considered to be overlap of RA plus SLE. It is considered to be an overlap of RA and SLE. The name given is Rupus hand. The name given to this is Rupus hand. Okay, erosions in SLE, it may be an overlap of RA and SLE. It is given the name of Rupus hand. Okay, now what is seronegative arthritis? Seronegative arthritis is a group of diseases characterized by negative rheumatoid factor. Okay, so they are known as rheumatoid seronegative, and they all have in common association with HLA-B27. They all have a family history. They all have a young age of onset, usually less than 40 years. And the kind of arthritis that we see in these patients is usually asymmetrical oligoarthritis, in contrast to your rheumatoid arthritis, which was symmetrical and poly. Here it is asymmetrical and oligo. The next is axial skeletal involvement. Axial skeletal means your spine. What about axial skeletal involvement in a patient with rheumatoid arthritis? And sacroiliac joint. They are classically involved in patients with seronegative, sacroiliac joint and axial skeleton. What about rheumatoid arthritis? In rheumatoid arthritis, spine is characteristically spared except C1, C2 or the atlanto-axial joint. Except C1, C2, spine is classically spared in a patient with rheumatoid arthritis. Okay. Then another important thing is enthesitis. Enthesitis is inflammation of the tendon and surrounding structures, which is again a feature of seronegative, more common in males or females. Ankylosing spondylitis is more common in males, mostly. Specific type it is different. Like ankylosing spondylitis would be more common in males. Reactive would also be more common in males. Inflammatory bowel disease, I'm not sure. Psoriasis, I'm not sure. So I think males, it would be more common in males than females. Okay, most of the types are more common in males. Enthesitis. Okay, enthesitis may be a feature. Now, what are the examples of diseases which are associated with seronegative? Examples of diseases are ankylosing spondylitis, then psoriatic arthropathy, Reiter's or reactive arthritis, Reiter's or reactive arthritis, enteropathic, that is associated with inflammatory bowel disease, and juvenile rheumatoid arthritis. Juvenile rheumatoid arthritis. How many types of JRA are present? How many types of JRA are present? How many types of JRA are present? Juvenile rheumatoid arthritis. It is divided into three types: the polyarticular form, the polyarticular form, and that associated with systemic onset. That associated with systemic onset. The polyarticular form is further divided into two types: type 1 and type 2. Okay. Now, systemic onset, polyarticular type 1, type 2, they're all rheumatoid factor negative. But the polyarticular type is further divided into two subtypes: RA factor negative and RA factor positive. So most of them are RA factor negative. Only some patients of our polyarticular type can have positive rheumatoid factor, and overall they constitute hardly 10 to 15% patients of JRA. So only 10 to 15% patients of JRA will have positive rheumatoid factor. Rest are all seronegative. Rest are all seronegative. Are these slides visible? Yes or no? These slides are visible? Yes or no? Yes. Now, what about ankylosing spondylitis? Ankylosing spondylitis. Ankylosing spondylitis is classically seen in young males. Classically seen in young males who complain of low backache. Who complain of low backache. What is the investigation of choice? Investigation of choice for ankylosing spondylitis. Investigation of choice for ankylosing spondylitis. Yes? Investigation of choice for ankylosing spondylitis. Yes, MRI of the dorsolumbar spine, which would show you at the earliest edema. It is not the X-ray. X-ray may be the initial. It is the initial investigation which is done, but the investigation of choice remains MRI, is MRI. Now, some clinical tests are done to look for the restricted movement of your dorsolumbar spine. One test is the Schober's test. Schober's test, which looks for the hyper extension or the extensibility, extensibility of dorsolumbar spine on bending forward, It is less than 5 centimeters in patients with ankylosing spondylitis. Okay, next is chest expansion. This is also decreased in patient with ankylosing spondylitis, less than 5 centimeters, chest expansion. Now in X-ray, you see squaring of the vertebra and the straightening of the vertebra, which is given the name of the spinal column, which is given the name of bamboo spine. Okay, so bamboo spine, squaring of the vertebra are seen on the X-ray. Okay. Finally, what is the treatment? The best treatment of choice is usually NSAIDs. For seronegative, NSAIDs are usually the treatment of choice, and the best is indomethacin. Indomethacin. But in ankylosing spondylitis, now even infliximab is approved. It has revolutionized the treatment of dripping wax appearance, and some other things are also seen in patients with ankylosing spondylitis. Let me just see this. Yes. Now after this, indomethacin is usually started in a dose of 25 milligrams. You can give up to 75 milligrams of sustained release of indomethacin. Fine. Now, reactive arthritis or Reiter's arthritis. Reiter's or reactive arthritis. Now, this Reiter's or reactive arthritis is classically seen after infections of some organisms. 1 to 4 weeks later, the patient complains of joint pain. After 1 to 4 week of infection by some organisms, and the organisms are either some GI tract organisms or some genitourinary tract infections. The GI tract infections are Shigella, Salmonella, some others, these are the common ones, and in genitourinary tract it is the Chlamydia. Most common is Shigella. Some books say Salmonella, but according to Harrison, the most common is Shigella, which is the most common organism. Okay, so there's usually a history of travel to the endemic zones. History of travel to the endemic zones followed by swelling of the joint. So Salmonella, Shigella, and Chlamydia. And what is the classical triad of Reiter's? The classical triad of Reiter's disease is arthritis, then eye involvement, which can be in the form of uveitis or conjunctivitis, and then last is genital involvement, which is in the form of urethritis in the males and cervicitis in the females. So this is the classical triad: arthritis, eye involvement, and genital involvement. Other than this, they can have other features like oral ulcers, then genital ulcers involving the glands, which is given the name of cercinate balanitis. Cercinate balanitis. They are vesicles on the glands which rupture to leave behind a ulcer. Okay, so cercinate balanitis is vesicles on the glands which rupture to leave behind ulcers. Then another features that they can have is inflammation of the digits, because of which the appearance of digits is given the name of sausage ductitis. Sausage ductitis. Sausage ductitis. Okay, so this is given the name of sausage ductitis. Cercinate balanitis, sausage ductitis. Then keratoderma melanoregica. keratoderma bilanoregica is also a feature of Reiter's or reactive arthritis. Keratoderma bilanoregica is hyperkeratotic presence of hyperkeratotic lesions, hyperkeratotic lesions on the palms and soles which heal by crusting. Okay, so hyperkeratotic lesions on the palms and soles which are associated with crusting when they heal. Okay, so this is keratoderma bilanoregica. Then they can also be associated with conduction defects of the heart. Conduction defects in the heart. They can have some pulmonary infiltrates also. So it is a multi-system disease. Fine. So now just few words regarding psoriatic arthropathy. In psoriatic arthropathy, first important point is that it can have two kinds of presentation. It can have asymmetrical oligoarthritis like your seronegative, but it can also have symmetrical polyarthritis like your rheumatoid arthritis. So it can have both the presentations. More common is asymmetrical, gout also we will discuss, don't worry. Most common is asymmetrical oligo, but they can also have symmetrical polyarthritis. Okay, a question was asked. Psoriasis is associated with asymmetrical oligo, symmetrical poly, both, none. So the answer was both. So both the patterns can be seen. One important complication in psoriasis is telescoping of the fingers, which is actually because of the collapse of the soft tissues of the fingers, and it is also given the name of arthritis mutilans. Okay, arthritis mutilans is collapse of the soft tissues of fingers, telescoping of the fingers. Okay, so arthritis mutilans. Now, if I summarize the joint involvement in RA, psoriasis and osteoarthritis. Osteoarthritis, RA and psoriasis. The joints which are involved and joints which are classically spared. So in osteoarthritis, the most common joint to be involved is distal interphalangeal joint, which is the Herbidon nodes, then followed by first carpometacarpal joint, that is the base of the thumb. So most common is DIP, which is the Herbidon nodes, then first carpometacarpal joint, then PIP, which is the Bouchard nodes. Okay, so in osteoarthritis you can have DIP inflammation, which is in the form of Herbidon nodes, then you have Bouchard nodes which are present in the PIP. Okay, so most common is DIP, then first carpometacarpal, that is base of the thumb, then PIP. And which joint is not involved in osteoarthritis? The metacarpophalangeal joint and the wrist joint. When we talk of RA would involve PIP, metacarpophalangeal joint and wrist joint. And what is classically spared? The DIP. And when we talk of psoriasis, any of these can be involved and none is spared. None is spared. Okay. So when we talk of psoriasis, none is spared. Any of the joints can be involved. So osteoarthritis is Herbeden nodes, Bouchard nodes, and first carpometacarpal joint. Now, lastly, gout, the crystal arthropathy, gout. Okay, so all of you have written it down. Can I change? Yes, so gout, what is gout? So gout is a crystal associated arthropathy which is associated with deposition of monosodium urate crystals in the joint. And this monosodium urate crystals, they are associated with the formation of tophi. The monosodium urate crystal, when they deposit in the joints, they cause the tophi formation. The most common site for tophi formation is the great toe, followed by the ankle joint. The other sites for this tophi deposition is subcutaneous tissues, in the tendons, ligaments, but never in the muscles. Never in the muscles. It can also be deposited in the visceral organs like kidneys. In kidney also you can have deposition of this, which leads to urate nephropathy. Urate nephropathy. Okay. Now, at the time of acute gout, gout is seen in patients who have hyperuricemia. But at the time of acute gout, the uric acid levels in the blood can be low, can be normal, can be high, and most commonly they are low. The uric acid levels in the patient with acute gout are mostly low, and the reason for this is that the cytokines which are released during acute gout, they have a uricosuric action, so the uric acid levels are low. Another thing is, it is the change in the uric acid levels which is important. So even when you give uric acid lowering drugs, it can lead to acute gout precipitation. That is why allopurinol is not indicated in patients with acute gout. Okay, because sudden change in the uric acid levels can also precipitate tophi and acute gout. Okay. Now, what is the most important diagnostic modality for gout? The most important diagnostic modality for gout is the polarizing light microscopy, and in polarizing light microscopy, the monosodium urate crystals, they appear negatively birefringent. And this helps in differentiating gout from pseudo gout, in which there is deposition of calcium pyrophosphate crystals which are positively birefringent. Okay, so monosodium urate crystals, they are negatively birefringent, calcium pyrophosphate are positively birefringent, so polarizing light microscopy would differentiate between them. Okay, this is the most important difference. Otherwise, earlier it was thought to be a gout only. Okay, later on when we discovered polarizing light microscopy, that only differentiated between them because the component of the deposits are different. Here it is monosodium urate, there it is calcium pyrophosphate. Otherwise they are same. Okay. Now next is when a patient is having chronic gout. In the X-ray, which sign can be seen? Radiologically, which sign can be seen in a patient with chronic gout? Yes, chronic gout, which sign can be seen? They can have the Martel's or the G sign. Martel's or G sign is a punched out lesion with overhanging margins. Punched out lesion with overhanging margins. Okay. So punched out lesions with overhanging margins, this is usually seen in chronic gout when the tophi has dissolved and a punched out lesion is left behind. Okay. Finally, when we treat gout, treatment of gout, if it is acute gout, the treatment of acute gout is NSAIDs except aspirin, because aspirin decreases the excretion of uric acid. So it is NSAIDs which is the treatment of choice. Other drugs which can be given are colchicine and steroids. These three things are given in acute gout. When we talk of the chronic gout, first group can be drugs which increase uric acid excretion, that is probenecid, or drugs which decrease the uric acid production, that is your xanthine oxidase inhibitors, the smartles of G sign is seen on X-ray inhibitors like allopurinol and febuxostat. Fine. And lastly, you have uricase enzymes. This uricase enzyme is normally present in the lower animals, not in human beings. It would convert uric acid into water soluble allantoin. So from the lower animals, we collect this enzyme and we give it to the individuals with hyperuricemia so that the uric acid is converted into water soluble allantoin. And the examples are rasburicase and pegloticase. And this gout is more common in males or females? Gout is more common in males compared to the females. It is much more common in males. Okay, it is not common in females. It is much more common in males. 20:1 is the ratio, especially the people who have alcohol. Okay. Pegloticase and rasburicase. So rasburicase and pegloticase, rasburicase. I think that would be all. So best of luck to all of you. I would give you my email ID. So if at all you have any problem, you mail me. If I would have the answer, so I would just reply you back. Okay. So DrRupaliMalik, Dr. Rupali Malik @gmail.com. So this is DrRupaliMalik. Fine. So Dr. Rupali Malik @gmail.com. Yes. So thank you all of you. If you are really saying that I was awesome. So thank you for that. So this is my email ID. If at all you have any problem, dialysis. I did not tell anything about dialysis, just the indications of dialysis. Indications of dialysis like hyperkalemia, uremic NCAF, volume overload leading to pulmonary edema, or CHF. Okay, blood urea levels more than 100. They are the indications for giving. They are the indications of giving dialysis. Then lastly, there are two types, the hemodialysis and the peritoneal dialysis. Hemodialysis, the most important complication is hypotension. Others can be electrolyte imbalance leading to cramps. For peritoneal dialysis, it is peritonitis, hypoproteinemia, etc. Okay. Fine. So thank you all of you and good night and best of luck.